Good morning. Good morning. Ridhima, are you there?
Yes, hi Saurabh.
Yeah, hi. Good morning.
Good morning.
Yeah, hi. I have another talk in the same this thing in Hall B.
Okay.
So can I start this session?
Yes, sure.
Because
No problem.
there also I have a third talk, so it will clash with the time.
Yeah, sure.
Yeah. So we'll start at 9 o'clock sharp.
Yes, we'll start.
Okay.
Just a second.
Good morning, Saili.
Good morning.
Good morning. Dr. Naina is here.
I'll just call her and ask her.
Yeah, sure. Saili, a request. I will start the session because I have another talk during the same time in another hall.
Oh, okay. No
So
problem.
I will just finish off my talk and then I have to change the, I think, Zoom meeting then.
Okay, sure.
Veena,
Good morning, ma'am.
Good morning.
So, ma'am, should we start the session?
Yes, please, I think it's the right time.
Good morning, everyone. So, this is, sharp at 9 o'clock, the first session in Hall C in this virtual MOSCON conference, one of its kind. So, we're going to start this session by welcoming our chairman, Dr. Naina Poddar, and we're going to invite Dr. Saurabh Patwardhan to present his first talk with all of us today.
Thank you very much. Should I start sharing, Dr. Naina, madam?
Yes,
Can I
please.
start sharing?
Yes,
Yeah.
all the best.
Thank you. So I'll be speaking about the triphobic and trifocal IOLs. This is a sponsored talk by Care Group. I've been using their IOLs for last many years, and this particular IOL I've been using for last few years. So I'll be sharing some technical aspects. Is it visible? Should I start?
Yes, Saurabh.
Yeah. In this presentation, I'll be speaking about the Triphobic HD IOL by Care Group and the magic of trifocal. Let's begin with how the multifocals really work. You can see this particular picture and you can see different images. You can see a large old man's head there, and also you can see the two persons, one lady and one old man standing there in front of that big man's head. And what happens is that you cannot see the large head and these two persons simultaneously. You know that it exists there, but still your brain shifts from one large image to smaller image when it wants to focus on one particular thing. And this is exactly how the multifocals work. So our brain selects what we want to see through a particular image. And in monofocal, because there is only one image which is falling on your fovea, you can see only at particular distance but no other image from other distances. So every multifocal is different from another, and the most important difference is the near addition. Before the trifocals came into picture, we just had the option of selecting one addition for that particular patient. So it always depended on the working distance of the patient. So if, for example, if the patient wants to work at 40 centimeters, the multifocal must have plus 3.5 or more addition. But if patient wants more of a intermediate vision, then patient needs to have 2.5 or less addition. And this is going to create a lot of trouble because we have to actually select a particular IL for a particular work. And then we had to also mix and match two different IOLs so that the patient can get different range of the vision. Also with the IL power itself, the addition, effective addition used to change. So for example, in high myopic eyes, if you choose a lower addition IOL, it is not going to work. But now I don't have to think about all these additions and different things, whether that patient will be suitable for this particular IL or not. And thanks to the trifocals, because trifocals are going to give us a full range of vision, not just far and near, but also the intermediate vision. And these triphobic and tridiff IOLs have essentially zero spherical aberrations. These are pupil independent IOLs based on diffractive refractive design, and it's a synchronized cast molded lens hydrophobic IOL. This is the triphobic IOL with its diffractive rings which are there, and these rings are peculiar because it has a additional alternate ring which is for the intermediate addition. It has a patented design with decreasing steps height and increasing angulation from center to edge, which gives us excellent light distribution for both near, far as well as for the intermediate, and also reduces the glares and halos. This is a typical defocus curve of a triphobic. As you can see that there are three different hills which indicate the focus. Here also same thing is depicted And this is the actual defocus curve which I found with the IDIF plus, which is a multifocal or bifocal I would say, and the TriDiff, which is a trifocal. As you can see in the TriDiff or trifocal, there is good vision maintained over different distances all throughout, right from 40 centimeters to infinity. Good range of vision or good quality of vision is maintained, while with the bifocal, as you can see, there is peak at the near at around 40 centimeters, but then there is no peak in between, so it has a very less range of vision. So in triphobic, basically 50% of the light energy is distributed for distance, 30% is for near, while 20% is for the intermediate. And the great thing about trifocal as compared to multifocal is that the total loss of light is less than 10%. The modular transfer function is much higher than what is minimum guideline. For example, for distance vision, the MTF minimum should be 0.43, but it is 0.60 in triphobic, while for near it should be at least 0.30, but it is zero point 0.5 for near, thus giving a excellent contrast sensitivity. Another good thing about this IOL is that you can get the actual power of IOL on the lens, and which I find quite useful, because for example in this case the 22 diopter IOL can be anywhere between 21.75 to 22.25. So if we have the actual power of the IOL, we can try to get as closer to the emmetropic IOL power of the particular patient. For example, if patient has emmetropic IOL power of 22.15, we can try to get something closer to that. Now, how the triphobic and tridiff has changed my practice? I feel that these IOLs are much better adapted by patients. There are very less glares, halos, and very less loss of contrast in these patients. Most of these patients are extremely happy after the surgery. In my practice, 15% of patients opt for trifocal IOL, and they have very predictable refractive results because they are based on the previous technology. Like for example, triphobic is based on acryolicy, tridiff is based on the IDIF plus or the hydrophilic platform of the lens, and we have already achieved very predictable refractive results in these IOLs. In my experience of 737 implants in last 3 years, 97% patients I have are spectacle free. They don't need glasses for any of the distance, and that's the great point about this IOL is that no patient has so far complained of any waxy vision or loss of contrast, which I used to get in some previous versions of the IOLs. The multifocal segment percentage in my practice has definitely increased with the introduction of trifocals. I think that is because of both the things, that because patients are now referring these patients back to us, and also because the surgeon's confidence in these IOLs has increased, which is which has resulted in increased number of patients going for these IOLs. And what I see in my, this was the case done I think 3-4 years back, one of my initial cases of trifocals. And what I see here is that they have got excellent range of vision. You can see the distance is 6 parts, the near is N6, and even the intermediate is 20 by 25, 20 by 16. And when I checked them on the contrast sensitivity charting, it was almost equal to a monofocal, almost equivalent, slightly less, but I think great contrast for a multifocal IOL and with great range of vision. Again, another patient similar to that, you can see the great contrast, 1.95, almost closer to what a monofocal would have, and that was the reason for very happy or satisfied patients that I had. So I think the future of cataract surgery is definitely refractive cataract surgery. And you have to really think, are you lagging behind if you are not going for these trifocal IOLs? So take home advice is that think of every patient as a refractive patient, improve biometry, optimize the constant and try to achieve better result. Work on the base monofocal lens, and earlier you start with trifocals, better you will be more predictable with the results and patient will be extremely happy. Thank you so much. Thank you very much. That was all from my side.
Very nice presentation, Saurabh. Simplified.
Thank you.
Who's the next speaker now?
I would like to invite Dr. Saili Lahane to present her talk on new winds of change, chandeliers and vitreoretinal surgery. Saili, if you can please.
Yeah, good morning. Okay, so can you all see the screen?
Yes.
Okay. So, good morning, everyone. So today I'll be talking about chandelier illumination in modern vitreous surgery. Um, as we know, there are types of illumination as the focal illumination, which we use routinely, that is endo illuminator or the light pipe, and diffuse illumination system is the chandelier system. The other ones which are available are slit lamp attachment for the operating microscope. Now, chandelier lighting system, it is a stationary and diffuse form of endo illumination from an anterior location, and this is available in two types. Like one is a single fiber system, which is used more commonly, and a two fiber system. What are the advantages of chandelier lighting? Basically, as it is self-retaining, so it frees up surgeon's hand, allowing true bimanual manipulation during the surgery. Surgeon can perform scleral indentation and achieve more controlled and smooth peripheral base shaving. It improves anterior chamber visualization for phaco-vitrectomy by using retroillumination, and theoretically it reduces the retinal phototoxicity because it is little away from the retina. Now, this is the first video wherein it is a sutured vitrectomy. It's an old case. The infusion cannula is having illumination so that bimanual dissection can be done in a case of PDR. For bimanual dissection, scissor and a forceps is being used. Because we are using a stationary illumination, we can use both the hands and do the bimanual dissection, especially in difficult PDR cases. This is another case. This is a MIVS, wherein again for a PDR, bimanual dissection is being done with the help of chandelier illumination. Now what happens when we are using chandelier is the shadows of the instrument actually comes and it hinders our dissections sometimes because the position of the chandelier is fixed, it is stationary. As we move the light pipe, it cannot be moved. So what I've done in this case is made two ports for the chandelier, one is superior, one is inferior, so that as and when required, the chandelier illumination can be changed from one position to the other position, so that it helps in dissection of the membranes and the shadowing is lesser. So this is as if a dynamic chandelier is being used because we are using it from two ports. Now this is another video where I've used chandelier for scleral buccal surgery. Basically, scleral buccal is an extraocular surgery wherein we don't make any entry inside the eye, but here I'm using a chandelier. So what we can do is, if we know where is the break, we can just put the chandelier opening exactly opposite in the opposite quadrant of the break, so that it becomes easier for the manipulation. Now with the chandelier, the chandelier illumination, we can use the wide fundus viewing system, and we can see the break, visualize the break. Do cryo, as you can see here, the cryo is being done. This is a highly myopic fundus, that's why the visualization is not very, the video visualization is not very good. But this is the cryo being done to the break. This is external drainage with the help of laser and scleral cut down. So the whole surgery, the whole scleral buccal surgery can be done under the microscope without using indirect ophthalmoscopy, without getting up, changing and seeing the fundus again. Again, at the end of the surgery, we can check the buccal effect with the help of chandelier through wide angle fundus viewing systems. So chandelier in the scleral buccal surgery, it offers wide, excellent visibility of the fundus. The whole surgical procedure can be done viewing through the microscope. It identifies preoperatively unidentified or unrecognized tears during the surgery. Scleral indentation can be performed by the surgeon. Visualization is excellent, especially in cases of non-dilating pupil, posterior capsular opacification, or early cataract. It precisely locates all the retinal breaks. It helps in undertaking cryopixy of the break, drainage of the subretinal fluid at the site wherein the height of detachment is maximum, and we can titrate the buccal height also. This is another video. First, right now, the light pipe is being used and vitrectomy is done for the vitreous hemorrhage. This is a case of intraocular foreign body. You can see the foreign body and retinal detachment. So now, after putting PFCL so that the posterior retina is not damaged for this. Now I have changed the illumination to chandelier, and then with bimanual using two instruments, I'm just trying to take out the intraocular foreign body. That's a break because of the foreign body. The sclerotomy is extended. Now with the help of intraocular forceps and the magnet, the foreign body is being taken out. So it becomes a little easy because both the hands are available for manipulation of the foreign body. Reducing the traumatization, we can remove the foreign body by using both the hands. Manipulation becomes a little easier with the help of chandelier. So in some cases of intraocular foreign body, definitely chandeliers can be used. Where else we can use chandelier? Chandelier illumination and bimanual vitrectomy can be used to remove dislocated IOLs. A proper bimanual vitrectomy can be done wherein we can hold the IOL with one hand using a forceps, and then with the other hand vitrectomy probe can be used to do the vitrectomy. Two forceps can be used to hold the lens and perform the handshake technique. It's also used in cases of removal of intraocular foreign body. What are the disadvantages? Basically, we are making one more sclerotomy. So bacterial inoculation into the vitreous cavity, vitreous incarceration at the site of sclerotomies. Repositioning of the fiber is often necessary to complete 360-degree peripheral manipulations. It needs at least one extra sclerotomy, which entails the typical risk of creating any sclerotomy, including leakage, hypotony, or infections. When we are using single fiber chandelier system, there is shadowing by the instruments which may interfere with the surgical field. In summary, chandelier helps surgeon do bimanual manipulations in difficult cases of diabetic PRD and PVR cases. It may reduce theoretically the risk of retinal phototoxicity because of the distance from the retina is more. It makes the life of ophthalmic surgeon easy with little additional burden. Thank you so much for this opportunity for presenting the talk. Sorry, ma'am, I cannot hear you.
It was a good presentation. Videos were good, simplified. Thank you.
Thank you, ma'am.
I would like to invite Dr. Hitendra Mehta to present his talk, please. So are you here? I don't think he's here.
Can we take the next speaker?
Yes, ma'am. So I will present my talk, which is next, and I am going to present, ma'am, is the screen visible?
Yes, please go ahead.
Okay, so I'm going to speak about the Biotech Icryl Phakic Intraocular Lens. I do not have any financial interest in this product. To start off with, refractive procedures are of two kinds. The first kind are the corneal laser procedures like LASIK, SMILE, surface ablation, and the second category are the lens-based procedures. The first common one that we're aware of is a refractive lens exchange, and the second one is a phakic intraocular lens. So what is a phakic intraocular lens? We are not replacing the natural crystalline lens of the eye. Hence, it is called a phakic IOL. We are adding an additional lens where the natural crystalline in a phakic eye, and hence it's called a phakic IOL. So when do we use phakic intraocular lenses? When the patient is not suitable for a corneal laser procedure. And what could be the causes for that? First is a very high refractive error beyond the limits of a corneal laser procedure, or if the corneal thickness is not good enough to correct the error, or if there is a suspicious topography where it's not safe to do a surface corneal laser procedure, or if the predicted post-operative keratometry is going to be flatter than 35 diopters or steeper than 50 diopters, where the cornea will be optically compromised after the procedure. In such cases, we will opt for a phakic intraocular lens. It is a good and preferred option in very high refractive errors. And the causes of this are, it is a more predictable procedure than a corneal laser procedure, wherein if we are correcting like a 9 diopter of error, we are most likely to undercorrect it slightly or even a high cylinder, whereas phakic IOLs are going to give you a more predictable and complete correction. Also, this correction is less likely to go into regression like a corneal laser procedure. So the refraction and correction is more likely to be stable. Also, it is a potentially reversible procedure, which is a very important and good selling point for the procedure. Versus a refractive lens exchange, this procedure offers the advantage of preserving accommodation, which is very important in young patients. Hence, it's going to preserve accommodation, and your cornea is not optically compromised, which can happen in very high refractive errors. The patient may end up with aberrations that are induced by the laser procedure. So this is a very good option in high refractive errors. So what inclusion criteria do we need to keep in mind? That the refraction needs to be stable, not more than 0.5 diopter change in a year. The age of the patient needs to be at least 21 years completed. The anterior chamber depth is the parameter that you need to look for on your topography or biometry to see that it is at least 2.8 millimeters before we go ahead and plan the procedure. The eye needs to have a good healthy endothelial count, open angles. The iris and pupil configuration needs to be normal before we plan for a phakic IOL. The contraindications would be any other comorbidity in the eye, like a corneal pathology or a cataract, uveitis, glaucoma, lens problems. So any other comorbidity in the eye would rule out the possibility of proceeding with this. It can also be used, these are off-label uses, but I have used it in a post-LASIK or post-PRK error, or in a stable keratoconus, in an early keratoconus where the quality of vision is good with spectacles. You can go ahead and correct the error with a phakic intraocular lens, but the keratoconus needs to be stable, or it should have been stabilized with cross-linking at least a year prior. It can also be used to correct a refractive error left behind after a cataract or a penetrating keratoplasty, but these are all off-label uses. So coming to the Icryl phakic IOL that we are discussing today, it's a plate haptic lens that you can see in this image. It is a hydrophilic acrylic lens, and it has a central hole, which is a very important feature so that you don't have to do a iridotomy or iridectomy at the time of the procedure. There are two orientation marks, and the orientation mark needs to be on the left side of the leading haptic and on the right in the trailing haptic. It has a large 6.5 millimeter optic, and it is available in 4 sizes from 12, 12.5, 13 and 13.5 millimeters. It can be used to correct up to minus 23 diopters of myopia. And there is also a toric version available in like 0.5 diopter steps, which will have orientation marks, and the usually not more than 10 to 15 degrees of rotation is required after implanting the IOL in the sulcus. So before planning for surgery, we need to get a good refraction of the patient and keratometry readings. Other than this, the two other important parameters are the anterior chamber depth that has to be measured from the endothelium, and the white to white diameter that determines the sizing of the lens. So anterior chamber depth can be measured from any topography that you're using or the optical biometer, but the optical biometer usually gives the anterior chamber depth from the corneal epithelium, and then the corneal thickness needs to be deducted from it so that you can use the ACD from the endothelium. Even imaging devices like the UBM or an anterior segment OCT can aid in giving us a good anterior chamber depth measurement. These imaging devices not just help us with the anterior chamber depth measurement, but also the iris configuration and any other pathologies that may be visible here. So it's a good idea to do this before planning for the procedure if one has access towards it. The important part in an ICL, I think everybody knows this, is the sizing. So we are placing this phakic intraocular lens in the sulcus, and the only instrument that can actually help us in measuring the actual sulcus diameter is the UBM. However, the UBM is highly operator dependent. It's not a very objective measurement. You need very highly trained staff to be able to give like a very nice horizontally balanced, vertically aligned image, and still the readings can be variable. So what commonly is used is the white to white distance. This can be used with the help of the most reliable measurement is using a caliper. If not the vernier, the more accurate measurement is a digital caliper as seen in the figure here. And we need to measure the white to white distance. So typically I would take 3 readings with the patient in the supine position, and it is not the brown to brown distance. So you make sure that you're just beyond that measurement, and you'll get an accurate white to white measurement reading with a caliper. If not, we can use a topography that can also give us not just automated readings, but even manually on the iris image, one can use a caliper and measure the horizontal white to white distance, or then an optical biometer, but usually it overestimates the white to white measurement by at least 0.5 millimeters. My suggestion would be to use a combination of all of these, whatever is accessible to you. More the readings, the more chance that you're not going to go wrong with the measurement. And then make sure that there's not too much difference in the two eyes when you're ordering the lens. So we are assuming here that there is a parallel pattern between a white to white measurement and the sulcus to sulcus. This is true in Caucasian eyes, but not in all races. So in Oriental population, the sulcus to sulcus tends to be much larger than the white to white distance, while in Indians, the sulcus tends to be much shorter than the white to white that we measure. So low vault is not that common, but high vault can occasionally be encountered in our population. So what is vault? Vault is the distance between the posterior surface of the phakic IOL and the anterior surface of our natural crystalline lens. This can be easily measured at the slit lamp. If you compare the corneal thickness, the vault, this distance should be between half to 1.5 corneal thickness in length. If it's more than 1000, it's called a high vault, and we can end up with a narrow crowded angle or glaucoma, whereas a low vault is when it's less than 125 microns, and then lens touch is common or anterior cataracts can happen, and toric IOLs with a low vault can rotate in the axis. Hence, sizing is very important to avoid these complications. I'm going to show you a small video of the procedure of the Biotech Icryl phakic IOL. So this is a toric version, hence I'm doing the axis marking here. The surgery is usually done under topical anesthesia with a temporal incision. So after the side port entry, I'm putting in some Oculan, and HPMC visco is the only viscoelastic that should be used at the time of the procedure so that it's easy to remove at the end of the surgery. The loading is very easy for this lens. As you can see, it's similar to a regular foldable IOL that we use. It's the same cartridge. Visco is put. Make sure that the edges of the lens are tucked in neatly at the side, and then just press the lens down and fold the cartridge. Through a temporal 2.8 millimeter wound, your hand has to be directed horizontally, and make sure that you keep an eye on the orientation marks. The left side orientation of the leading haptic, and then put some viscoelastic over it. So now you know that the lens is convex surface up. looking at the orientation marks. And then with a special instrument, each haptic, each of the 4 is tucked behind the iris one by one, and do not touch the central optic. As you can see, the lens is now being aligned with the marks. At the end of the surgery, just make sure that you do a good irrigation aspiration, wounds are hydrated, and no iridotomy is necessary for this lens. So these are my results, wherein at 1 year post-op, 95% patients were 6/9 or better after the procedure, and the safety was excellent. No patient lost a line. In fact, typically with high myopia, there can be a gain in a line or two of your best corrected vision, because with thick glasses, usually there's quite a bit of aberrations, and the vision tends to usually improve after a phakic IOL. The predictability of the procedure was excellent. The average refractive error ranged from 0.09 to minus 0.21, and the predictability for the spherical equivalent shows that 80% patients were within 0.5 diopter of spherical equivalent, while 95% were within 1 diopter. Similarly, for the toric version, 60% patients were within 0.5 diopter, while 100% were within 1 diopter of targeted astigmatism. This last graph, the E1, shows the stability of the procedure from after the surgery to 6 months later. The refractive correction was maintained. So usual concerns can be the vault issues and halos and glare, but now with the central hole in most of these lenses, the risk of pupillary block or lens opacities is quite reduced, and I haven't encountered any with this lens so far. So why choose this particular lens? Because it has very good results. It is easy to load and implant, as you just saw. We are all familiar with the injection system, and it has a central hole, so you don't have to worry too much about pupillary block or doing an iridectomy at the time of the procedure. It has a large range of errors that can be corrected, and it is very stable in the sulcus. An online and offline app is available to calculate, and it's very easy to go ahead and order the lens. The company will support you with whatever calculations you may not be well versed with. So to conclude, phakic IOLs definitely have a wow factor, and it's a surgery that comes easily to cataract surgeons with excellent results. So I hope we can all offer this to our patients who come and are eligible for this procedure. Thank you very much for your attention.
Thank you, Dr. Ridhima. Excellent presentation, and thank you for the practical tips. Can we move on to
Thank you,
the
ma'am.
next speaker?
Hello. Is Dr. Hitendra going to be there for his talk on eccentrics?
So I'm not sure. I think you can present your talk, sir.
Okay. Right. Okay. I hope my slides are visible.
Yes, sir.
Yes, sir.
Okay, so it's really a pleasure for me to speak on the latest molecule on the block for neovascular AMD, which is brolucizumab, or it's called as Pagenex in India. It is basically a great molecule because I've described it over a period of time how it comes to. Now, when as a retinologist we are treating when we are treating a neovascular AMD, basically we are looking at two things: either to reduce the number of visits for the patient when it comes to the clinic. But if we do that, we chance that the activity might be missed and patients might lose visual acuity. And if we reduce the number of injections, we can have a suboptimal outcome of the vision. As you can see from this graph, whichever the country in the second year, as the number of the injection and the visit fall down, the visual acuity starts becoming lesser and lesser. So there are a lot of unmet needs even today, in spite of so many years of starting intravitreal injections. We want to give lesser injections. We want to reduce the number of visits for the patients. We want less activity with the amount of intraretinal fluid or the subretinal fluid becoming the least. And we want the macular thickness fluctuation to be the least as much as possible. And these are the basic challenges that we are having to maintain for the patients on neovascular AMD. And I think that's where the single chain antibody fragment of Pagenex would come very handy. And this particular molecule is the smallest functional unit of the antibody, and it's a really engineering marvel that it has been invented for neovascular AMD. I'll just take you through a very brief thing on it. So if you take a typical human monoclonal antibody, it has got two arms, the FC arm and the Y-shaped arms. These Y-shaped arms are basically made up of a single chain, heavy and light fragments. And its main active sites are these two sites here, which have got the complementarity determined regions, or the CDRs. So with the biotechnical technical engineering, what they have done is they have actually isolated only these ones from a human monoclonal antibody, and they have mounted a new CDR or a novel CDR which has got a higher affinity towards vascular endothelial growth factor. And that gives you a fantastically small molecule as compared to the other molecules that are available. Now, why a small molecule is very important? Look at the size of this is just 26 kilodaltons versus 50 kilodaltons of eccentrics or 100 kilodaltons of aflibercept. And the small size means you get more molecules per injection. So you can have a large number of molecules here, about 11 to 13, versus just a single molecule which can be there in aflibercept, or just about zero point 0.5 to zero point 0.6 in ranibizumab. So you can imagine that you can pack in more power with the same dose when you're using a smaller molecule. And a smaller molecule also means more penetrability, more potency, and more binding to the vascular endothelial growth factor. Now, this molecule has been very extensively studied, and it has gone through phase 1 to phase 2 to phase 3 trials. And these are the phase 3 trials that we're going to discuss, which are called as the Hawk and the Harrier trials. Now, these are the two parallel trials, which are the phase 3 multi-center randomized double mask trials that were done. And they are the two parallel trials which were done in different parts of the world. The Hawk trial was done in US and some sites all over the world, like in Israel, Japan, Australia, New Zealand, Latin America. Whereas the Harrier was done excluding US, it was in Europe and some parts of Asia. So you can see there are two parallel trials which are done in different parts of the world. And there were a total of 408 study sites for these trials. Each of these had about roughly 1,775 patients which were enrolled. And what was the basic point of taking these trials? One was to prove that it is not inferior. It is not inferior to aflibercept, which has been around for a long time for neovascular AMD. And it also wanted to say how the visual acuity improved, how the activity of the CNVM went off. And the activity can be made out by knowing intraretinal fluid, subretinal fluid, or the subretinal pigment epithelial fluid. And it wanted to know how the activity is controlled by getting these three fluids out. And that would amount to the change in the central thickness of the fovea. It wanted to see how long the injections can be prolonged to, whether we can maintain the patient on 3-monthly injection, and can we predict how many patients will remain on 3-monthly versus someone who will need more frequent injections. Now, this was the study design for this. Each patient was first given 3-monthly loading doses. And once the 3-monthly loading doses were given, at week 16, the patient was evaluated for disease activity. If the patient had a persistent disease activity, then the person who was on brolucizumab arm was shifted to every 12-week interval of for every 8 weekly dosage, just like aflibercept. But the person who was shifted to a 8-weekly injection remained on 8-weekly injection and was not shifted back to 12-weekly. So it's very simple to understand. The patient was given brolucizumab at every 12 weeks until and unless the disease activity was high at 16 weeks, whereas the aflibercept person remained on 8 weeks throughout. So how do you get the disease activity? Well, there were stringent criteria used at week 16 and a slightly less stringent criteria which was used on week 20, 32, and 44. So the basic criteria were whether the best corrected visual acuity has been lost by more than 5 letters, which carried out throughout. Whereas at week 16, they even saw the fluid and they left it to the investigator to decide whether the disease activity was there and whether there is a worsening. So this is how the study design comes in. You go and give it first 3 loading doses, evaluate at 16 weeks. If disease activity is there at 16 weeks, then you shift them to 8 weekly. If disease activity has 20, you shift them to 8 weekly, so on and so forth. And similar one was in the Harrier study. Now, there was one basic study we had to understand between Hawk and Harrier. In Hawk, they used brolucizumab 3 milligram, 6 milligram, and aflibercept to compare with each other. Whereas in the Harrier study, they used only 6 milligram of brolucizumab versus aflibercept to study. So that was the basic difference between both of them. So in Hawk, we had 3 arms. In Harrier, we had 2 arms to study. Now, what are the key findings? Let's go directly to the key findings. The best corrected visual acuity at week 96 versus aflibercept showed that brolucizumab was not inferior to aflibercept. So the primary endpoint was achieved. It showed that the brolucizumab arm was not inferior to aflibercept in terms of visual gain in both of the studies. In fact, even the 3 milligram brolucizumab did as well as aflibercept when you studied it at week 96. How about the people who got more than 15 letters or who got more base corrected visual acuity of more than 84 letters at week 48? And there you see that 33% of the patients of Hawk gained 15 letters or more at week 48 as compared to 25.4% in aflibercept group. So here it gives a slight edge to brolucizumab 6 milligram, not the 3 milligram, over aflibercept, where you can see there's a slightly more visual acuity gain as compared to the aflibercept arm in the Hawk study. Now, how about the macular thickness? This is where the basic difference came in. When you see the patients who were on brolucizumab, you can see there is a sustained sort of a decrease in the macular thickness versus the patients who were on aflibercept, where you had a seesaw-like a pattern between each injection. So you can say that between each injection, the macular thickness increased till you gave another injection where the macular thickness went down, as compared to the brolucizumab arm, which had a sustained decrease. And this sustained decrease, we know, leads to a more tighter control on the disease activity. It amounts to lesser fluid collecting between injection, and it amounts to lesser visual acuity damage that can happen between the injections. When you compare the intraretinal and subretinal fluid, we can see that the brolucizumab arm had about 35% patients who had less amount of intraretinal or subretinal fluid on OCT than patients who were on aflibercept. And similarly, if you see the Harrier trials, you will see about 35 to 40%. Now here the percentage is slightly more because you have to understand you're using 6 milligram here, whereas here you're using 3 and 6 milligrams both. Similarly, if you see subretinal fluid, you can see there is a superior anatomical outcome when you see at subretinal fluid as compared to aflibercept arm. So basically what it means is that brolucizumab gives you a better drying of the retina. It means a better amount of control on the disease activity. Even when you see the sub-RPE fluid, it proves the superiority of brolucizumab. So what you mean is that the intraretinal fluid is much lesser. And why that is important, because anatomical outcomes like this, where the fluid is lesser, means better vision. And that has been proven in many, many studies that any presence of intraretinal, subretinal, or pigmentoepithelial detachment has led to visual acuity loss or no visual acuity improvement. So if you get a better draining of the retina, it means that there's a better amount of visual acuity improvement. Now, can we predict how many of the persons who are put on 3 monthly injections will continue on so? So if 50% of the brolucizumab 6 milligram patients were maintained up to week 48 on 3 monthly injections, and that means that many lesser amount of injections to our patients. And if the patients completed up to week 48 with every 3 monthly injections, then up to the first 3 months, we know that 80% of the brolucizumab patients who completed the first injection interval remained on the 3 monthly injections up to week 48. So if you see that the first injection itself, you can put the patient on 3-monthly injection, then the chances of that person to remain on 3-monthly injections is about 80%. And that's a huge number of patients whom we can offer 3-monthly injection. Over 75% of the brolucizumab patients who completed week 48 continue to remain on 3-monthly injections up to week 96. So it means what? It means that lesser number of injections for the patients. We can confidently call the patient after every 3 months. So that means lesser number of visits. So that actually translates to the unmet needs of AMD, which was one of the first slides I showed. It means that what we want in treatment of neovascular AMD is lesser injections and lesser visits. And that's exactly what this brolucizumab will help us achieve. So in summary of this study, it proved that brolucizumab was not inferior to aflibercept. We had a better amount of visual acuity, a better amount of drying of the intraretinal subretinal fluid, a superior control on central retinal fluctuation of the thickness, and we can actually extend a dose now to 3 months. It was not possible with any molecules till today. Now, this is just to share my initial experience. This was a 67-year-old male who had a CNVM with intraretinal and subretinal fluid. And after giving injection Pagenax, within 10 days, you can see that most of the CNVM activity is gone and there is no intraretinal or subretinal fluid that is left. And it means that you have a safe and effective molecule, but you really need to rule out any intraocular inflammation prior to choosing this as your drug of choice. And why so is because there was an alert which was given by the American Society of Retina Specialists, which saw that after the brolucizumab injection was marketed in the United States, they reported about 11 cases on February 23, 2020 and on March 30, 2020 who had occlusive vasculitis. And this occlusive vasculitis was a result of intraocular inflammation, which was subsequently reported by many studies, which can happen on patients who get Pagenax. And because of this event, then Novartis went ahead and formed a safety review committee, which had neutral people who were global retina and uveitis specialists and imaging experts who could come to a conclusion as to why we are getting this sort of inflammations, and yet we are not yet very sure why these inflammations come in. However, they have cautioned that if you use it in patients with lesser amount of signs of inflammation, of previous signs of inflammation, then the incidences have come down. The information about this is on a website called brolucizumab.info, which gives you a live tracker of this amount of inflammations. And if you actually see them, it is just about 4.5 per 10,000 injections, which goes to 0.45 or 0.3 if you see retinal vascular occlusions. Now, if you take a 3 events total, it goes to about 0.136%, but we don't want even that to occur. And that's why it's very important to find out if there's an early inflammation, because if there's an early inflammation which becomes uncontrolled, that goes to vascular occlusion. And that's why before you inject Pagenax, you should see to it that there are no AC cells of flare. The anterior segment should not show any features of old uveitis. Even the posterior segment should not show any uveitis. So basically you have to rule out any prior inflammation in these eyes, and therefore you should even avoid it initially in one-eyed patients before you choose to give Pagenax, because these are the patients who have a higher chance of inflammation, and the incidence of these inflammations has come down after this advisory has been put into practice. So how do you start with this new molecule if someone wants to? Select a compliant patient who can report to you with any sort of ill effects or any symptoms that the patient will feel. Maybe it is redness, watering, or even slight amount of blurring of vision. They should immediately come in. You can definitely add poor responders to anti-VEGF to see how this molecule works. We have to counsel the patients properly, do a meticulous pre-checkup, and explain all warning symptoms to the patient in detail and evaluate the patient time to time to look out for inflammation. Going ahead, it's a fabulous molecule. It is already approved in more than 40 countries. They have priced it very well, and this is the MRP. It comes to much lesser, about 23,000 for the ophthalmologist. And this gives them a great opportunity to reduce the injection burden to about 10,000 rupees per month for the patient if you can manage to put on 3 monthly dosage. And that gives you a great of opportunity to shift to a totally approved drug rather than using the drugs which are not approved like Avastin or Zaltrap. It will give you a good amount of response, a better control with a better pricing for the patient. Thank you. Thank you very much.
Thank you, Dr. Nishikant Borse, for making the study also interesting, the hop and the harrier. And let us move on to the last speaker. I think last 10 minutes left.
Dr. Nikhil Beke, ma'am. Dr. Nikhil Beke to present a talk on eccentrics.
Fine. Okay. Please start.
Hello, am I audible, ma'am?
Yes,
Yes,
please,
Nikhil.
Dr. Nikhil.
Yeah, just a second. Is my screen audible? Is my screen visible?
Yeah. Can you expand it? Right.
Yeah. So would be addressing primarily the points of enigma of similarity. So do the RCTs translate into real world practice both for the innovative molecule, that is eccentrics, as well as the biosimilars which are now widely available in the Indian market? Right. So I think it would not be wrong to say that the medical practice changed for better, and there's no going back to the pre-anti-VEGF era since 2006 when ranibizumab got its approval. And the goalpost of medical retina treatment literally shifted from loss of prevention of loss of vision to betterment of vision. So this was for the first time that in seemingly incurable disorders like NAMD, that we started talking of improvement in vision, and that was a big thing that was credited to introduction of ranibizumab. So ranibizumab continues to enjoy a lot of evidence, a very robust evidence, and evidence of efficacy that is durable in the long term. And that's why the ranibizumab goes from strength to strength. That from its first approval in 2006 for neovascular AMD, now most recent approval is for retinopathy of prematurity and even proliferative diabetic retinopathy. So the ranibizumab has the largest number of clinical trials, the largest amount of real-world patient data, and it has the most extensive patient follow-up program. So this has been proven to have a good efficacy and safety both in clinical trials as well as in real world. So, but how do the randomized clinical trials differ from the real world study, and is there any real need of even studying real world data? So basically, the real world data needs to be studied because these are non-interventional and usually open label studies, because we want to what's seen in clinical what's seen in randomized clinical trials is it's difficult to employ the same protocol. It's difficult to employ difficult to get the same kind of compliance that the patients have in RCT. And the real world studies, then there are representative of more broader patient population. They have variable patient adherence and compliance protocols. And this assesses the performance in routine clinical practice. So the real world studies which are available, they conform more to your clinical practice. And these are in fact more useful in taking patient decisions rather than proving the drug's efficacy and so. But at the same time, it would not be wrong to point out that because these are open label studies, these are sort of most of them are retrospective studies. So they don't really conform to the tenets of research methodology, and they bring in the bias in the study. So the real world studies, so they better inform the treatment practices in routine clinical settings. So when it comes to making a choice for your patient, I think the real world studies, they are more informative, they are more useful, and they enrich the evidence from RCTs to optimize the patient outcome. It also informs and helps the decision makers such as healthcare professionals, payers, regulatory authorities such as insurance bodies. So this was the largest prospective observational study in medical retina, Luminous. So this was a global 5-year multicentral observational study across all the approved indications to evaluate long-term effectiveness, safety, and treatment patterns which were associated with ranibizumab usage, 0.5 milligram as it's available in India. So what was observed is for most of the indications, the real world data or the real world improvements, they were largely in line with the improvements which were seen in the clinical trials. And a curious observation that was seen for most of the indications, almost all of the indications, was the visual acuity gains were greater in the patients with lower baseline visual acuity. What it means is if your patient has poorer vision as compared to another with relatively better vision, your patient stands to gain more with the injections. And this is in line with, this what obviously has a bias such as ceiling effect of the drug, where a patient at 6'9" vision won't be able to improve as much as a patient with 660 vision. But even setting off all these biases, the drug seems to be helping more with patients with lesser visual acuity. And this stood true irrespective of the indication that we looked at, including the retinal vein occlusions, diabetic macular edema, and myopic CNVM. So the evidence for randomized control trials, they are in synchronization with real world evidence. So I think we can continue to trust ranibizumab with our clinical day-to-day clinical decision making. So our newest kid on the block was biosimilars, which was introduced in India about 5 years back. And before going into any discussion about biosimilars, we need to know what they are so that we can take an informed choice and we can conscientiously advise our patients regarding the choice between innovator molecule, that is Eccentrix, and the biosimilars. So the biosimilars, before going to biosimilars, we need to know what are biologics. So biologics are basically pharmaceutical products which are derived from or composed of a living organism. The example of latter is the vaccines, the live vaccine that we speak of. So the oral polio vaccine would be a biologic because it contains a living organism. On the other hand, more so in terms of medicine and specifically in terms of ophthalmology, these are products of a genetic engineering process, a bioengineered designer molecule that is oftentimes an antibody. And what biosimilars mean are they are highly similar versions to an existing FDA product biologic, which is referred to as innovator molecule or a reference product. So they should not have any clinically meaningful differences. The biosimilar should not have any clinically meaningful differences from the reference product in terms of quality, safety, and efficacy. This is how you define a biosimilar. But we can discount some minor differences in clinically inactive components. For example, this is an image which has been taken from one of the European regulatory authorities website, wherein you can see that the majority of the structures are similar, but one of the side chains, they are a little dissimilar. But as long as this side chain doesn't account for any kind of meaningful difference in terms of quality, safety, and efficacy, I think we can ignore it. And these two products still, the product on the right hand side still qualifies as a biosimilar product for the product on the right, on the left. So the biosimilars are typically manufactured by a different company when the patent and exclusivity expire on the original reference molecule. By definition, it has to have the same mechanism of action, the same approved indications, same routes of administration, the same dosage forms, and the same strength, as in the strength in terms of microgram or milligram that can be administered at one point in time, as that of the originator. So as we can easily see that the biosimilar manufacturers, they have a lot less degree of freedom than would be given to other molecules which are manufactured after the patent expires. So the facility, manufacturing, processing, packaging, and storing biosimilar must be of the similar quality of that of the originator. So this is in theory that should be the case. So let's see whether in practice such norms are followed. So a question that is routinely asked to me is, are biosimilars same as generic medications? Because the generic medications are the medications which get manufactured after the patent on the innovator molecule or the initial molecule expires. So there is a striking difference between the two, and it's important to understand that difference before we understand why biosimilars may or may not be similar in efficacy and quality and safety as that of the original or innovator molecule. So the generic drugs basically are very small molecules. The biosimilars, they are they're similar to a biologic entity. And these are rather large molecules. These are quite complex molecules. So the manufacturing process of a generic drug is a chemical process. So that can make predictably identical copies. The manufacturing process of a biosimilar is a specialized biological process, and it provides a similar copy, but the copy might not exactly be identical. The stability of a generic drug is very high. The stability of a biosimilar it might not be very stable because it's sensitive to storage and handling conditions. And unless you know them precisely to be of the same as that of the innovator molecule, the stability might not exactly be reproduced. These are also immunogenic, as in these are all proteins, despite of, other than being antibodies, these are proteins, and you can always have neutralizing antibodies to proteins. So your biosimilar molecule in lab might work as good as the or as well as the originator molecule, the innovator molecule. But in clinical practice, because of these neutralizing antibodies, it can face issues such as tachyphylaxis or no effect whatsoever. So the tests which are done for quality control, because it's a simple molecule, generic drugs require less than 50 tests, while the biosimilar being complex molecules require more than 250 tests. And approval requirements, these are relatively, getting an approval for a generic drug is relatively very easy. For a biosimilar, you require large clinical trials involving lots of patients before they are deemed similar to the innovator molecule. So this is what the theory justifies, this is what the theory dictates. Do we have such kind of robust literature or robust evidence to justify the use for biosimilar as a vis-a-vis the innovator molecule? Well, at least when it comes to the published research, we have a single randomized clinical trial, and that too, which has been done only in AMD patients. So this was a study which was carried over just 3 months, wherein in the real practice you require at least 1 year long study. It conformed to the older norm of prevention of vision loss, when the current studies actually evaluate best corrected visual acuity gains. So this product was deemed a success if it just helped maintain patient's vision, whereas the current studies require that you show some sort of improvement over the baseline, at least in a significant proportion of patients. So this study.
Can we wrap up, Nikhil?
Yeah, so this study basically was a very small number of patients as a required rather than having a lot of patients. So what we went in was a multicentric retrospective open label study from Disha Eye Center, and this was a comparison of innovator versus biosimilar. And in real world, in diabetic macular edema, it was shown that you have lesser of a visual acuity improvement with a biosimilar than the innovator. The improvement in the visual acuity was again significantly better with innovator molecule. The improvement in one or more line was significantly better with innovator molecule. And the adherence to the treatment, as in whether the patient decided to continue the same treatment beyond first few injection, that was significant. The compliance of the patient was much better with an innovator molecule. So although the mean number of injections, this was more with the innovator molecule as compared to the biosimilar. And similar were the results with AMD. That in terms of the visual acuity improvement and in terms of better compliance, I think the innovator molecule outperformed the biosimilar. So this was the result of an open label retrospective study from Disha Eye Center from West Bengal. The randomized control trials, they are not in synchronization with real world evidence. So when it comes to cost comparison, because of multiple complexities in manufacturing and storing the drug, the typical biosimilar would cost about 65 to 80% of the original molecule. And so in comparison, the generics cost only about 10 to 15% of the original molecule. So because when it comes to an informed choice, we need to weigh the cost, the cost that is offset by use of a biosimilar versus the efficacy and the patient compliance that is to be expected out of a biosimilar. So this is the comparison. This is the choice that we need to make as a clinician. Thank you.
Thank you, Dr. Nikhil. This was the last speaker of the session. I thank all the participants and the speakers and Dr. Ridhima for making this today's session interesting. Thank you all.
Thank you, ma'am.
Thank you. Thanks a lot.
Thank you.
Hello, everyone.
Hi, good morning.
Good morning. Hello, I think everyone is here. So let's begin with an interesting ocular surface session, and we have all the dignitaries and faculties over here already logged in. To begin with, we'll begin with Dr. Sangwan, and all of the speakers in this ocular surface session really do not need any introduction, but just to complete the session, I would introduce Dr. Sangwan. He is presently a consultant of cornea and ocular surface at Shroff Eye Hospital, Delhi. As we all know that he used to be at LVPI. He was a director of the Delhi Prasad Eye Institute, Hyderabad, as well as Dr. Paul Dubot, chairperson, chair professor. He is known for his work on limbal stem cells and the one who innovated SLED and changed the world of limbal stem cell transplantation. He's also a receiver of Shanti Swarup Bhatnagar Award, which is one of the highest award in science and technology, and also a receiver of P. Shiva Reddy Researcher Award. And he also has hundreds of international peer-reviewed publications in his credit till now. Over to you, Dr. Sangwan. So he would be presenting on management of complex ocular surface diseases, especially his pet area, that is, he would be presenting about the simple limbal epithelial transplantation. Dr. Sangwan,
thank you very much.
you're there?
Yes, I'm there. I'm there. Can you see my slides?
Perfectly.
Yeah,
Yes,
thank
we
you.
can see.
Thank you very much for kind introduction, Swapnil, and good morning to all dear friends and those who are logged in this Sunday morning to listen to us. I'll very briefly talk about surgical management of complex ocular surface disorders. When we say ocular surface, what we mean by is the front part of the eye, and there are three essential elements for the normal maintenance of the ocular surface, which are corneal, the surface epithelia, ocular surface sensitivity, and ocular surface and the tear film, the preocular tear film. The surface consists of the front part of the eye exposed to the environment, and the epithelia consists of mucocutaneous junction from the lower lid, palpebral conch, fornix, bulbar conch, limbus, cornea, and the same upper half of the eye, the front part of the eye. I won't talk much about the basics of the limbal stem cell or limbal stem cell deficiency. Maybe now it is better, my contrast. I was facing the window. All right. The spectrum of these diseases consists of a variety of disorders which need medical treatment, or you may have immune mediated disorders, or you may have a chemical burn. If you intervene early, you may also stop the progression of for them from a medical to a surgical disease. So many of these, like for example, a VKC, which is severe disease affecting the limbus, on those situations you should use systemic medications to prevent and to stop the progression. The meibomitis obviously needs a very different treatment. It doesn't need surgery. Similarly, when the patient presents to you with a sort of a pannus on the covering the entire ocular surface, If it is very clear from the history that there has been injury with some sort of a chemical, then it's a very clear-cut diagnosis. If it is not very clear, patient doesn't know, then you need to be very careful in ascertaining what are you dealing with. And many times you may have an underlying immune-mediated disease such as healed Moran's ulcer and or VKC. Those kind of patient you would need some sort of systemic immunosuppression if you end up doing surgery to keep the surface happy and quiet. These are two patients. The left side photo represents a patient with OCP and the right side represents a patient with BKC. And both of them looks, they need, like, especially the right one, looks like that you need a surgical intervention. But unless you do a good medical control of the inflammation, the surgery would not be a good idea. For OCP, it's always systemic immunomodulatory drugs which will help, and that will also help it to prevent progression to a further deterioration of the ocular surface. Another example of very active immune-mediated allergic eye disorder, VKC, and we see these kind of patients, about 3 to 5% of our VKC patient in clinical practice, which would need aggressive treatment with topical or systemic immunomodulators. Now, when you decide surgical treatment, the first thing that you want to differentiate is whether the surface is wet or bone dry. So bone dry surface we will not talk about, is primarily in situations like Stevens-Johnson syndrome, maybe OCP, maybe viral fevers which caused this condition. And by and large, other conditions, whichever you may be dealing, would have a wet surface. In addition to the wet surface, also please check some sort of ocular surface sensitivity, which we talked. So now out of 3 elements, we have 2 elements, surface sensitivity and preocular tear film. Now we're trying to replace the epithelia. And the condition, what it calls is limbal stem cell deficiency, and which we'll talk about it. Then another question that you have to deal with in ocular surface, complex ocular surface diseases, are you dealing with a medical or surgical condition? Here, obviously on the left side, both the situations are medical treatment, while in the left side, it is a OSSN which requires a surgical intervention. Most of the OSSN, nowadays there is a chemo reduction strategy where you can use different medications. The techniques for dealing with complex ocular surface disease surgically is variety of tools you have. Amniotic membrane, limbal transplant, which is in different ways, cadaveric, live related or cultivated, or a slat. So rest of my talk, I'm going to talk about the slat only, and it's the technique and the outcomes and how does it help. Before that, let's just briefly touch on a video. How do you suture the amniotic membrane if you don't have a fibrin glue? And this you may need for a variety of different indications, including slat. So this is a video showing you the suture placement, which is circumferential 10-ohm monofilament. And now if you have the fibrin glue and if you're patient and you can afford, I think that's a much better option. Otherwise, using sutures is not bad idea at all, and it works absolutely fine. The suturing technique, as you have seen in this video, is slightly different, and probably taking some circumferential sutures is much more efficient and helpful. If you're going to use the fibrin glue, this video shows how you are going to use the fibrin glue, basically mixing both the component. Then after excision of the pannus and or whatever the lesion you are excising, and then you achieve the homeostasis, and then with gentle cautery, and then use the amniotic membrane. First the fibrin glue, or you can spread the membrane entire ocular surface, then lift one half and put both the components of the glue, and then you iron it out. Similarly, you can repeat on the other half. There are different ways and means of putting the fibrin glue, and there's no particular way better than the other. As you are comfortable, as you think it is working, is perfectly fine. The cultivated limbal epithelial transplantation, which is one way of treating limbal stem cell deficiency, is to grow the cells in the lab. And this is what a technique we have developed in LVP since 2001, where these cells are, limbal biopsy is taken and it grows for 2 weeks in the lab on the amniotic membrane. And the cultivated cells offer variety of advantages. Especially it reduces inflammation, scarring, and amplification of the stem cells. Then the outcomes of this technique have been fabulous, reported widely in the peer-reviewed literature, and in excess of 70% success rate over 5 years and even up to 10 years. The drawback of the cultivated limbal epithelial transplant is two. Mainly, the cost of maintaining a clean or a CGMP grade facility is very expensive, and two surgical procedures for the patient, and the gap of at the cap of two. And then pediatrics LSCD is not very amenable to CLAT. So we started doing the simple limbal epithelial transplantation, which was demonstrated briefly here. And maybe I will skip the video since the SLED videos are now widely available. And if required, I can share those videos separately, those of you who want. So I will go ahead and skip the video. I mean, rest of the technique is same which I showed you with the amniotic membrane. On top of that, you just put the explants cut into small pieces. And on top of that, you put the fibrin glue and then put the bandage contact lens. You can expect these kind of outcomes. This is about 6 to 8 weeks follow-up. This is 3-year follow-up before and after. When you compare these three techniques on these 7 parameters, almost all, either the SLET is better or is equal to other techniques. Essentially, SLET, you know, what it has done is it has combined the advantages of both CLOW and CLAT and neglected or negated the downsides of both these techniques. This is a study which is under publication now, and where we shown the cost of SLET in India is about $700. Cost of the CLAT in India, when you take all the costs in condition, is about $7,000 US. If you compare CLAT using Holoclar as a product, which is approved in Europe, this is about 80,005 euro. So obviously the costs are a tremendous change. How these cells heal or the surface, and this is a very good study published by Vikas Mittal and his colleagues, where he showed over a period of time, if you observe these cells, there's a clearing or there is epithelialization of the surface at all around 360 degrees. And in about 10 to 14 days, like in CLAT, where you get to a good layer of cells in 10 to 14 days, you get a monolayer here. And this is exactly grows same manner as CLAT in the lab and SLET in the patient's eye. Also, when you compare with CLOW, the CLOW, there is a unidirectional flow of cells. Only from the cut edge, the cells grow. Rest of the area is not healing. Here in SLET, you have each explant giving rise to epithelial cells in 360-degree manner. So therefore, the rate of healing is much faster, and peripheral central cornea heals at the same rate. How it might be working? We think that these implants have growing cells all around, which is visible to everyone. They also, the mesenchymal cells contained within the explants are the ones which are contributing to the success. And they have variety of growth hormones, exosomes, and variety of growth factors. Another observation that we have made is the explants last or implant lasts much longer in case of SLET as compared to the CLAT. And these act as like a mini limbus. So in summary, the SLET is effective, replicable, repeatable, affordable, and a reliable technology where there is no patents. The surgeons can collaborate, contribute in further innovations. And there are many further innovations in SLET, which are published in peer-reviewed literature. Thank you very much.
Thank you so much, Dr. Sangwan. It was really a nice talk, and thank you for innovating SLED, because all of us who are sitting in periphery and doing their private practice, we could have never really thought of doing CLET in our practice. So it is really benefiting patients in the periphery as well. Thank you so much.
Thank you.
We'll take questions at the end. Now we have next speaker, Dr. Namrata Sharma, I don't think anyone in the audience need introduction of Dr. Namrata Sharma. So she is an cornea cataract and refractive surgery services in charge at Dr. RP Center for Ophthalmic Sciences at AIMS Delhi. She has done her fellowship from Moorfields Hospital, London, and she's now everybody knows she is an honorable General Secretary of All India Ophthalmological Society. She has multiple international awards at her credit, as well as she has published more than 450 international journal publications in international peer-reviewed journals, and she has also authored around 11 books. So, one slide is not enough to really introduce her. So let's move on to the talk, and Dr. Namrata, over to you. Her talk is about management of acute Steven-Johnson syndrome. Ma'am, you started? We can't hear you yet.
Can you hear me now,
Swapnil? Yeah, we can hear you.
Yeah, we can hear now.
Thank you, Swapnil, for envisaging this very good symposium. I would be talking to you about Steven-Johnson syndrome management in the acute stages. And Steven-Johnson syndrome or toxic epidermal necrolysis is, although it is rare, the incidence is 2 per million, but 70% of them may have ocular disease, and it is associated with the use of antibiotics, anti-epileptics, and NSAIDs. And 10 is, of course, more severe form of acute SJS, and SJS is when less than 10% of the body surface area is involved. Now, it can present in various forms, going from severe to less severe to more severe. And we come across all such presentations, and more so if you're attached with a hospital, with a general hospital which has a dermatology department or has an emergency department. In these cases, you would see more commonly. Now, generally, acute ocular SJS will occur within 2 to 6 weeks after symptoms. The ocular manifestations are seen in 50 to 80% of the cases, and 1/3 have long-term ocular sequelae, and there will be multiple ocular features which include bilateral conjunctivitis, conjunctival and corneal ulcerations, and pseudomembranes, early symblepharon formation, phonics, shortening, as well as meibomitis. Now, this is the natural history of SJS, which was very beautifully put into a paper by my mentor, Dr. John Dart, under whom I did fellowship. And there are variable degrees of scarring which can lead to dry eye. This is just to show that if you don't treat acute SJS, what is going to happen? So it is going to lead to entropion, keratinization, there will be dry eye, there will be variable amount of discomfort, and finally there will be limbal stem cell deficiency, and the patient will not be able to see at all. So it is important that you treat this in the acute stage itself, so that it doesn't all this sequelae doesn't happen of dry eye, epithelial defects, infections, delayed surface failure. Then recurrent SJS, acute episodes can also be there on chronic SJS. Then lipid keratopathy can be there, and sometimes you can even have a ocular cicatricial pemphigoid, which is a pseudo pemphigoid in these cases. So all these problems need to be addressed, and at times you can even have a late scleritis, which is common in these cases. The goal of therapy is that you want to control the acute inflammatory reaction, restore the intact surface, prevent long-term complications. And principles of treatment are that timely slit lamp examination is required for epithelial defects. Aggressive systemic and ocular medications may be required to achieve faster recovery, and regular sweeping of the fornices may be required. You have to prevent the formation of pseudomembranes and symblepharon in these cases. And the following are the treatment modalities which I'll be discussing with you. So in the acute phase, it is important that you do daily conjunctival hygiene, give prophylactic antibiotics. Role of topical steroids is uncertain, but amniotic membrane transplantation is now well proven beyond doubt that it should be given. And IV methylprednisolone has also been tried by some people. So in acute SJS, whenever there's recurrent conjunctival inflammation, or there is early or late ocular pemphigoid, or there's scleritis, immunosuppression is mandated, which could be with azathioprine, mycophenolate, or cyclosporin. And this is what Dr. John Dart advocates, that you can use steroids to begin with, and then later on can shift the patient to a alkylating agent or antimetabolite, or one of these agents, which I will discuss subsequently. But one should not mix the drugs in the same color groups because they tend to interact with each other. And this is the sheet which we had at Moorfields, which we used to follow for the various side effects of these immunosuppression charts. And suffice it to say that whenever you are actively immunosuppressing the patients, you would need the help of a internist. And this is the chart for side effects. So this is just to show how the steroids need to be tapered most immediately, but other immunosuppressive agents can be gradually tapered. And this is the step ladder treatment or immunosuppression, which is done in cases of ocular cicatricial pemphigoid as well as in cases of acute SJS, if required. Now, there are new immunomodulatory agents also which can be given, and these are given only in anecdotal reports with which do not have many randomized control trials. Now, we did a study. We found that in these cases of Stevens-Johnson syndrome, they tend to have infection. So we did a study to look at the conjunctival microbial flora in SJS at our center. And we found that staph was the most common organism in these cases. There are various kinds of lubricants which have been tried in acute cases, and this is the summary of those. It actually doesn't matter whichever you try. The only thing is that the lubrication should be frequent during the day, and also you should cover it with the various kind of gel combinations at night time. This is a study which was done for steroid pulse therapy in ocular SJS with the intravenous methylprednisolone. And although it was a small cohort or small subset of patients, this was at follow-up of 1 year, and they said that it did prevent ocular complications. And these are the pictures from the same study. Now, systemic steroids have also been tried, but again, there are no randomized studies to prove the same. And we did a study or randomized control trial in 82 cases, and this is under publication. We found that all the parameters, when we compared topical cyclosporin A versus no topical cyclosporin, and this was 1% topical cyclosporin, all the parameters were comparable except for corneal vascularization, which was less in those cases where topical cyclosporin A was given. Then amniotic membrane transplantation has a very important role to play and should be done for all cases of acute SJS, as has been shown in various studies. And we also did a randomized control trial to look at the adjuvant role of AMT in acute ocular SJS. And we took 50 eyes. In group 1, we did amniotic membrane transplantation with standard medical therapy. In group 2, we just did standard medical therapy, which consisted of topical steroids and lubricants. And I would like to acknowledge the idea of Dr. Virender Sangwan, who said that you have these patients in your dermatology wards, and you can just go there, and even in the wards itself, you can go and at bedside only put amniotic membrane and do this study. So this was mainly inspired from him. And these are the cases in which we did amniotic membrane just to show AMT group. Tear breakup time was found to be superior in the cases where amniotic membrane transplantation was done. Schirmer was found to be much better in those cases where amniotic membrane transplantation was done. Conjunctival congestion was far less in those cases. If you compare the amniotic membrane group versus the medical therapy group, you can see that the congestion, there's marked difference. And in between, we felt that why are we actually not putting amniotic membrane, because it didn't seem to be correct not to be putting it in these cases. Then this is again the medical therapy group where look at the simulafron, it is present, and in none of the patients in the group where we had put amniotic membrane had any simulafron. And then again, corneal hose, this is in the medical therapy group, and limbal stem cell deficiency, again in the medical therapy group at 6 months. And like I said, corneal vascularization, again, we also studied and found it to be significantly different. And this is the medical therapy group at 6 months. Conjunctivalization also was much more in those cases where AMT was not done. So this is to just summarize the factors which were better with the amniotic membrane transplantation, and almost everything was better, as opposed to the lid changes, which were not, which were found to be similar with both the groups. So this is again to show the same cases that in which amniotic membrane transplantation was not there. So we concluded that amniotic membrane transplantation is something which should be undertaken in all the cases without fail. And again, I would also like to bring to your attention the outcomes of COMET in eyes with chronic Stevens-Johnson syndrome that we have recently published in American Journal of Ophthalmology in 45 cases. And again, this is shown to have good results, but this is more for chronic SJS cases and not for acute cases. Then another thing that we did was that we gave a new classification of multi-step grading for evaluation of SJS cases, especially the chronic SJS cases, because we felt that our cohort of cases was much more severe as compared to the cohort of cases which original classification was given by Sotozono. So we gave a multi-step 5-grade or rather 6th grade, because it goes from score 0 to score 5, 6th grade classification for the chronic Stevens-Johnson syndrome. So again, this is the study from the COMET that we did, just to show how the patients become more comfortable and they start to see in cases of chronic SJS, and to show the results from the same. And in all the parameters, again, we found it to be far superior as compared to if you did not do a COMET in those cases. So in acute SJS is to be best managed by amniotic membrane transplantation, and we do believe that there's different grading which is required in Indian population as our diseases are more severe, and lubricants are the mainstay for symptomatic improvement. But in the acute stage, it is best to do amniotic membrane transplantation. And if you do have acute episodes later on in the course of the disease, then you may even have to resort to immunosuppression in these patients. So these are some of the review articles in which insights into Stevens-Johnson syndrome are given. This is a rare disorder, and ocular manifestations of these are not very much described and not studied also very well are not studied because of the rarity of the disease. And I would like to acknowledge Professor John Dart and all the people who have worked on Stevens-Johnson syndrome from the Department of All India Institute of Medical Sciences. We have already 2 PhD students who have finished their 2 PhDs, and there are 2 yet another one who are working on the same. So thank you very much for your kind attention.
Thank you, Dr. Namrata. It was really a nice talk, and I think everybody who is working in a multi-speciality hospital should be aware about this condition and how to manage that condition, because they are the ones who come in, who treat them first. So our next talk is by Dr. Sayan Basu, who is the Director of Center for Ocular Regeneration, and Virender Sangwan, Chair of Regenerative Ophthalmology. He is a dynamic ocular surface specialist who is developing novel stem cell therapies with, in collaboration with some international institutes. And he has achieved an achievement award from American Academy of Ophthalmology, as well as he has more than 100 peer-reviewed international publications to his credit. Over to you, Dr. Sayan. So he would be presenting about preventing recurrences in pterygium.
Can you enable my screen sharing?
It is enabled. I'll just check it again. It is enabled, Dr. Sayan.
Yeah, you just have to stop. You just have to click a green share screen, Sayan.
No, it's enabled now. It was disabled. My video is also disabled. Okay, can you see my screen now?
Yeah, we can see.
Excellent. So first of all, I would like to thank Swapnil for inviting me to present at this virtual seminar. It's always a pleasure speaking to all of you. And Swapnil asked me to talk on pterygium, which is a fairly common topic. And this is something that is encountered by ophthalmologists, irrespective of specialty, on a regular basis, and they come in all shapes and sizes. So I'm going to take you through what my understanding of pterygium is and how it should be treated, and give you some pointers along the way on to how you can achieve good cosmetic and functional outcomes with pterygium surgery, and what are the pitfalls that we need to be aware of and avoid. So what we know about the pathophysiology of a pterygium, and this is fairly well understood, although there's not 100% consensus on it, is that there is UV light mediated damage to the limbal stem cells, particularly nasally, and in some cases temporally, and in some cases both, because of the particular shape of the cornea and how the UV rays get focused on the ocular surface. And because of this destruction of the limbal barrier, the conjunctiva grows over the cornea, and that results in a progressive pterygium. And at some point of time, in most cases, the progression stops and the pterygium becomes atrophic. And in some cases, it may continue to progress and involve the visual axis, although that is fairly rare. The pathognomonic feature of a pterygium is elastotic degeneration, which you can see here in panel E, which I'll show you again in detail later on. And that is how the diagnosis of a pterygium is made. It has a head, which is the part of the pterygium which covers the cornea, the neck, which overlies the nimbus, and then the body of the pterygium, which you basically see over the ocular surface, bulbar conjunctivally. And like I said, the pathognomonic feature on histology, which is elastosis, is nothing but this whitish deposits. It's all denatured collagen that you see in the substance of the pterygium under the epithelium. You can see it clinically also in many cases, which are very progressive and fleshy. This elastosis on OCT is hyperreflective, back shadows, and on histopathology, you can see it either as very dense deposits at the base of the neck or head of the pterygium, or you can see as this amorphous denatured collagen in the mid of the stroma of the conjunctiva. Now, before we go on to understand and discuss what are the indications for treatment and what are the treatment modalities available, it is important to understand what is not a pterygium, and therefore avoid situations where you are dealing with something more sinister, and how we can do that. So always look for unusual locations. Usually a primary pterygium is nasal or temporal. If you have an unusual location like inferior or superior, if it is associated with dry eyes, if there is unusual pigmentation on the lesion, if the surface is irregular, or if the morphology is epithelial rather than subepithelial and OCT. These are all pointers towards other etiologies which you need to differentiate. For example, here on your screen, you see two cases of ocular pemphigoid, which was misdiagnosed and treated at pterygium, which led to worsening of the situation. And then it becomes very, very difficult to manage once limbal stem cell deficiency develops. Again, it has to be also differentiated from ocular surface neoplasia. On your left, you see the typical primary pterygium, and you can see here the epithelium is normal, and it is basically subepithelial, whereas in an OSSN, you see that most of the lesion is epithelial, and you can see the continuity with the corneal epithelial here. So this is one way of differentiating it from more sinister lesions like pemphigoid and ocular surface neoplasia. Now, the indications for treatment of pterygium, if you look at it theoretically, how it is written in a textbook, you have a lot of causes that are mentioned, but we know that primarily patients come with cosmetic concerns that we are trying to address, and rarely it is because of high astigmatism or progression towards the center of the cornea, or observed growth by the patient or the ophthalmologist, and so on and so forth. But mostly it's a cosmetic concern, especially in young and middle-aged patients. And therefore, the surgery that we do should give a good cosmetic outcome. This is very, very important to understand, that if we give a suboptimal cosmetic outcome, even though you remove the lesion and there is no recurrence, the patient will not be happy. Now, the problem with simple excision of the pterygium is recurrence, and that is the bugbear of pterygium surgery. Any surgical technique that is developed and is evaluated is on the basis of the recurrence rate, and therefore this is something that we have to keep in mind. Recurrent pterygium is nothing but a fibroblascular pannus. It is basically nothing but scarring or granulation tissue, and this recurrence is very different from a primary pterygium, which is mostly elastosis or elastotic degeneration, which is a UV light mediated degeneration, which is also seen in the skin in elderly individuals, which we clinically diagnose as freckles. They're nothing but elastotic changes, which causes a little bit of folding and pigmentation of the skin. Whereas secondary or recurrent terygium is essentially fibrosis. So it occurs because of the surgical trauma. It is not a recurrence of the primary lesion. You do not see the elastosis in the recurrent terygium. And therefore, you have to understand that it is poorly performed surgery or inadequate surgery which results in excision and not a recurrence, true recurrence of the same pathology as we use the term recurrence in, let's say, tumors or in dystrophies and so on and so forth. So it is the iatrogenic trauma of removal of the pterygium. So when we do a simple excision of the pterygium and we create a bascleral defect of the ocular surface, this heals by granulation. And it is this granulation tissue, the granuloma or the symblepharon, which basically resembles a pterygium morphologically, but pathologically is a completely different process. That's why if you operate on a recurrent pterygium, you realize that it's much more difficult to remove. It is much more adherent to the underlying sclera and also to the muscle. Now, how do we prevent this fibrosis or recurrence? By addressing the surface defect or by excessive fibrosis. So we need to cover the surface defect by a graft, which can be most commonly used as conjunctiva and also amniotic membrane. Other grafts have also been used and are reported in literature. And to prevent excessive fibrosis, you can use antifibrotics either alone or in combination with the grafts. Now, most commonly used antifibrotic is mitomycin C. 5-FU is also used, and there are others. But what we know from the published literature, and there's a lot of it, is that adequate private, especially if you're dealing with a primary pterygium, adequate excision with conjunctival coverage or doing a conjunctival autograft has the best outcomes, including the limbus per se at least does not have an advantage in treating of primary pterygia. Whether it has any advantage in recurrent pterygia, the jury is still out on it. We do not have a consensus on that. There's not enough data to show that. But mostly a conjunctival or well-done conjunctival autograft is the best treatment. And if the defect is covered well, antifibrotic is not required. Might be a role of antifibrotic in very advanced recurrent pterygia. So I'll just show you a simple way of doing the surgery. We typically have the patient on a topical anesthesia, put from some brimonidine to achieve a little bit of vasoconstriction, inject a little bit of preservative-free lidocaine in the mass of the lesion, and then excise the neck of the pterygium using simple Vanna scissors. And here we are basically excising just the superficial part of the epithelium and subepithelial tissue. And then you kind of pull the underlying elastotic tissue because it extends beyond what you can see, and do a dissection which involves as much of that elastotic tissue as possible. You can mostly eyeball the tissue that is degenerated and elastotic, and sometimes you can use other visual aids like using trypan blue dye, because the trypan blue will usually stain what is degenerated. And you can see here the whitish elastotic mass, and you can draw that out without going close to the rectus and excise it, and then peel the pterygium off the corneal surface. Usually in the primary pterygium, the younger the patient, the easier it is to peel this off. The older the patient, there might be more fibrosis and scarring. And then you measure the graft and you mark an area on the superior bulbar conjunctiva. It is always a good idea to inflate this area and also mark this area so that you don't lose orientation of the graft. And at the same time, you do not dissect the graft with too much of Tenon's capsule. The more tenons there is in your graft, the more fibrosis there will be, and the more vascular will be the appearance of the graft after it has taken up. So we excise the conjunctival graft again using Vanna's forceps, and here again trying to do as superficial a dissection as possible with taking as little tenons in your graft as possible. And once this is done, you can secure this graft on the bare sclera that you have created, either using fibrin glue or using sutures. Using sutures is not necessarily a bad idea, but it needs to be removed post-operatively because you typically want to use nylon sutures that do not self-absorb. Micro sutures become loose very fast. This can also be secured in place by autologous serum or blood that is there on the bed. I do not have any personal experience with it, but there are some people who are very experienced and are able to do it with a lot of reliability. It requires massaging of the graft on the bed for quite a while, so it takes a little bit longer time. Again, fibrin glue is not really that inexpensive and not readily available, so those options can be explored. And usually you want the, here what I'm doing is removing a sliver of the conjunctival graft from the limbus. This is a very common mistake that people make of leaving the conjunctival graft on the limbus. The limbus should be left bare so that it epithelializes from the cornea, and the cornea, when it epithelializes, it looks circular or oval rather than D-shaped, which what happens when you leave the conjunctival glove on the limbal surface. And then you can put a contact lens at the end of the surgery, not mandatory. Again, just taking you through the steps of surgery. Vasoconstriction before surgery using primordial is a very good idea because it is very little bleeding during surgery, and you can, otherwise in some cases the bleeding is so much that you kind of get lost. Injecting lidocaine, if you're doing on a topical, is very important. It kind of allows you to do completely painless surgery. Removal of the tenons is important, particularly the pathological tenons where you can see the whitish elastotic tissue. Without hurting the muscle, and by creating a very clean base clear defect where you put the graft. It is important to the graft to be adequate in size, and therefore you will see that when I'm putting the graft on the eye, you have the patient abducted. So even if you're doing the surgery under peribulbar anesthesia, make sure that you hold the eye with a forceps and turn it in the opposite direction so that you can measure the exact size of the defect. If the eye is in the primary position, you tend to undersize the graft, which leaves a bare area towards the nasal side, which then heals with granulation. So you get suboptimal cosmetic outcomes. And also remember to clear the limbal area. So about 2 millimeters from the gray clear cornea, that should be free of the graft so that when the graft epithelializes, it has a nice, good cosmetic outcome. A slightly more radical form of this surgery is called the perfect technique, where a much larger conjunctival graft is taken and is sutured in place. The idea here is that the edges of the graft, the superior edge is covered by the upper lid, the inferior edge is covered by the lower lid, and the nasal edge is so far back into the fornix that you can't see it, and the canalicular fold is reformed. This was described by Lawrence Hurst, who probably is the most experienced pterygium surgeon in the world. Fantastic outcomes with this technique, usually recurrence rates of close to 0 and very nice cosmetic outcomes. So what you're looking for basically is a eye which looks like this preoperatively. And if you look at the semilunar fold over here, that semilunar fold is dragged temporarily because of pterygium. And when the surgery is done appropriately, that semilunar fold falls back into position and you get a nice cosmetic outcome. So this is the kind of cosmetic outcome that you're looking for, where the patient's eye looks like the other eye or a normal eye without there being any remnant of the congested or bulky tissue of the pterygium, or there being any noticeable scar from the surgery or any sequelae from the surgery. It's important that the donor area is treated well. Like I said, just insufflate it with some fluid so that you don't take too much of tenons to avoid granulomas at the donor site, which can happen. And again, if you leave bare areas at the recipient site, that can also develop granulomas. These are basically, and they're not very difficult to treat. If you give intensive topical steroids or subconjunctival injection of steroids, they will usually go away and the conjunctiva will. So please remember this, that the pterygium is a benign degenerative lesion. It is mostly a cosmetic problem, and therefore it is important to do a good cosmetic surgery to give the patient a good, clear, clean cosmetic outcome. And there is no effective medical prophylaxis as of now. Surgical excision is the only treatment. The problem is recurrence, and the recurrence can be addressed if we do a clear, clean dissection and cover the entire area with a nice conjunctival graft. So gentle but complete removal, delicate graft harvesting, adequate coverage of the bare sclera, and proper anatomical realignment, and leaving no exposed bare area open. Just if you are going to deal with a patient who has pterygium and cataract, which is usually advanced, age, and these are usually atrophic pterygium, just if you're going to combine it, the indications would be where the visual axis is covered, or if it is progressive, and if the patient has cosmetic concerns along with that. You have to make sure that the patient understands that the poor vision is not because of the pterygium, because sometimes if you do only cataract surgery, or if you do only pterygium surgery, that and the patient's vision does not improve, both ways you may land up with an unsatisfied patient. And is the astigmatism regular? So if the astigmatism is regular and stable, then you can just go ahead with doing a cataract surgery. The pterygium is not very advanced. Restore the vision and then deal with the pterygium later. It usually will not change much. And if the astigmatism is very irregular, then you might want to do the pterygium first and then the cataract surgery. I think I've touched upon all the points, and if there are any further questions, then we can take them later. Thank you, Swapnil, for the opportunity, and thank you everyone for your attention.
Thank you so much, Dr. Sayan. It was really an excellent talk on preventing recurrences. So we'll move on to next talk by Dr. Kuresh Muscati. So we all know Dr. Muscati as an president of All India Ophthalmological Society. He was also president of Cornea Society of India between 2017 and 19. And practically there is no higher society in India where he was not, he has not served as a president. He has also received a Lifetime Achievement Award from International Society of Cornea and Keratorefractive Surgery. So it's nice to have him here, and he's one of a person who has started KPOs in India very early. And today he would be talking to us about keratoprosthesis, which to do, how to do, and when to do keratoprosthesis. Over to Dr. Muscati. I will be running your slides. Kindly let me know when to change the slide.
Okay, go ahead. Thank you, Swapnil. So we'll, in a brief 12 minutes, we'll try and cover a little bit about which keratoprosthesis to be done when and why keratoprosthesis as well. Let's go ahead. Next. So there are many different types of K-Pro in the market. The ones most widely used, still remains by far the Singh was champagne cork keratoprosthesis popularized by Dr. Daljit Singh. And in a personal communication with me, he told me he had done about 6,000 to 8,000 of these, but there's not too much published literature in this. The OOKP, Strempellis, which was modified by Falcinelli, which is now called the MOOKP, which was popularized in the early 90s by the Shankar Netralaya group, and abroad, of course, by Chris Liu in UK previous. And the Pintucci keratoprosthesis, which I started doing since 1997, which is a Dacron skirt haptic. It's now called the Chavan Muscati Pintucci keratoprosthesis, or CMP. And the Dolman or Boston K-Pro, which is completely different and which is done for different indications. Very popular now in India after Anthony Aldave introduced it in the late 2000s. Next. So should every opaque cornea be offered a K-Pro? No, because corneal transplant gives very good results in a majority of cases, both lamellar and full thickness. In fact, if you can't use a K-Pro, only natural cornea to save the eye, then you can think of a K-Pro later. And all successful corneal transplants have much better field of vision than K-Pros. Next. Let's start with the Boston K-Pro. The Boston K-Pro is an easier K-Pro to do and can be done in any patient with stem cell deficiency. Vascularized failed grafts, if there are multiple failed grafts, any opaque cornea, a lot of vascularization where you know it's going to fail. Any patient with stem cell deficiency, even if it's 360 degrees, and is not a good candidate for a stem cell transplant. But the eye should have good visual potential, no glaucomatous optic atrophy, posterior segment should be normal on B-scan, and importantly, it should have a good tear secretion, good lid blinking and apposition of the lids. So everything else should be okay except that the cornea and the stem cells are fired. These are excellent indications for Boston K-Pro. Next. So obviously there are some contraindications. So those with a poor tear secretion, like you see on the left, those with keratinized cornea again on the left, those with a history of previous cornea melts, as you can see the melted cornea on the top, and those with poor lid blinking or malapposed lids like the gentleman below, who's a lot of adhesions, lids don't blink, not a good candidate for a Boston K-Pro. Next. Contraindications for all K-Pros, Boston as well as Pintucci, is patients unable to follow up with a good eye doctor, even if the patient is from Jumritalaya or wherever. If you can communicate with the local doctor there and he follows what you're and he's willing to follow your advice, then it's important, because the patient can't keep coming back to you for small things. So either there is no qualified eye doctor in the area where the patient resides, or the patient himself is unable to realize the importance of compliance to treatment, And of course, in my case, because I'm in private practice, if unfortunately they can't afford my unreasonably high fees, they are not good candidates. Next. Next topic, yeah. So these are some of the patients who do very well. Chemical burns, that patient pre-op, and you can see the same patient post-op, and below Boston K-Pro as it looks like with the sutures in, and Boston K-Pro with sutures out. This was a patient who was HIV positive with bilateral stem cell deficiencies. She did very well with the Boston K-Pro, the one lady in the center. Next. So how do you evaluate the patient? You take that's Dr. Tony Aldave, who taught us all this in 2008, 2009 by coming to India and holding our hand, literally holding our hand and helping us do the procedure. So visual acuity, one thing I learned from him, sometimes you get patients with ankyloblepharons. You can't actually take visual acuity because the four directions of light he can't see. If you can consistently see the nasal projection, it gives you a guideline that there is no end-stage glaucoma. And I found this very useful. So I've done patients with the poor projection of light, but excellent nasal projection, and the patients have improved to 6/9 post-op. So IOP where possible, blink mechanism, tear secretion you need to know. You also need to know whether the patient is phakic, pseudophakic or aphakic, because the Boston K-Pro comes in two types, the aphakic one and the pseudophakic one. So if there's a good pseudophakia with a PCIOL in the bag, you don't disturb it. You ask for a pseudophakic model, which comes in one diopter, 45 diopters. And if it's an aphakic, if it's going to be aphakic or the patient has a cataract or a clear lens, you render them aphakic, in which case you have to send the axial lens to the manufacturer and they give you suitable power, maybe 50, 52, 54 diopters. Look for retinal detachment, cupping, etc. Next. So these are the parts of the K-Pro. The first, third and fourth part comes with the in a sealed box. The corneal graft is what you need to get from the eye bank. So you need a cornea as well. So the front part is mushroom shaped. The back plate has got holes in it. It used to be 8 holes, now 16 holes. And there is a titanium locking ring at the base. Next. So ideal cases, again to reiterate, one or more failed grafts with poor prognosis, vision less than 3/60, and preferably one-eyed patients, though I have now started doing in the last 5-7 years, patients with good vision in the other eye because their vision is fairly good and cosmesis is not so bad with a Boston K-Pro. Next. The contraindications, again, to reiterate, because this is what everyone is not going to do a K-Pro, but everyone should know the indications and contraindications. So for a Boston K-Pro, autoimmune diseases, pemphigoids, Stevens-Johnson, Sjogren's syndrome, very severe chemical burns like the girl above who is from Pakistan whom I operated for a Pintucci. So the face, the eyes, the lids, all were gone. Poor blink mechanism. If the vision is good, better than finger counting, you don't do this. And kids where cases where follow-up is not possible, and phthisis, of course. Next. So this is my first, I've done about 50 cases since 2009. This is the first patient, the pre-op with a divergent squint with a, he was one-eyed, and we thought we should attempt this. Perhaps it's amblyopic, but to our surprise, he improved to 6/12 in spite of the divergent squint. And you can see the cosmesis is not so bad. This second patient was had a history of collagen disorder. We were advised by Anthony Aldave not to do this, but we were eager to do this, so we went ahead and did this. She did have corneal melts on 3 occasions. She's been on immunosuppressives since the last 10 years. Treated with methotrexate, is maintaining post-op 6/18 vision. Not an ideal case, but sometimes you don't have a choice. Next. Next, Swapnil. So there are indications where you can't do the Boston K-Pro. I've been doing the Pintucci keratoprosthesis, done 85 cases of the classical Pintucci since 1997 to 2017. Then after Pintucci expired, we started making the prosthesis in India, and then we call it the CMP keratoprosthesis, Chavan Muscati Pintucci keratoprosthesis. Next. So the Pintucci is a whole new ball game, and this is a patient with Pintucci Capro 6/9 vision in the only eye. The other eye has got an optic atrophy. Vision 6/9, but is depressed because the cosmesis is very poor. And she says when she goes to play cards in a kitty party, people stare at her face. She forgets that she was PL vision prior. So generally patients with keratoprosthesis are unhappy for various reasons. Next. So the indications of my study were 85 bilaterally blind Asian patients with accurate light perception. All were unsuitable for or had history of failed PK and B scan showing attached retina. Initially, even for failed grafts, we did the Pintucci till the Boston Capro came along. Next. So the Pintucci Capro is a cylinder of PMMA and with a Dacron mesh, something like a Medu Vada sitting little off center, which is 0.6 millimeters in thickness. So you can see the picture on the right of what it looks like. It comes sterile. Next. I've done it in various neighboring countries. In Malaysia, this was a patient who had 17 previous surgeries. He was a milk vendor, and we did this sometime in 2006. Restored vision. I had to go because it's a two-stage procedure, so you have to go twice to operate. And then 5 years down the line, he was vending his milk at 5 a.m., crossing the road, and a truck came and hit him and knocked him off, and he died. So as I told you, Pintucci keratoprosthesis patients are either depressed because of cosmesis, or it's injurious to health if they see well because their field of vision is tubular, only about 30 degrees. Next. So the indications are bilaterally blind, and the tear secretion and lid apposition is not mandatory over here. So even a patient with poor tear secretion and with adhesions, ankyloblepharon, are still good candidates for the Pintucci. Next. Visual results are fairly good in the sense that no improvement of vision in 12 patients out of the 85 odd patients, but 3 feet to 20/60 in 64 patients. So most of them had working vision, and about 9 actually achieved this. So the results are not as good as the MOOKP, but the MOOKP is a more laborious procedure which involves the canine tooth from the mouth. It cannot be done in children, therefore only in adults, while here we've done it in children who are 9 years and above. Next. Every Pintucci has got more than one complication, so we've had lots of complications: retroprosthetic membranes, extrusion of the KP, KP drop into the vitreous, detachments, choroidal detachments, buccal mucosal overgrowth, glaucoma, phthisis, and panophthalmitis, and whatever you name it, and we've had these complications. Some of these have been treated by further surgeries. We've had 10 retinal detachments out of 85, and all of them, 9 of them have been successfully operated at Shankar Nethralaya. Next. So these are some of the secondary procedures that were done in these patients. That B scan shows a dropped Capro, like you would see a dropped IOL. I'm the only person in the world to have 3 dropped Capros in my series. Next. So though we see many more cases of bad chemical trauma as compared to cases of OSD and ocular pemphigus in the Pintucci series, the Pintucci KP works quite satisfactorily. Almost 65% of the patients showed some improvement in visual acuity. Next. Thank you so much for your attention. Swapnil, you're on mute. Yeah.
Yeah. Thank you so much, sir. It was really a nice presentation, and it's amazing work what you have done since many years. Next talk is by me only. So I would be talking about using a conjunctival flaps for ocular surface disorder. So why this particular topic is pertinent in this era is this particular surgery I found most useful in COVID era, because since March to almost September, October, we didn't even have therapeutic tissues to operate on the patients, and only thing which saved many corneas all over world is these days in last 6 months is conjunctival flaps probably. So conjunctival flaps is is a technique which is used for more than 100 years as a treatment modality for challenging disorders of ocular surface. The proper technique was described by Gunderson in 1958, and then it got popularized. So mainly indications of conjunctival flap are persistent epithelial defect, bullous keratopathy with poor visual prognosis, a severe chemical injury, and small corneal perforations where you are not able to get the tissue or anything to immediately, secure the globe. So this is how the conjunctival Gunderson flap was described first, where you have to just one second. So here is how it is described. So on the suprabulbar conjunctiva, you have to make an incision into the fornix and then near the limbus, and then extend this incision up to the 9 and 3 o'clock. Also, you have to give cut on the inferior conjunctiva, so that you have to do a 360-degree periotomy, and then mobilize the flap and then suture it onto the cornea. So you can suture it at the limbus, and then it gets secured very well. I do it slightly different, and I would be just showing my technique. And this is a very good procedure which every one of us should know, and it is very good for blinding eyes. So after applying a traction suture, turn my globe downwards. So I usually apply a clear corneal or limbal traction suture with 6O Vicryl. Now I have inflected the conjunctiva with lignocaine and adrenaline, and then I'm trying to excise the horizontal part which is into the fornix. And then I'm trying to separate the conjunctiva from the tenon. So this is also very important not to take a lot of tenons, because this is going to be a very large bare area, and if you touch the tenons a lot, it would find it very difficult to heal. So try to separate it properly, sparing the tenons, and go on dissecting towards the limbus. And also you have to take care that you go till 9 and 3 o'clock, so that you would be able to mobilize the conjunctiva properly over the ocular surface. So here I am trying to dissect up to the limbus. Hold the conjunctiva very nicely, take care of it, otherwise if it gets buttonhole, then you have to suture it and it will add to the extra complication. So this is the excision at the limbus, so I am doing a periotomy. So I usually do a partial periotomy rather than doing a 360-degree periotomy, and then try to free the flap from the limbus. But keep this pedicle on, and the pedicle should be thick enough so that it would not break down. Remove the complete epithelium. This is very important, because if you leave behind some of the epithelium, it might form epithelial cyst. I usually use a fibrin glue to adhere the graft completely over the cornea. Make sure that glue is spread all over, and then slide the pedicle flap. So you can see pedicle on both sides, and then try to iron it onto the cornea. But yes, make sure that there should not be any traction on both the sides, otherwise it will retract immediately. So try to reduce the traction by giving an incision, extending incision up to 3 and 9 o'clock. I usually secure my graft with 10-0 nylon sutures, which will go through the corneal stroma, circumferential suture. So here it is how it is taken. So take a long bite through the half stroma. And then, why 10-0 nylon suture? Because it induces less inflammation, and you can remove it later. You can bury the suture properly. This Vicryl suture, they are polyfilament sutures, induces a lot of inflammation, as well as it creates a lot of discharge also. So this is how I secure my Gunderson flap onto the cornea. And this is how post-operatively it appears, and it should be. These patients usually get a really good painful blind eyes really get good results, and they get really relieved by their pain. And I'm sure many of the cornea surgeons over here, they must be fan of these conjunctival flaps, because they are really a rescue in these desperate situations. Some key points, you have to mobilize the flap without traction. Do not give the traction. Determine preoperatively how much conjunctiva is available, because most of the time, many times in these ocular surface disorders, the superior conjunctiva will be fibrosed or scarred, and then you won't be able to mobilize it properly onto the cornea, and then it would be very difficult to really make a proper flap. If necessary, you can also inferior flap, and it may be combined to provide adequate coverage. Suture buttonholes, if any. Sometimes if there is some scarring in between and you might get buttonholes, so try to suture them. Remove corneal epithelium completely to prevent the epithelial cyst formation. This is also very important. So you can also combine the Gunderson flap with the tattooing. It can give slightly better cosmetic appearance because it would not look completely white. So this is one of the blind eye patients where I made the flap as I described onto the superior bulbar conjunctiva. Try to make it as thin as possible, particularly in this scenario where you are going to do a tattoo also, because through that transparent conjunctiva, that tattoo will give a better cosmetic appearance. And then you can spread the dye over the deepitalized cornea. And now all of us are equipped with Chinese weapon, that is those Chinese tattooing machine, which almost all of the cornea surgeons we have now, and we can do a nice tattooing with that machine after setting the needle depth and everything. This is how the tattooing is done, and just I'll wash it to see how much area is covered. So this pretty good area is covered with tattooing. And then over that tattooing, I put the fibrin glue and I'll mobilize the conjunctival flap. And rest of the procedure remains same. Once you mobilize the conjunctival flap, iron it out properly all over the cornea. And as I said, I usually put a three circumferential 10-0 nylon sutures so that it won't retract from the periphery. So you can put it in the periphery, the circumferential sutures. This is how the flap is sutured, and you can see the blackish appearance of the flap because of the tattooing which is done underlying the flap. There have been some modifications of conjunctival flaps. So suppose you have this epithelial defect or persistent epithelial defect or corneal thinning or melt. So you can always mobilize a pedicle flap like this, cut it from here, keep the pedicle intact, mobilize it and suture it with radial sutures onto the particular area where required. Or you can mobilize a bipedical flap like this, which is in D and E picture. So you can mobilize this flap, take it onto the corneal pathology, and then suture it onto the corneal pathology. So this is very good in even autoimmune conditions where you have corneal melt and you don't have the tissue immediately to go ahead and do the definitive procedure. This is a modification which was described in 2009. So where if the superior and inferior bulbar conjunctiva is scarred, you can always give a 8mm incision on both sides of the cornea and then mobilize these two flaps, bring it to the center and then suture it to the center. So this is a slight modification of the conjunctival flap where you would be covering from the sides, and you can always cover the bare tenons with AMG so that it would hit properly. So to summarize, it is a very underused technique, but believe me, it was one of the technique which helped me most in this COVID era, because since March we faced severe deficiency of tissues, and at times we had to do conjunctival flaps for non-healing ulcers, fungal keratitis, and many painful blind eyes, or even for severely symptomatic PBKs also we did a conjunctival flap. Now once the tissues are available, we can remove the flap and again do our desec or demec, whatever definitive surgery is possible. So it is an alternative to more mutilating surgeries like evisceration or all those things. So it's always better to learn this simple technique. It's not difficult to learn, and everybody should do it whenever it is required. Thank you so much. So I think next, I think Centaur is going to show some video before Dr. Pahuja's talk. Dr. Pahuja, you are here?
Yeah. Can you hear me?
I think someone from Centaur called me to, they want to show some video. Can you please start?
Yeah, I mean, okay.
You are only starting, sir? Okay. Yeah.
Yet another quality product in anti-glaucoma portfolio. Eyes are beautiful. You keep them so. Centaur commits support. Thank you.
Thank you. So next we have Dr. Kishore Pahuja. He's an ophthalmologist practicing in Pimple Saudagar since 1989. He's director of Natasha Eye Care and Research Center at Pimple Saudagar and director and chief ophthalmologist at Mirame Hospitals Private Limited, Chindwad. He's also trustee and president of Netradham, a charitable organization providing eye care for poor, and he's member of societies like CIOS, IRSI, GSI, and MOS and POS. He's one of a practitioner I have known who really takes interest in academics and really gets new things his armamentarium and try to experiment new things and really I really admire him. Dr. Pahuja, over to you. His talk is about medical management of dry eye. Sir, can you unmute, please?
Yeah, can you hear me now?
Yeah. I still can't see your slides.
Let me just share my screen. I can't see my presentation. Can you see my presentation now?
No, not yet.
Can you see now?
Yes, now we can see.
Okay. Thank you, Dr. Swapnil, for making me part of this, and thanks, Centaur, for inviting me for this. I'm actually an odd in the ocular surface experts over here, so probably I'll just try to justify what we understand for a dry eye disease is a multifactorial disease. There's a classification. I mean, there's a definition given by Deuce too. So we understand that there are 43 words to define dry eye disease. Whether can you just unmute all, please? Okay, so I mean, it has 43 words to define dry eye disease. So it's very unjust to do an justice in only in 12 minutes. But if you understand initially how we used to classify dry eye disease, one we do an Sharma and say is an aqueous deficiency, or we do an TBUT and say that it's a evaporated dry eye. But use two classification change our concept that how the patient presents, whether it's asymptomatic, symptomatic, and then there is a mixed type of an disease as an aqueous deficient and an evaporator. So how the patient presents to us, complaints of an blurriness, sensitivity to light, dryness, fatigue eyes, difficult to wear contact lenses, red eye, excessive tearing, gritty feeling. So if we understand that how this happens, I mean, it's how Professor Sagwan said is an ocular surface system. So in that ocular surface system, what we understand is an lacrimal gland, accessory lacrimal gland, meibomian gland, tear film. And these all are a part of an ocular surface system, and an stratified squamous non-keratinizing epithelium and a cornea and conjunctiva. So that makes it an stable tear film. And how it's maintained, because the lacrimal function unit is in a sub part of an system, in that it's controlled by a central nervous system, by lacrimal gland and the meibomian gland and the efferent and efferent nerves. So how the ocular surface system fails? This is Professor Sagwan's paper, which was published. There is when there is an squamous metaplasia, so that means a normal non-keratinized ocular surface epithelium changed to keratinized, which is a non-wettable surface, and or there is a limbal cell stem deficiency from a normal corneal phenotype. It is by an invaded conjunctive epithelium. So there are also various ocular surface disease. They coexist and intermix with each other, like a dry eye disease, and then are a non-dry eye disease like an MGD and allergic conjunctivitis. So we have to understand that how we classify or how we diagnose and then treat them accordingly. Meibomian gland dysfunction has been really taught nowadays, that this is one of a reason only for an evaporated dry eyes, and there's an alteration of an tear film, eye irritation, clinically apparent inflammation, and then ocular surface disease, including a dry eye. So when in an OPD we see a patients like this who has an thick orifices which are just coming out. So you press it and see that how much the secretion is coming out, and then you can probably understand that this is one of a severe form of an MGD. And then also we have a lot of equipments in our armamentarium now that we diagnose, we do an interferometry, meibography, NIBUT, and also we can just probably monitor the changes of an inflammation like blepharitis. Now, I think probably dry eye disease is the most frequently but underdiagnosed eye disorder, because it has a wide disparity and worldwide prevalence, like from 10.8 to 15.57. And India, approximately 30% of the patients in ophthalmology clinic, and in rural also they are more now. And hypermetropes more than myope than an emetrope. This was an study done by Professor Gupta, and it is also more common at high altitudes. In India also, if you see, that varies from 18.5 to almost about 50%. So considering this, what we understood this as American Academy of Ophthalmology, they did in a survey. So we also tried to do in a survey in last 2 months, where we saw about 90 patients a day, and then we calculated, and within dry eye disease like symptoms were almost about 58 patients. So that means it's very, very common, but we have to probably give an importance and stress to this. And also there was a utility assessment. What's a utility assessment is, they do it like, how severe the disease is and how severe the symptom is, which is affecting the quality of life. What they put it is, that's almost is almost close to an patients with an angina. And if you understand, is dry eye becoming an epidemic for an Indian conditions today, because there are a lot of computer use, pollution, and old age, and then alcohol, and also the AC draft and the dry weather, which is just making a part of it. And also other risk factors like contact lens wear, refractive surgery, systemic medications, and then a preserved eye drops. And tear film instability and hyperosmolarity, they are the core mechanisms of it. And then if you understand this, what is the core mechanism that usually causes an inflammation, has a lot of the interleukins on the tear film, which causes an inflammation, and then causes a dry eye syndrome. And according to Dewes, if you see the severity grading which has been given, from 1 is in a mild and 4 to a severe, where also they have advised in a surgical intervention. The role of history and symptoms cannot be stressed, because it is really, really important in diagnosing a dry eye. And these are the studies which started, and then now now probably the OSTI and the DEQ are very common, which are being really used in our OPD practice. And then accordingly, when patients gives and how much is the score, and accordingly we decide that probably we have to do some tests for this. So when we do this test, we also have some really limitation to the diagnostics, like Shermer, tear breakup time. It doesn't, I mean, the result depends upon the stimulation. So, some patients have more of a tearing. So Shermer becomes, falsely positive. Tear breakup time is highly depend upon the conditions of testing and also an inter-observer variability. Ocular staining like rose Bengal, it has sometimes it doesn't give you the way you want it. And when non-invasive tests, they came because they were really not giving a good result. So tear meniscus height, corneal topography, and tear interferometry and tear evaporimetry, we did that. But these also we have to understand they're very expensive and not available at a general practice level like us. So, but when we do a tear stability analysis within a topography, we understand whether it evaluates only tear stability due to an lipid layer or an overall tear film, which is still not really being seen out there. These are the advanced where we do a breast cytology, flow cytometry, and but then what challenges we face today in diagnosing an dry eyes is there is no gold standard, lack of well-defined cutoff values, and no separation between non-dry and dry eyes, vary depending upon the clinical circumstances, and ocular surface sensitivity in advanced cases is reduced, and less known non-invasive tests, and most diagnostics are poorly standardized, and severity, sensitivity, specificity, and repeatability, they vary. I mean, we don't have it. So if you understand the fact what we're going to talk, what's the goal of therapy? We want a relief of a discomfort, restore the homeostasis, and provide a smooth ocular surface and prevent the structural damage to an ocular surface. So this is our goal. So what's the treatment menu today is an artificial tears and an artificial tears. But I mean, it all depends upon that how you select the artificial tears. And so there are various polymers in an artificial tears like cellulose, CMC glycerin, and PVA, PGA, hyaluronic acid, which is really new in our armamentarium now. So because they are the mainstay of an trial treatment, and this was a study done which said that I mean, if you baseline assessment if you do and you do a treated, and after that the rose Bengal staining has reduced with an artificial tears. So what's an ideal tear supplement as an ocular lubricant? Ideal, no added irritation, increased dwell time, no blurring, symptom relief, increased tear breakup time, ease of use, it is cost effective and neutral to a slightly alkaline pH. So there was a few studies which were done. I'll just give two of an studies over here. Clinical trial which is done to compare the efficacy and safety of a multi-ingredient formulation of a CNC and an HA, which says it was a double-blind randomized study, and the conclusion was combination of an CNC and an HA eye drops. It reduces the significant symptoms of an ocular dryness post-LASIK. The another clinical trial which was done, which did about 305 patients, and that also that also proved that the best performance in ocular symptoms score was within a combination. The another clinical trial which was done, a prospective and a randomized multi-center phase 3. This also they conclude that CMC and sodium hyaluronate combination is more favorable than an HA alone eye drops. So if you understand an DUCE classification or as per the DUCE treatment, what you wanted to tear film instability will cause an hyperosmolarity, so that will cause an ocular surface inflammation, ocular surface damage, and also a neurosensor abnormalities. So what we have in a tear film instability, we should use a sodium hyaluronate and a CMC. And for a hyperosmolarity, you should have an erythritol and an L-carnitine. And so this really gives a benefit for treating all of the problems which has been explained by NDUs too. So and also it has a synergistic action as a sodium hyaluronate plus a CMC. And the another thing which is the oral supplements. So when they used it, so this was an dream study. It wasn't a contradictory, but then if you see that, I mean, if you if you really torture the data long enough, it will probably confess to anything. But then if you compare the other studies, they have proved that, they really work for this. And what are the recent trends that we do it? Immunomodulators, which is cyclosporin combinations. We have repopamide in our armamentarium now. Trehalose is really getting a lot of importance today. And then autologous serum. This is really, I feel that this is being used at an institute, and we as a private practitioner probably don't have that facility there. We can do that, but we have started doing it at our place, and it really helps us for the severe dry eyes. And then you have an intense pulse light therapy, LipiFlow, and amniotic membrane transplant, what Swapnil has advised. So conclude, ocular surface system failure leads to ocular surface disease. Utility assessments found that it's similar to a severe angina, and classical diagnostic test for dry eye system has many limitations, and lubricants is really in a mainstay of a treatment for this. And thank you very much. But I mean, at the end, I'll say that probably still we want something where we just fix it, and in dry eye disease within some drops, which can solve our patient's problem. Thank you very much. If there's any questions, I'm there.
Thank you, Dr. Pahuja. It was really a nice talk, and I would welcome if there are any questions, though many of the speakers left for their respective other talks. If anyone has any questions, I would try to answer in ocular surface, or otherwise we can conclude the session.
If there are no questions, then probably we'll just conclude it, because the time is over. We can just give it to the other session.
I think we have no questions. Rohit, you want to say something, or you are starting a next session?
I'm the moderator for the next session.
Okay.
Yeah.
I think it's time as well for the next session.
It's 12:30. So Dr. Rohit, you can just carry on. Thank you very much.
Thank you.
Thank you very much.
Can I invite Dr. Somyava Basu, who is the chairman for the next session, along with Dr. Anand Subramaniam, who is co-chairing the session on understanding ocular inflammation. I can already see Rohit and Samyak, who are here as the moderators.
And you can see me also now.
Hi Anand, good morning.
Hi
Is Dr. Somu there?
Dr. Basu will join in some time. He had an emergency OR today. And,
lucky guy.
And Dr. Parthoprati Majumdar is with us. So, and I hope Dr. Chetan Vedekar has joined us. Chetan, are you there?
Yes, Dr. Rohit, I'm there. Can you hear me?
Yeah. So we can start then. So at the beginning, I would like to thank Dr. Somyava Basu, Dr. Anand Subramaniam, Dr. Gaurav Shah, and Dr. Samir Mulkatkar for agreeing to be the co-chairs, the panelists for the session. We have a fantastic faculty. So if anybody has any questions, I would encourage them to type in the chat box. Today, we are going to start with the first talk that's on non-granulomatous anterior uveitis, clinical pulse by Dr. Chetan Vedekar. Chetan has done his post-graduation and then a fellowship at the prestigious L.V. Prasad Eye Institute. He's currently practicing in Nanded. Chetan, over to you.
Should I start sharing the screen?
Yeah, please go ahead.
Just let me know when you can see the screen.
Yeah, the screen is visible and we can hear you.
Shall I start?
Yeah.
Okay. First of all, I would like to thank Dr. Rohit for such a kind introduction, and I would like to thank all the. Hello? Can you hear me? Hello?
Yeah, Chetan, we can hear you clearly.
Okay, great. Thank you. Yeah, just wanted to confirm. Thank you. So I wanted to thank all the MOS organizers to give me an opportunity to present in this session of uveitis. I'm I consider myself really fortunate to participate in this session because of the other great stalwarts, many of whom are my teachers and many of whom are my seniors also. So the topic given to me is non-granulomatous anterior uveitis clinical pearls. I would just briefly introduce the topic first. So all of us know that anterior uveitis is anatomically the inflammation of the iris alone, which is iritis, or the anterior part of the ciliary body, that is cyclitis, or both of the structures, that is iridocyclitis. So basically, it is the inflammation of the anterior part of the uvea. In a typical patient of anterior uveitis, normally the vision is reduced during the acute stage. Usually it is easily manageable, but it is the sequelae of the anterior uveitis which leave a long-lasting impact and which can create a problem, as we will see. So there are different ways to classify anterior uveitis, and I thought it is important to just briefly enumerate them. It can be a typical limited acute attack, which lasts less than 3 months, or a persistent uveitis, which lasts more than 3 months. We should be specific in using the terms recurrent and chronic. The difference in here is when we are treating the patient and the patient responds, and there is a disease-free interval of 3 months without treatment, and then he has another episode, then we define it as a recurrent uveitis. However, when the patient has a recurrence before 3 months after stopping the treatment, then it would be a relapse and the disease would be chronic. That means the disease never subsided. So the recurrent disease subsides for 3 months and the chronic disease does not subside. So this is the first word. Now coming to etiology, the commonest etiology is autoimmune with only ocular involvement or associated systemic involvement. Rarely other infectious diseases can be associated, such as TB, and rarely other etiologies which are common are associated with other conditions like after trauma or after an eventful or traumatizing surgery, or uveitis can also be lens induced or drug induced. The classification that is pertaining to this presentation is basically a pathological classification. It will depend upon the nature of the keratic precipitates. The KPs can be either granulomatous or non-granulomatous. I will elaborate about this in further slides. So I would like to explain some pearls regarding the symptoms. Most common symptoms in a patient of anterior uveitis are pain, photophobia, redness, and blurring of vision, and sometimes watering. As is the clinical picture we see most of the times, the patients many times have repeated attacks. They have sought consultation with multiple ophthalmologists at multiple places, and they have used topical steroids and or systemic medications. And many times they are not properly treated, so they just keep on going here and there. Now coming to blurring of vision, most common cause of blurring of vision is the turbidity of aqueous which loses its transparency because of the inflammation. The other associated causes can also be corneal edema when the IOP is high, or sometimes there can be a complicated cataract also. The mechanism of photophobia usually is because of the ciliary muscle spasm, but it can also be because of the AC infiltration or epithelial edema or involvement of the pupillary muscles by inflammation. The pain usually is of varying degrees and it is attributed to the spasm of the ciliary muscles. Usually it is a dull aching pain, but sometimes the pain can be very severe when the IOP is very high. Coming to the pearls regarding signs, lid edema is usually not associated very commonly with a typical case of non-granulomatous anterior uveitis, but sometimes if the inflammation is severe, then it can be there. Coming to circumcorneal congestion, it is always important for all of us ophthalmologists to differentiate the circumcorneal congestion of anterior uveitis from the congestion of the entire conjunctiva, which happens in conditions like conjunctivitis. It involves the peripheral bulbar conjunctiva as well as the palpebral conjunctiva. So, on examination, it is easily differentiable. Usually, circumcorneal congestion is because of the enlargement of the episcleral vessels in the region of the ciliary body. That is why it is typically circumcorneal. However, sometimes in anterior uveitis, it can be absent also. Some examples are Fuchs heterochromic iritis and JIE. These are typically called as white eye uveitis. Now coming to the most important part, that is keratic precipitates. What are these? These are basically cellular deposits on the corneal endothelium. They can be of two types. One, fine keratic precipitates, which are found in non-granulomatous type of inflammation. These are basically formed from accumulation of lymphoplasmacytic cells. On the other side, the KPs can be large or mutton fat. There, in addition to lymphoplasmacytic cells, there are epithelial cells also. And then we classify the patient who have granulomatous inflammation. Old KPs are usually appearing colored or brown pigmented KPs. They indicate the past episodes of uveitis. Usually AC inflammation, if it is mild, it is in the form of cells and flare. But however, many times it can be very severe, forming a fibrin reaction in AC or forming many times a hypopion also and forming a pupillary inflammatory membrane. Usually, many times there can be iris atrophic patches also, and they are indicative of chronic iridocyclitis, and they are usually due to ischemia. Sometimes there can be myosis also, and it is because of the reflex spasm of the sphincter to inflammation and due to vascular distension of the iris vessels. Posterior synechiae, when we see, they are usually the result of adhesions between the lens capsule and iris, and they extend sometimes all around, which is 360 degree, and then we call it as occlusio pupillae. And sometimes occlusio pupillae is also seen, which is basically a condition where there is a complete pupillary membrane formation and it obscures the lens surface. Now it is important to always dilate these patients and see, because what is appearing on slit lamp is basically AC inflammation, may be associated with other things also. Some examples are there may be vitreous cells, there may be pars plana exudates or snow banking. Then these patients basically have intermediate uveitis and they have just a spillover of the inflammation into the anterior chamber. Other examples are there can be dyspedema or hyperemia, there can be vasculitis signs like sheathing or exudates, or there can be retinal or choroidal involvement in presence of a retinitis patch or choroidal infiltrates. Even there can be an exudative retinal detachment also. Such conditions are usually panuveitis, and the anterior chamber inflammation is a part of that. Some examples are sarcoidosis, tuberculosis, Rickets syndrome or sympathetic ophthalmia, and toxoplasma retinochoroiditis. So always make it a point to dilate and examine the posterior segment in detail to make sure that it is only anterior uveitis. Now coming to the most important part regarding the investigations. The first attack of a patient. So when we ask the patient how many times this has happened to you, the patient says this is the first time. Then we don't have to give him CBC, ESR, VDRL, LFT, AFT, HIV. We don't have to investigate extensively the first attack of non-granulomatous anterior uveitis. And the treatment is basically symptomatic, and we can treat with topical medications. The indications of investigation in a non-granulomatous anterior uveitis patient are very specific. When the patient is having recurrent attacks or when there is alternating uveitis in one eye and then the other eye, then we can investigate. On the contrary to this, when the patient has granulomatous anterior uveitis, whether it may be bilateral or unilateral, those patients we have to investigate. The most common investigation in a non-granulomatous anterior uveitis patient is basically HLA-B27. And the reason is HLA-B27 patients are usually having severe uveitis and recurrent nature. So we have to counsel the patients that the disease can occur again and again, and then it can be severe, and they will need long-term treatment. Coming to the clinical pulse regarding management, mostly the mainstay of treatment is topical steroids. The topical steroids work by reducing the inflammation. Along with that, we usually give cycloplegics to the patient. The main role of cycloplegics is to relieve the ciliary spasm, which gives symptomatic relief from pain, and also it helps to prevent or break the posterior synechiae. The most commonly used topical steroid molecule is prednisolone. Usually, it is preferred over betamethasone or dexamethasone. The reasons being its higher anti-inflammatory effect and potency, and also the corneal penetration of prednisolone is more than the other two molecules mentioned. One important point that I would like to mention is only plain steroid, giving plain steroid drops is enough. We don't have to give steroid antibiotic combinations, as we all know that it will always gives rise. It always gives rise to more and more antibiotic resistance. So pure topical steroid is good enough. How to use the steroids? This is also very important. We have to give the steroids 1 hourly or 2 hourly, depending on the intensity of the inflammation. As we very commonly see, 4 times or 6 times is not enough because most of these patients are having severe inflammation. Also tapering, sometimes the patients receive steroids and then stop after 1 week or 2 week or 1 month. This is not right because it is not going to help the complete resolution of inflammation. We have to use steroids till complete AC inflammation is gone, and the tapering has to be very slow. We can taper even 2 weekly in initial stages if the patient responds, and the tapering has to be over a period of weeks. Also, regarding cycloplegics, most of the times the patients need atropine and it is preferred over homide because of the ciliary spasm. The ciliary spasm is also very strong in the patients of non-rheumatic anterior uveitis, and many times they need atropine. In some selective patients where there are multiple recurrences or there is systemic involvement also, they will need oral medications in the form of oral steroids or oral immunosuppressions. So these are the few points which I think we all should keep in mind while treating the cases of non-rheumatic anterior uveitis. Over to Dr. Rohit, and I thank all of you for your patient listening.
Thank you, Chetan, for sharing the clinical pearls on non-granulomatous anterior uveitis. I would request our expert panelists to share their comments and views.
Rohit, I think we can take all the questions at the end, or do you want to take
Sure.
this?
Yeah, definitely. We can take the questions in the end. So moving on to the next
We have
presentation.
not had any questions in the chat box.
Yeah. Sure. So we will move on to the next presentation. Dr. Samyak Mulkutkar is with us. He is a senior consultant in vitreoretina uvea at PD Hinduja Hospital in Mumbai. He has done his post-graduation and then a senior residency from PGI Chandigarh. Samyak is going to make the enigma of intermediate uveitis easy for us. Samyak, over to you.
Thank you, Rohit. Can you hear me?
Yeah.
Okay. Can you see my slides?
Yes.
Yes. Thank you, Rohit. I thank Team MOSVIRCON, the organizers, and all the scientific committee, especially Rohit, for giving me an opportunity. And it's a pleasure talking to all of you, a lot of esteemed faculty here at the MOSVIRCON virtual conference. So I'm going to talk about how, just in a nutshell, about intermediate uveitis and how we can make it a bit easy. As we've seen, and Dr. Chetan has said, that any inflammation of the uveal tissue is called as uveitis. Now, according to the standardization of uveitis nomenclature, the predominant site of inflammation is very, very important. So if the predominant site of the inflammation is the vitreous humor, then we label it as intermediate uveitis. So you may have cells, snow banking, snowballs in the vitreous to label it as intermediate uveitis. How do we see that is on the slit lamp, we focus the slit beyond the posterior lens surface. And we can see the cells in the anterior hyaloid phase. We may see cells on retroillumination, and we may see vitreous haze. Snowballs are aggregates which are inflammatory cells in the mid-vitreous cavity, or you can see them on the inferior periphery. These are called snowballs. What are snow banking? Snow banks are pars plana exudates, so which are made up of loose fibroblast, fibrovascular tissue, along with mononuclear cells and fibroblast-like cells. And they are seen more of inferiorly because they gravitate down, and they signify severe disease, and they require aggressive treatment. There are a lot of times that you may miss snow banks, and that is why indirect ophthalmoscopy, sometimes with the peripheral scleral depression, is very important. in diagnosing snowbanks. Now, what may be the etiology? The etiology may vary from infective etiology or non-infective etiology to something which is non-infective. So there are a host of etiologies, but the data, if you see the literature, will vary according to geographic locations, the pattern of referral, genetics, and so on. So let's look at what the etiology says. Now, what is pars planitis? And all of us may have been hearing the term pars planitis synonymous with intermediate uveitis. Now, intermediate uveitis in the absence of any detectable systemic disease is called as pars planitis. That you have worked up for A, B, C, D, E, F diseases, you tried to find a diagnosis, but you didn't, and then you label it as pars planitis. And this terminology was used a lot previously, but in developing countries like us, we are seeing a different scenario. Let's look at a few examples. This is a 52-year-old Indian female. She presented with diminished vision in both the eyes, more in the left than the right. She had cells in the anterior chamber, and there was vitritis in the both the eyes. On OCT, she had macular edema on both the eyes. And if you look at a wide field fluorescent angiography, you may see a lot of leakage from the peripheral vessels. And you also see the cystoid macular edema on the wide field fluorescent angiography. So vitritis being the vitreous is the major site of the inflammation here. But you should remember that the peripheral vascular sheathing and the peripheral vascular leakage, it doesn't change the classification of intermediate uveitis. In fact, peripheral vascular leakage and macular edema are very much a part of the intermediate uveitis spectrum. She was evaluated further. She was a diabetic, uncontrolled. So the basic investigations like chest X-ray suggested mild accentuation of markings. The MON2 was negative, almost like negative. TPHA was negative. So we went ahead with a CECT chest, which revealed multiple discrete and large lymph nodes, bilateral and hilar in location. So she was investigated further, sent to pulmonology, where they did a EBUS, a guided biopsy of the lymph nodes, which turned out to be gene expert negative. Now, gene expert is a test that you do for detecting tuberculosis. So she was negative. She was a diabetic. So she was given Ozurdex in both the eyes because of the uncontrolled sugars, and she responded well to this treatment. Now, what does the literature say? If you look at the literature, if you look at literature from the North India and from the South India, there have been extensive studies done over years, and you can see that the commonest etiological diagnosis in our country has been tuberculosis. And there has been an overall decreasing trend of the idiopathic variety or the pars planitis because of the better diagnostic accuracy, or you may see the the way we chase diagnosis today. It would have been not imagined over the past decade. And this is one of the again large number of patients of intermediate uveitis. Most of them were bilateral, 67% of them, and all of them had vitritis, snowballs, and some of them will also have an anterior segment spillover. We have to know what are the other features. You may have a cataract, you may have glaucoma because of the associated steroid induced glaucoma, or an epiretinal membrane in the late cases. So the approach, when we approach a diagnosis of intermediate uveitis, you need a detailed ophthalmology examination for the primary inflammation. You need ancillary investigations like the fluorescein angiography, OCT, UBM, and the USG. And nowadays, you have a lot of literature speaking about wide field fluorescein angiography. And why wide field fluorescein angiography? Because if you do an angiography of the posterior pole, most of the patients you will be missing the inflammation on the posterior pole, whereas in intermediate uveitis, you have a lot of leakage in the periphery. Of course, you see central cystoid macular edema in the center. On the UBM, you may see cyclitic membranes, or you may see atrophy of the ciliary process. Now, atrophy of the ciliary process is a poor prognosis because it suggests severe amount of inflammation and long-standing inflammation. Well, cyclitic membranes carry a relatively good prognosis. Make sure that the patient has a general physical examination done. You may need to involve an internist or at times a rheumatologist to have the patient evaluated. What I prefer to do is, from my side, I get a MON2 or TPHA, get a chest X-ray and a CECT chest, either of them. Most of us would prefer a CECT chest because it gives us more information, and again CBC and ESR. We should not miss, and this is very important because in the elderly, the primary intraocular lymphoma may masquerade as intermediate uveitis, which at times responds very nicely to steroids and it keeps on coming back again and again. So the further investigation like an MRI imaging or sampling of the vitreous at times, a vitrectomy may be needed. And again, optic neuritis or periphlebitis also, again, a very much important cause of intermediate uveitis. So just in brief, how is it different in children? I'm not going to harp a lot because Dr. Mayur Morekar is going to speak on it, but you may see a lot of opacified hyaloid in children, especially they have a very, very strong vitreous reaction, and they may have these peripheral aggregates which are very strong. At times they may run into retinal detachment. So cyclitic membranes may cause retinal detachment in almost 10% of the patients, or they may cause inferior retinoschisis. You have to differentiate intermediate uveitis from GIA, which is a predominantly anterior chamber inflammation and presents with chronic arthritis. But they have severe visual impairment. Toxocara in children can also have, chronic inflammation with the peripheral granuloma and traction bands. Always rule out a foreign body in a child because at times the child is not going to give you the history of a foreign body. So when you have a lot of inflammation, not able to find out, try ruling out a foreign body. Intermediate uveitis requires a stepwise and a graded approach. Steroids are the mainstay of inflammation, whether you use topical steroids, periocular steroids, intravitreal steroids, oral steroids, or systemic immunosuppression. So this is a stepwise or a step ladder approach. Pars plana vitreous surgery, yes. In fact, we have a lot of studies now which where we find pars plana vitreous surgery as a good therapeutic approach or a good diagnostic approach. You have to make sure that you explain the pros and cons to the patient. Make sure that the systemic condition requires treatment. So if you have a adjoining systemic condition which is contributing to the inflammation, treat it. So recurrences should be taken care of and they require long-term treatment. And you should always remember the vision-threatening complications like cataract, glaucoma, an epiretinal membrane, and at times a retinal detachment due to the traction on the peripheral retina. Of course, those with pars planitis where you have not found any diagnostic, any underlying diagnosis, they don't have a benign cause. The inflammation keeps on happening again and again. So we should all be prepared for that. So just recapitulating the clinical pulse, thorough clinical examination is very, very important. Make sure we don't miss out on indirect ophthalmoscopy. The peripheral leakage or cystoid macular edema on the fluorescein angiogram does not change the diagnosis of intermediate uveitis. In fact, it is very much a part of the diagnosis of intermediate uveitis. Intermediate uveitis in the absence of systemic disease is called as pars planitis. Currently, we should chase the etiological diagnosis wherever possible in a prudent manner. We should treat in time because it may have visually debilitating complications, and we should watch out for recurrences as well. Steroids, immunosuppressives, or steroids in local or systemic manner should be given prudently. We should treat the cause because it may help us prevent recurrences. Last but not the least, there has to be a team approach. The ophthalmologist should involve the rheumatologist, get labs done, and other specialists as and when required for the treatment of intermediate uveitis or rather any form of uveitis. Thank you. Thank you, Rohit.
Thanks, Samir. for beautifully summarizing intermediate uveitis. The next, we have Dr. Partho Pratim Majumdar with us. As I call him Partho sir, he is a uvea consultant at Shankar Netralaya, Chennai. He is going to talk about viral anterior uveitis. Partho sir, over to you.
Thanks, Rohit. I hope my slides are.
I'll request all the other audiences to mute their mic.
Okay. So first of all, I would like to thank Rohit and Team MOSVIRCON for giving me this opportunity. I'll be talking on the viral anterior uveitis. So Rohit, I hope my slides are visible.
Yes, sir. You're audible and your slides are visible.
So viral uveitis is puzzling and sight-threatening ocular disease, and the signs of this uveitis can be very subtle compared to the other bacterial uveitis. The viral uveitis can be subtle, and you need to have a very high index of suspicion. So I'll try to, because time allotted to me was 8 minutes, so I'll quickly go through the clinical phenotypes, the laboratory investigation in a single slide, and I'll just highlight the treatment protocol used in viral anterior uveitis. So coming to begin with, I just want to show a case like 60-year-old gentleman who presented with recurrent attacks of redness and ocular pain, and he was treated with topical steroid, rebound inflammation on ceasing the topical steroid. So with this history, when he came here, on examination, we found cells, 1+ cells, flare, and multiple pigmented capes scattered all over the endothelium. As you can see here, he also had a high IOP of 32 millimeter of mercury. And so what we did, we started him on anti-glaucoma medication and the topical steroid with cycloplasm. So aqueous aspirate was done, and it was in PCR, it showed the presence of varicella zoster virus. He was started on oral acyclovir 800 mg 5 times a day, and you can see the result here. This was the picture at presentation, and you can see the picture here. So to begin with the clinical phenotype of viral anterior uveitis, I'll just share 5 or 6 pearls. You have to understand that the viral anterior uveitis is almost always unilateral. It typically responds to topical steroid and recurs when you stop or taper the topical steroid. So this is number 1. So and when while comparing the viral uveitis with the most common cause of anterior uveitis, that is HLA-B associated uveitis, one must remember that anterior viral uveitis occurs only in a particular eye. It is confined to a particular eye, right or left, whereas HLA-B27 associated uveitis by nature, it is recurrent, and it is known for alternating between the eyes. So the patient will come and tell that, no, my right eye was involved 3 months back, and now the left eye has started redness. Whereas in viral anterior uveitis, if you take a elaborate history, it is almost always confined to a single eye. Another important point is the high intraocular pressure. Any anterior uveitis with high IOP should be evaluated thoroughly and properly. And as you know already, that it can happen from the trabeculitis, and usually this kind of high IOP responds very well to anti-glaucoma medication and topical steroid. But one needs to evaluate and rule out the steroid response in cases of viral anterior uveitis. So viral anterior uveitis, usually the IOP rise responds very well to anti-glaucoma medication and topical steroid, because sometimes it can resolve the trabeculitis. The number 3, various kind of capes can be seen. There is no particular like only pigmented keratic precipitates you will see. It can have various presentations, starting from mutton fat. It can present with fine. It can have a medium sized keratic precipitates. As you can see here, there are a lot of topography of these keratic precipitates. But one thing you must pay attention, that the diffuse distribution, that is very unique to viral anterior uveitis. viral anterior uveitis is known to present with keratic precipitates which will have diffuse corneal endothelial involvement. So this is one important point. The next important point is the iris atrophy, or in absence of iris atrophy, sometimes the patient can present with dilated pupil. And if you take the history, the patient don't give you history of using the dilating drops. So a dilated abnormal, classically sometimes called as a D-shaped pupil, or presence of iris atrophy, it should point towards the viral uveitis. So as you can see here, and one needs to differentiate this viral uveitis with iris atrophy from other causes of viral uh, iris atrophy, which includes anterior segment ischemia, leprosy, trauma, previous attacks of angle-closure glaucoma, etc. The number 5 is the corneal involvement. It can be corneal edema. It can present with endothelitis. It can have a Decimet's fold, disc-shaped opacity. And so the main point is, if you see a patient with anterior uveitis who has a corneal involvement, always suspect or rule out viral uveitis first. So these are the few pictures you can see. I have placed them sequentially. First one was presented with corneal opacity, and it was due to herpes simplex. The next one is varicella zoster and the CMV. So sometimes it's very difficult from the topography of the corneal opacities. In textbook, various descriptions are there that this kind of opacity can be there, but any corneal opacity should be evaluated, and one should rule out these 3 viruses. One example is that CMV anterior uveitis. Classically, it is said that the CMV uveitis can present with coin-like opacity, like here. You can see coin-like opacity, which usually occurs in the center of the cornea, usually have a translucent halo, and the capes are brown pigmented overlaying the opacity. But variation can be there. So if you see this kind of circular corneal opacity, one should rule out the CMV anterior uveitis. Similarly, a study showed that the positive predictive value of observation that anterior uveitis and ocular hypertension resistant to topical steroid, not suggestive of HSV and VZV, makes the suspicion of CMV. So this observation, they found a positive predictive value of 94.7%. But you have to remember, it is not as easy, because they have mentioned that not clinically suggestive of HSV or suggestive of VZV, which is very difficult clinically to differentiate from CMV sometimes. So coming to laboratory investigation, I just want to show one slide and let it be very clear that serological tests are of no use. So if you are suspecting viral uveitis, torch titer has hardly any role. Why? Because these viruses are ubiquitous virus, and our 95% of the population is exposed to these viruses, primarily the HSV, VZV, and CMV. So positive result or raised titer of a particular virus does not make any sense in your clinical decision in a ocular involvement. So PCR is helpful, most sensitive. Also, PCR plays a role that to highlight the treatment. It helps you to differentiate between HSV, VZV, and CMV, and thus you can decide the treatment for the patient. Now, what is the treatments we have? The topical antivirals are not beneficial for treating HSV, VZV, but it is used for CMV. The rebound effect is very common with topical steroid, as already mentioned. And mainly our treatment for HSV, VZV is guided by a study called herpetic eye disease group, which recommended oral acyclovir at a doses of 400 milligram 5 times a day for several weeks. So several weeks, we'll discuss it now. The CMV anterior uveitis, on the contrary, can be managed with ganciclovir, systemic and topical medications. So now coming to herpetic eye disease, if you go through the study, it says that in cases of stromal, endothelial, and anterior chamber, that is anterior uveitis involvement, one should give oral acyclovir for a long term. Now, this is very important. One, you have to remember that the herpetic eye disease study, which was a multi-center prospective randomized study, it was conducted almost 20 years back. And the study assumes the clear delineation between epithelial and stromal keratitis. And if you see in your clinical practice, sometimes it's very difficult to differentiate between your epithelial and stromal keratitis. Sometimes the epithelial keratitis may have some stromal component, vice versa. So, and also the study was conducted when the sophisticated medications like antivirals like valacyclovir, ganciclovir was not available in the in scenario. And also the study was conducted long back, the vaccination for children and for adult was available. So that's how it's a question remains that how effective is herpetic eye disease study in determining the treatment outcome in your in viral uveitis in present scenario. So mainly we use acyclovir. You have to remember that acyclovir requires thymidine kinase for virus activation. So it is not effective for CMV anterior uveitis. So valacyclovir is in better drug compared to acyclovir, and ganciclovir, mainly the topical concentrations are used, which ranges from 0.15 to 2%. It is applied 6 to 8% a day for induction, and it can be used for 1 to 4 times a day for maintenance. To conclude my talk, I just want to mention 3 points. One is that diagnosis of viral uveitis requires high index of suspicion. Next, the serology has limited value and PCR is the most sensitive test, and herpetic uveitis usually requires long-term antiviral treatment. Thank you.
Thank you, Partho sir. It's always a pleasure and a fantastic learning experience, to hear your talks. Next, we have Dr. Soumya Basu to speak on ocular tuberculosis. He's the best guy to enlighten on also ocular TB. He has innumerable publications on various aspects, ranging from investigations to treatment aspects for ocular TB. He's currently the director of uveitis services at LV Prasad Eye Institute. Basu sir, over to you.
Thank you, Rohit, for the invitation and getting me to speak in the MOS forum again this year. Yes, I think I'm ready to share my screen. I've slightly changed the title of the talk. It's called TB or not TB, what I have learned about this controversy. Of course, a lot of things can be said, but I'll keep it short, keeping in view the time restrictions for this talk. So first thing is TB uveitis. It's a real disease. It's caused by Mycobacterium tuberculosis. And we know there are multiple clinical presentations, some of which are given here. The granulomatosis anterior serpiginous like choroiditis, and retinal vasculitis. And all of these respond to anti-tuberculosis therapy, either by direct resolution of the lesion or by non-recurrence, especially when anti-TB therapy is given along with corticosteroids. They will respond by non-recurrence after you have stopped the anti-TB therapy. So this is in short about TB uveitis. So the key questions that we'll address in this talk are what actually causes ocular TB, because people keep saying that ocular TB is related to latent TB. We'll see if that can happen. What should be the approach to diagnosis? Do we focus on the clinical signs or do we focus on the immunological tests? Does PCR help in accurate diagnosis? And what is the role of anti-TB therapy in managing what we call as ocular tuberculosis? Now, the first question is about latent tuberculosis. When we ask this question, first thing we should try to ask is, does latent TB cause disease anywhere else in the body? It does not. Unfortunately, we have assumed that just because we do not find Mycobacterium tuberculosis in the ocular fluids, it means there is no Mycobacterium tuberculosis in the eye. But we should remember that there are histopathological studies that have shown that the bug is actually present in the eye in all the clinical manifestations that come to us, although it's a paucibacillary disease, and anti-TB therapy works in these patients. Now, anti-TB therapy will never work on latent tuberculosis. It will only work when the bacteria are actually multiplying. So this shows that whatever we see, and if it is due to TB, then it is due to active bacterial multiplication and not due to latent tuberculosis. Want to know more about this? I suggest you read the small letter to the editor published in BGO in 2012. The second question is about where should be the focus? Should it be on the clinical signs or the immunological tests? Now, it is very, very important here to emphasize that always start with the clinical sign. If you are getting a clinical sign, say you're getting a sunset glow fundus, think of leakage. Don't think of TB, even whatever be the Montu or quantiferon results might be. You are getting a non-granulomatous antirevitis with fibrin in the antechamber or a hypopion in the antechamber. At least first think of the possibility of HLA-B27. Do not think of tuberculosis in these patients. On the other hand, if you have a patient where a retinal vasculitis comes with these retinitis lesions over the blood vessels, there's a serpiginous lesion like you see here with a central healing and a peripheral activity, or you see granulomatous antirevitis, think of tuberculosis and chase the diagnosis of tuberculosis. Don't stop just because some tests were negative. So start with the clinical sign. Remember that immunological tests can be false positive. Of course, all of us know that and all of us keep harping about this. But also remember that immunological tests can be false negative. And this is not just due to faulty technique, but there are several biological causes for which your Montu test or the quantiferon TB gold test can be a false negative. And we'll look more into it in a case that comes up next. Most importantly, whatever has been your conclusion, always rule out non-TB entities for a given clinical sign. So, for example, if you are getting a granulomatous antirevitis in an adult patient or a retinal vasculitis, think of sarcoidosis, think of Behçet's disease. Rule these out before you label something as tuberculosis. So this should be the broad approach when you try to diagnose something as ocular tuberculosis. So as I mentioned earlier, all these tests can not only be false positive just because we are in India, but they can be false negative as well. If we give a subcutaneous injection instead of an intradermal injection for Montu, you'll get a negative result. The tuberculin quality is poor. There is disseminated TB, or there is overall immunosuppression due to HIV or drugs or neutropenia, diabetes, and also in young patients. The younger the patient, higher is the chance of having a false negative Montu or quantiferon TB gold test. Also remember that all these, worry about BCG vaccination causing false positive test is not quite true. There is hardly any effect of BCG vaccination if it was given in infancy, as we do in India, if your patient is more than 10 years of age. So please be very clear about this. And this is not an opinion. This has come from very objective clinical trials that are mentioned below. So here's a patient who was tuberculin skin test negative, 28-year-old male. All tests were negative, but we saw these choroiditis lesions in the right eye and in the left eye, a larger one in the left eye. But since all tests were negative, this patient was treated with steroids and did very well. However, this patient came to me about a year later with this large lesion involving the macula and vision dropping to pointing finger close to face. So even though these tests were negative, we looked back at the previous picture and looking at a choroiditis lesion like this, and also looking at the possibility of TB contact in this patient, we started this patient on anti-TB therapy, and this without any corticosteroids. And this was the result. Within 2 weeks, you see how quickly it is melting away, and by 5 weeks, it was gone completely. So don't get distracted by a negative tuberculin skin test or a quantiferon TB gold test if you strongly suspect that the lesion is suggestive of tuberculosis. Trust the clinical sign. About PCR, now we all know that we hardly get a positive PCR from our aqueous fluid. And there are various reasons for this. The most important reason that I need to highlight here is the inappropriate sample. And this is an example of this is, say you have a serpiginous like choroiditis, where there is no inflammation in the aqueous. And in this patient, if you take an aqueous sample and try to do PCR on that, it will invariably be negative for Mycobacterium tuberculosis, because your inflammation is at the back of the eye. You need to get a vitreous sample for this patient if you want to look for Mycobacterium tuberculosis. But it's not just about false negative here. You can also have false positive results. And this we have seen in studies that we did at LVP, where we took patients who had culture-proven non-TB endophthalmitis. And these samples, when we tested for Mycobacterium tuberculosis, we found 16% of these samples were positive. We assume that this positive result came from bystander MTB DNA, because in TB endemic countries, the dead MTB can move on in the macrophages, and they can come into the eye and picked up in the vitreous sample if there is excessive inflammation. So as of now, the bottom line is that we are really not depending on PCR in making our diagnosis of ocular tuberculosis. Finally, the role of anti-TB therapy. As I said earlier, this can either be due to resolution of inflammation, where you have given anti-TB therapy alone, you know that the drug is working. But when you're giving both anti-TB drugs and corticosteroids, you know that anti-TB therapy works when there is no recurrence of inflammation, at least for a period of 2 years. Here is an example, retinal vasculitis with a retinitis lesion overlying the blood vessel. We treat these patients with anti-TB therapy alone, and when treated like this, we see that the lesion disappears very well without any need for corticosteroids. Another patient, large tuberculoma, not one but two of them lying here involving the macula. 2 weeks later, after starting anti-TB therapy, you see how the lesions are melting away. And in 2 months, they have almost completely disappeared with anti-TB therapy alone. We feel that in that retinal vasculitis and in such a tuberculoma, these are infection-driven manifestations of tuberculosis, and they respond very well to anti-TB therapy alone. On the other hand, when you have a lesion like this, where there's a central scarring and activity around it, which is called a serpiginous-like choroiditis, it is imperative that you add corticosteroids to that, because there's a very high risk of what we call as paradoxical worsening in these patients. And these should be aggressively treated with corticosteroids along with anti-TB therapy. To summarize, ocular TB is caused by active infection, not by latent disease. The clinical signs are always more important than the immunological tests. Of course, we should also rule out all the non-TB entities that could possibly give rise to a given clinical sign. PCR for ocular tuberculosis is yet to be proven useful in clinical practice, and anti-TB therapy is effective either alone when it's an infection-driven mechanism such as retinal vasculitis or tuberculoma. On the other hand, in conditions such as serpiginous-like choroiditis, we have to give anti-TB therapy with corticosteroids. Thank you for your attention.
Thank you, sir. It was it made ocular TB quite simple for us. Next, we have Dr. Mayur Morekar with us. He is going to speak on pediatric uveitis. Dr. Mayur Morekar is an associate professor of ophthalmology at Bombay Hospital. He is one of the leading uveitis experts in our state of Maharashtra.
Yeah, good morning, Rohit. Rohit, can you hear me?
Yes, sir.
Yeah. Is the screen visible?
Yes, the screen is visible.
Okay. So thank you all for having me here. And I have started all my presentations with acknowledgements to my teachers from Sion Hospital, from L.V. Prasad Eye Institute, Hyderabad, and my teachers in counseling psychology. So the topic for today is pediatric uveitis, and I have put the co-authors here because the take-home message for today is that a uveitis specialist cannot be a standalone treating clinician in pediatric uveitis, because pediatric uveitis is a special scenario where you would require many experts, because there are going to be systemic features, there are going to be dosages of drugs which would require a pediatric ophthalmologist. Of course, you would require management of amblyopia, which there is always a risk of setting in, so you always need a pediatric ophthalmologist along with you. And towards the end, we realize how we at times would also require retina surgeons. So coming to the first sort of a take-home message, that in autoimmune pediatric uveitis, there is many a time that one would require immunosuppressives, and therefore, if one is into management of pediatric uveitis, then one should not shy away from using immunosuppressives. The immunosuppressive of choice in pediatric uveitis is methotrexate, and the second drug which may be added is oral mycophenolate. The important thing to understand here is that the biologics are reserved for the more relentless cases. Herein you see a picture of a 15-year-old boy with non-granulomatous anterior uveitis and inflammation, controlled, presented to us with a band-shaped keratopathy, and the inflammation has been controlled using 15 milligram per week of oral methotrexate. The other thing that's important to understand is that follow-ups are going to be long. This is a 6-year-old girl who presented to us first in 2015, and she had a bilateral intermediate uveitis with disc edema. We suspected her to have ocular Behçet's, and she was actually B51 positive, had a rheumatology reference, and then was started on oral methotrexate. She actually then turned out to have a bilateral panuveitis, as the USGB scan showed choroidal thickening in both eyes and minimal subtenon fluid, suggesting a scleritis. What is important to understand here is that these cases are going to require immunosuppressives. You can see over here that the patient required an immunosuppressive in terms of methotrexate for a full 2 years. And then after that, we have almost a 3-year follow-up where the patient has now been free of immunosuppressives and has been maintaining a remission of inflammation. Coming to the topic of more interest to the general ophthalmologist is what to do when one needs to do a cataract surgery in a case of pediatric uveitis. One needs to understand that these eyes may sometimes be extremely inflamed, and that is why a thical change can also be triggered after a routine cataract surgery. And this change would be more likely to happen if the eye is operated upon in the state where there is an uncontrolled inflammation and where cyclitic membranes are present. And that is why all this inflammation needs to be tackled pre-operatively, and then one can have a good outcome. So I'm going to show here 3 case scenarios trying to highlight this. The first is a 15-year-old boy who presented to us with a left eye intermediate uveitis.
What we did first was control his inflammation. The control required almost 20 milligram of methotrexate and prednisolone up to 5 milligrams per day. The challenges in this case were that patient also had an additional glaucoma. There was a macular scar, and there was a little rebound inflammation that required us to step up the oral steroids post-operatively. This is the outcome at 3 months post the surgery, and the patient had a final best corrected visual acuity of 20 by 120 N18 with a glaucomatous optic atrophy, what was limiting the best corrected visual acuity. This is another patient, a 12-year-old boy with a bilateral granulomatous panuveitis, wherein we achieved control of inflammation, and the eye was operated when it was quiet, and we were on 10 milligrams of prednisolone per day with subcutaneous methotrexate 12.5 milligram per week and oral mycophenolate at 1500 milligrams per day. The challenges in this were the small pupil, which was there throughout surgery, the difficult capsular excess, but yet the surgeon, who is a pediatric ophthalmologist, was able to achieve in the bag IOL. This patient turned out to be what is Blau syndrome or a juvenile sarcoidosis, where it is always difficult to achieve remission. As you can see, there is a little hypopyon in a quiet eye after the cataract surgery is done. But yet we were able to achieve a good outcome. The patient ultimately landed up with a best corrected visual acuity of 20 by 40 and N12 in the affected eye, despite those corneal scars which are obvious. The third was a more challenging case where we required our retinal surgeon to intervene. This is a 13-year-old boy who's HLA B27 positive on spondyloarthropathy, left eye, a very severe non-granulomatous panuveitis. At the time of surgery, he was on oral prednisolone 7.5 milligrams per day, sulfasalazine 2 gram twice a day, subcutaneous methotrexate 20 milligram per week, and had been on about 6 doses of fortnightly adalimumab at the time of surgery. There have been many challenges in this. You saw the use of iris hooks that happened. You can see the fibrous anterior capsule, which the retinal surgeon is trying to tackle with a vitrector. Preoperatively, this patient had a UBM which showed cyclitic membranes, and the preoperative B scan was suggestive of a pre-thesical change, and that is the reason why we got the vitreous surgeon into the picture. The IOL here was implanted in the sulcus. The main reason the IOL was implanted was because it was planned to have silicone oil in the eye after a vitrectomy, and the IOL was put in to serve as a barrier to the silicone oil. This is the eye, how it is 4 days post-surgery. The patient is still on follow-up and maintains a quiet eye, although does not have a vision which is helpful. He has a vision of about counting finger 1 meter, but a quiet eye. So the take-home message from this presentation is that there has to be an extremely effective sync between the uveitis specialist, the pediatric ophthalmologist, and the pediatric rheumatologist required to achieve control of inflammation in cases of pediatric uveitis, and the role of the vitreoretinal surgeon in tackling cyclitic membranes when present in a child with a complicated uveitic cataract cannot be overemphasized. We're open to take questions. Thank you.
Thanks, Mayur sir, for a wonderful talk on pediatric uveitis. I have personally learned a lot from you and over numerous phone call discussions, especially on pediatric uveitis. Thanks for everything. Next, we have Dr. Vivek Dave, who's going to present on TARS versus endophthalmitis. It's a scenario where most of us can panic when we see a patient with severe inflammation on day one. So Dr. Vivek Dave is going to simplify for us because the treatment for both can vary significantly.
Yeah, good afternoon, everyone. Thank you, Rohit. Rohit, am I audible?
Yes, sir, you're audible, sir.
All right. I hope my screen is visible too.
Yeah.
All right. So I thank Rohit, thank the panel and MOSVIRCON for giving me this opportunity to share a few of my views on TAS versus endophthalmitis. So in this 8-minute span, I will touch upon both these entities with respect to their presentation and general management guidelines or outline of the general management, and end the talk with a couple of non-TAS endophthalmitis masquerades that one needs to keep in mind. Once in a while you may encounter them, and it should not crop up as an absolute surprise. So, as is common knowledge, endophthalmitis is inflammation of the inner layers of the eye with exudation in the vitreous cavity, and is an infectious entity by clinical definition. It has been classified largely as exogenous and endogenous. We will restrict the further discussion largely to the exogenous post-cataract surgery related endophthalmitis. That is where TAS would become a differential. Other ways of classifying obviously are acute or chronic, and also based on the timeline, it could be early or delayed, and as per etiology, bacterial or fungal. Now, when we look at post-cataract surgery endophthalmitis in the acute setting, it is the endophthalmitis by definition occurring within first 6 weeks of cataract surgery, or as the endophthalmitis vitrectomy study put it, first 6 weeks of either a cataract surgery or a secondary intraocular lens implantation. So how do we suspect? Now, all of us, when we operate, have a general idea of what is the average or the mean inflammation, or the mean, if I can put it, post-operative discomfort that the patient tends to have in our hands. Each one of us will have our own clinical curve. So any unusual inflammation post-surgery above and over what you would otherwise expect, any increasing pain or any decreasing vision, because as you treat any post-operative patient with your post-operative regimen, your pain should go down and vision should subsequently keep rising. So if you have these combinations of symptoms and signs, this is something that should give you the first red flag that something may be going on wrong in this particular patient. Now, first 2 weeks for most cases post-cataract surgery are very crucial. If you pull out studies related to acute endophthalmitis, there's a plethora of literature, but if you pull that out and try to do a meta-analysis and see what was the presenting time frame, then most of them, though the definition says 6 weeks, most of them will fall in the first couple of weeks. So this was also paralleled as an observation by the two big studies, the endophthalmitis vitrectomy study and the ESCRS study. So if you look at the days to onset of endophthalmitis, most of the cases are occurring in the first couple of weeks. So this becomes something where it's very important to impress upon the patient that in case he or she feels any deviant feeling, any deviant symptom, he should get back to you immediately, either telephonically or physically as may be appropriate, and not ignore them. If we look at the constellation of signs and symptoms, which again should alert us that something is going wrong, is blurred vision, which was found to be over 90% of cases in both the EBS and the ESCRS. So also on examination, a hypopyon, which we know is a sine qua non of presence of endophthalmitis, and haziness of the media. So when we talk in terms of media haze in this setting, we are going by the assumption that the cornea and the anterior chamber per se are not really obstructing your view, and everything IOL onwards towards the retina is giving you a haze or giving you a lack of clarity, which helps you, raise a thought in your mind that this patient is having an element of vitritis, which becomes a part of endophthalmitis. And as the topic goes, the differential to endophthalmitis, though not very common, but it is often spoken of in the same breath as endophthalmitis is spoken, which is the toxic anti-segment syndrome or TAS. TAS, again, as we are aware of, onsetity tends to occur within the first day of the procedure, and classically presents with a limbus to limbus corneal edema, a high IOP due to an associated toxic trabeculitis. There is classically no vitritis. This is difficult to pick up clinically because of the corneal haze, but a B scan would show you either an anechogenic vitreous cavity or a vitreous cavity with minimal echos on par with the echos seen in the other eye. Uh, TAS is often also spoken of as a diagnosis based on therapy or a therapeutic diagnosis, wherein you start the patients on steroids and there is a dramatic improvement in the overall clarity. That is because the underlying pathomechanism of TAS is, as the name suggests, toxicity to the endothelial cells of the cornea, causing an abrupt apoptosis of the cells. So that leads to this sudden occurrence of corneal edema as the endothelial pump would malfunction. And as steroids come in and the inflammatory toxicity is brought down, the cornea tends to get back to its routine functioning. Usual reasons, I would come to them in a little detail later, are unclean instruments, imbalanced pH solutions, pH in the solutions that are used intraocularly, and sometimes the presence of preservatives in the ocular fluids and medications. So there can be multiple sources, and each source can have its own cause pathophysiologically as to why it leads to TAS. And the common increment factors are the irrigating solutions or the ophthalmic viscoelastic devices, contaminants in your ophthalmic instrument due to residues from cleaning, or denatured viscoelastic which is stuck on the instrument. Ocular medication in case your reconstitution goes wrong. Say you're attempting to use intracameral cefuroxime 1% and you do not reconstitute it correctly, or intraocular lenses which come with their own chemicals around them, that in a particular eye or a bunch of eyes can set up a TAS. If you put them head on, there are certain factors that do help you swing either ways, either be TAS or infective endophthalmitis. Like we said, TAS invariably starts on the first post-operative day. Infectious endophthalmitis not very usually on the first post-op day, but tends to occur largely in the first couple of weeks. TAS is classically a painless condition, though there could be a little discomfort, whereas endophthalmitis tends to have various degrees of pain. A very important observation is lid edema, as inflammation in TAS is restricted to the anterior segment of the eyeball. Adnexal inflammation is not classical of TAS at all, whereas adnexal inflammation is very commonly seen in infectious endophthalmitis. So lid edema is a good differential between the two. Conjunctival congestion again will be very prominent in an infectious endophthalmitis. So also there would be relevant iris changes, changes in IOP, and the fact that therapeutically speaking, topical steroids will give a dramatic lasting response in TAS, whereas in an infectious endophthalmitis, steroids may just cause a temporary sort of masking of the underlying infection. Rough or overall management guidelines in endophthalmitis would be diagnosis, which would be by a clinical suspicion by looking at the constellations of signs and symptoms that we just discussed. Initiating an appropriate antibiotic and steroid therapy. This therapy should include empiric intravitreal antibiotics, and then as guided by the biopsy, which would either be taken as a standalone step or as a step in part of a vitrectomy. The biopsy microbiology evaluation would guide you as to what specific antibiotic would you use in your subsequent injections and as to whether you would add dexamethasone. Most practices would start off with vancomycin and ceftazidime as the empiric combination. And in case your smear or culture alludes to a particular type of group of organisms, then you would either drop your ceftazidime or drop your vancomycin and appropriately add an intravitreal dexamethasone. In fungal endophthalmitis, usage of intravitreal dexamethasone is controversial. Most practices would refrain from using intravitreal dexamethasone, but there are randomized studies which suggest that usage of intravitreal dexamethasone in a fungal endophthalmitis can also have a beneficial effect with respect to a quicker resolution of endophthalmitis, though the final achieved visual outcome may not be better than when dexamethasone is not used. When you look at TAS as a contrast, this usually tends to occur as a cluster because you may have multiple cases where a particular autoclave or a particular set of instruments may have got reused, and that would trigger off this reaction in the anterior chamber for subsequent patients. So the mainstay is prevention. The steps would include staff sensitization, strict instrument cleaning protocols, cleaning of the ultrasound water bath and the autoclave water reservoirs, and also to set up sort of an epidemiologic investigation to see and track back all the processes that happened before the TAS was diagnosed to get back to the increment source so that that can be taken care of. Again, differentiating how would you look at preventing an endophthalmitis? One of the common causes that endophthalmitis can occur is if you are operating in a milieu where there is a possible risk of infection. Uncontrolled diabetes can pose as a big risk. So by a general working guideline, random sugar less than 200 milligram percent is advisable as a start point to plan a surgery. Regurgitation over the nasolacrimal sac is important to be checked. If positive, cataract surgery should be postponed and done after a DCR or a DCT is done. Syringing is not recommended just before taking the patient for surgery because there can be residue from the nasolacrimal system which would be there in the cul-de-sac. Appropriate sterile OR and clothing. Batch numbers of all consumables that are entering the eye should be diligently noted, and there should be a low threshold to suture the wound, because if the wound is leaky or there is a negative pressure, it is likely to suck fluid from the cul-de-sac into the interiors of the eye. And all pre, intra, and post-operative findings need to be documented very well for any medico-legal requirement in the future if endophthalmitis occurs. So also for the general understanding of the operating surgeon as to what techniques or what measures that he or she took went fine, or what deviation from those measures possibly led to an infection. As of current knowledge, the only acceptable prophylactic or preventive pre-operative preparation, if we can call it, for preventing endophthalmitis is usage of povidone iodine 5% for 3 minutes on the skin and periorbita. It should reach till the boundaries of the hairline, tip of the nose, nasolabial fold, and the ear, and should be allowed to dry. So this is a very important clause, that one should allow it to dry and not quickly clean it off before draping. Also, in the conjunctival sac, 5% povidone iodine should be instilled and left there for about a minute. As far as other prophylactic factors goes, ESCRS study did look at multiple prophylactic factors, and the strongest prophylactic factor that came out that reduced the risk of endophthalmitis was intracameral injection of a prophylactic antibiotic. In the ESCRS study, they had recommended cefuroxime, relatively often used alternate now is intracameral moxifloxacin, as cefuroxime 1% often requires reconstitution to get your 1% dose from the injection, whereas moxifloxacin has to be used directly as the formulation available as drops, and from that point of view is technically easier. In case one gets an endophthalmitis, and prompt retina referral and management is important. A clear dialogue with the patient and the relatives, explaining to them that the condition is still treatable and that patient cooperation and possible referral may be required. It is important to have a peer review with another colleague, appropriately refer to a higher center, falling back on one's own OT, culture all solutions that are used, and I would repeat, document diligently to sort of cover you up in case there is any medico-legal action that occurs going ahead. As far as TAS is concerned, like we said, it is best prevented. This is important that the entire surgical team is aware about all the instruments that are used and have the know-how of all handling protocols. A regular sensitization of all staff handling instruments and their sterilization is important, especially if that staff of yours has a turnover. In case you don't have a very stable staff, the next person coming in may not be aware of what all is currently the protocol. Water reservoirs that our sterilization apparatus has need to be cleaned regularly. Usage of disposables as far as possible. Cleaning with oil-free compressed air, and documentation of all sterilization endeavors that were taken by the surgeon for any future cross-check and back referral. I will end my talk with two endophthalmitis masquerades, not necessarily TAS, but something which is a little similar and something where once in a while we may come across one of the masquerades, and the other masquerade is something which we come across relatively commonly. So this slit lamp photograph shows you a patient with day 1 post-cataract surgery. Look at the amount of fibrin that is present. Most of the pupillary area is covered by this whitish fibrin. There is also an associated hypopyon. If you look at the color of the fibrin, it is more whitish than yellowish. It is restricted largely in the pupillary plane, with the peripheral areas of the anterior chamber relatively clear. This patient had a normal B scan, and the adnexal inflammation was minimal to nil. So this patient was started on topical steroids and cycloplegics after having taken an AC tap. The AC tap did not reveal any organisms. The cycloplegics and steroids over the next 24 to 48 hours changed the view of the anterior chamber, like what is shown in this photo from what was at the presentation. 48 hours further down the line, this is what we had. So we saw that there was a retracting fibrin now present only in the central part of the pupillary area with clearance all around. And apologies for the hazy photograph, but this was what it was 2.5 weeks down the line, with the entire fibrin completely cleared. So inflamed looking anterior chamber, minimal adnexal signs, AC tap that was negative, response to cycloplegics and steroids alone, no antibiotics. So what did we exactly look at? We looked at an entity called as non-infectious endophthalmitis, not a very commonly used terms in clinical circles as such, but this is an entity which on slit lamp evaluation would look like an endophthalmitis. Posterior segment will be clear, AC will look hot, AC tap would be negative, and topical cycloplegics and steroids tend to take care of the condition. So this is more like an extreme post-operative inflammation as against a full-blown infectious endophthalmitis. The reference that I have put up here is a very interesting read which describes this entity in further detail, and would help us pick this up in clinical practice as and when we come across. Last 1 minute on this patient. So this patient was referred as a possible delayed onset endophthalmitis with this whitish sort of reflex in the capsule. And the history said cataract surgery was done around 8 to 10 years ago, and this was noted by the patient just in the last few weeks. The patient had a mild discomfort in vision, complaints were of mild fluctuating vision. On evaluation, otherwise, the anterior chamber was quiet, B scan was completely normal, and actually through the upper two-thirds of the pupil, the fundus was very clearly visible with a clear posterior segment. So this was thought of by the referring surgeon as a possible plaque in the anterior, in the lens capsule, and thus the suspicion of a delayed onset endophthalmitis. But I would just want to put your attention to this slit. If you can see, the slit is convex posteriorly. So that tells us all of this is material that is accumulating in the capsule and actually stretching the capsule behind the IOL. So this was the anterior segment OCT of this same patient. You can see the IOL. You can see that there is a zipper closure between the iris and the IOL surface. And this is the whitish material that is present, collected all behind the IOL. This is clear fluid behind the IOL. So what exactly were we looking at here? This was a capsular distension syndrome, which masqueraded as clinically a delayed onset endophthalmitis. Not a very common occurrence after cataract surgery, but once in a while you can have it due to residual epithelial cell metaplasia. Decrease in vision here could be due to a refractive shift or collection of turbid fluid in the capsule. So the vision doesn't really fall down to PL or hand motions, but it is a slight reduction in vision. So the entire clinical constellation can divert the treating physician to think that this is actually a delayed onset endophthalmitis. Nd:YAG capsulotomy is a very straightforward treatment for this condition. This is how the patient was post Nd:YAG capsulotomy immediately post procedure and had settled down subsequently. So this is how the anterior segment OCT then looked with all that material flushed out from behind the posterior capsule. So thank you for your kind attention. I would like to take questions in case any. Thank you.
Thank you, Dr. Vivek, for a wonderful talk. We would have the discussion after all the talks are over. And next, I would like to invite my dear friend, Dr. Rishikesh Kaza. He's a rising star in the uveitis specialty. He's going to speak on how to approach a patient of vasculitis. We also have with us
Yeah,
Dr.
Dr.
Preetham
Rohit.
Sawant, sir.
Hello.
Yes, sir.
Can I just take 30 seconds before Dr.
Sure.
Rishikesh starts?
Absolutely, sir. Absolutely.
So first of all, this session, you don't have to hurry up because we have a good buffer between 1 to 1:30. There is nothing happening here. Can you hear me?
Yes, sir.
Yes, sir.
Yeah. So there is the next session on retinal surgery is at 1:30. So don't hurry up your speakers. Take your time.
Thank you. Thank you. Thank you, sir.
And I'll take another opportunity to thank Dr. Rohit for conceptualizing this session. Thank you, Rohit, and for inviting and gathering all the faculty. My personal gratitude to Dr. Samyava Basu, Dr. Subramaniam, Dr. Gaurav Shah, Dr. Samyak. I can see my Samyak. Dr. Chetan Videkar, Dr. Partho Pratim, dear Partho Pratim, Dr. Mayur Morekar, Dr. Vivek, I heard your talk, fantastic. Dr. Rishikesh Kaza, thank you everyone for making this session and the conference a grand success. Thank you so much. So please go ahead, Dr. Rishikesh.
Thank you, sir. Thank you for your support.
Hi, good afternoon, everyone. I would like to thank the MOS conference panelists and everyone involved in the management. And I would like to thank Dr. Rohit for giving me the opportunity to present here. And I'll be sharing a few points on an approach to retinal vasculitis, which we follow in the clinics here and to an extent which I practice. So I'll start with this case. And just briefly, I'll touch upon the case and then we'll go into the approach from here. So there's a 30-year-old male gentleman who presented with left eye diminution of vision, but his visual acuity was 20/20. He complained of minimal floaters. And he's a pilot by occupation. He did not give any history of TB or TB contacts. But what we can see here is that if we look along the vessels, you'll find areas of perivascular sheathing and hemorrhages, multiple hemorrhages lined along the vessels with focal lesions and probably a few candle wax drippings, again along the temporal quadrant. Now, taking it from here, let's, the presentation will be divided into multiple questions and we'll try and answer them in the format such as this one. So the first question is, why is it important to diagnose retinal vasculitis? So the first point to note that is it's one of the most common posterior uveitic entities. And if we look at isolated retinal vasculitis, if you look at the literature, it's about 10% of all uveitis. And in addition to this, you can have it as a secondary manifestation in various diseases like intermediate uveitis. It can occur along with retinitis or chorioretinitis lesions. Now, considering retinal vasculitis, is there any similarity or differences when you compare systemic vasculitis to it? So if we look at the differences between the two, you'll note that systemic vasculitis is actually associated with vessel wall destruction, whereas retinal vasculitis, you don't have any evidence of vessel wall destruction. That is a histopathological evidence, which is necessary for a systemic vasculitis to be diagnosed. Etiology-wise, most of these systemic vasculitic entities are predominantly non-infectious, whereas it's the other way around for retinal vasculitis, where it's more commonly infectious, at least in our country or our population. And but you can have both infectious and non-infectious variants. Systemic vasculitis is predominantly arteriolar, whereas the retinal vasculitis is predominantly venous. And as I mentioned, that histopathology is necessary for a diagnosis of systemic vasculitis. So how does one evaluate a case of retinal vasculitis? These are the 5 points which I consider, and I'll keep this talk clinical. I will not be going into any investigations or systemic or ocular. So first, look at perivascular sheathing or cuffing. Second, try and decipher whether it's arterial, venous, or mixed. The third point to note is the morphological pattern of presentation, and we'll come to what are the morphological patterns later. The fourth one are the associated ocular and ocular features, clinical signs, which are presenting with the retinal vasculitis. And finally, looking at the systemic features and signs associated with it. So we try and deal with the first point, that is perivascular sheathing, and let's try and look at what is perivascular sheathing. So it's basically disruption of tight endothelial junctions, which causes a loss of the inner blood-retinal barrier, which leads to exudation and infiltration. Now, the Foka-Robin space is actually infiltrated with the lymphocytes and exudation coming out of the cells. And this is between the, just outside the vessel wall. So occurring between the pericytes and the glia limitans of the vessel walls. Now, the second point to look at is whether it's venous, arterial, or mixed. Now, when you look at the most common variants in our clinics are the venous predominant retinal vasculitis. And the main causes here include tuberculosis, sarcoidosis, or it can be undifferentiated. There can be a huge list to it, but I will not go into the entire list. But remember that TB and sarcoid are the most common ones here. If you're looking at an arterial predominant retinal vasculitis with perivascular sheathing, you can have it without or with perivascular sheathing. If it's with perivascular sheathing, then you also have to look for any retinitis lesions in the retina, maybe in the periphery, and you will eventually find one, and it can be associated with acute retinal necrosis, toxoplasmosis, or sometimes cat scratch disease, where the artery occlusion can occur right at the point of the retinitis lesion. If it does occur without the perivascular sheathing, then suspect a vasculopathy, because these may not have prominent vasculitis, but cases like diseases like SLE, where it's not very common to present with perivascular sheathing, though it can have, but these generally present with vascular occlusions, more commonly arteriolar occlusions. However, this is a case of SLE presenting with arterial involvement, but with perivascular sheathing, which is not quite prominent, but still can be noted. Now, the third variant is a mixed, where you can have arterial plus venous retinal vasculitis. And these are the cases where Behçet's disease is one of the examples where that can occur. And in addition to this, you can have idiopathic retinal vasculitis and neuroretinitis, or CMV retinitis, again, the viral entities which can have this kind of a picture. Now, let's look into the most interesting part of this, the morphological patterns of retinal vasculitis. So this is a whole list of them, but let's look at them individually. So the first one is skip vasculitis, where you can have segmental involvement with areas of apparently unaffected vessel. Here, one must remember that treatment of primary pathology is required, and you may not need treating retinal vasculitis separately, like in the case of toxoplasmosis, where you deal with the retinitis lesion, and eventually the segmental skip vasculitis will also disappear, which many of you would have noted in your practice. But it can be seen also with tuberculosis and sarcoidosis. The second entity is confluent vasculitis, where the whole vessel or the entirety of the vessel is involved. And in these cases, you might need treatment of the retinal vasculitis as a separate entity in a few cases. This can be seen with tuberculosis and sarcoidosis also. The third variant is called frosted branch angiitis because of its similarity to the appearance of a frosted branch of a tree. Now, this is characterized by severe perivascular infiltration, and the vision does not correlate generally with the amount of vasculitis that you see. Though they may not have any central CME or central retinal involvement or posterior pole involvement, the vision may be very low as compared to the involvement you see. The most important known etiologies of of frosted branch angiitis include CME retinitis, toxoplasmosis, SLE, leukemias, and lymphomas. However, it has been noted in tuberculosis also, but it may not be associated with any etiology where it would be classified as undifferentiated. The next variant is Chiarelli's vasculitis or Chiarelli's arteriolitis. In this, there are more often than not, you will find foci of retinitis or retinochoroiditis or chorioretinitis generally associated with it. But these can last much longer after resolution of retinitis lesions, as seen with toxoplasmosis or acute retinal necrosis or tuberculosis, in fact. Here, the deposits have been found to be within the arteriolar walls or intraluminal, or can be perivascular, but recent evidence suggests that they are actually within the vessel wall itself. An interesting point to note here is that these do not leak on fluorescein angiogram like the other causes of vasculitis. The last two variants, one is called hemorrhagic vasculitis, which is usually venous in nature. And one of the mechanisms proposed is proliferation of the intima of the blood vessels or endothelial cell proliferation, which can lead to occlusion and lead to hemorrhages occurring in the periphery. This can be noted with tuberculosis and Behçet's as the most common causes of hemorrhagic occlusive venous predominant vasculitis. Now, another variant of occlusive vasculitis, more commonly involving the arteries or arteries and veins both, where it's associated with the low flow circulation and a general profound or a diffuse ischemia, which may be more systemic than ocular itself or retinal itself. So systemic ischemia, which leads to choroidal perfusion abnormalities leading to occlusions. And here, typically, there is no perivascular exudation, and it's seen with Takayasu's arthritis, Aplas syndrome, and sometimes Suzak syndrome. Now, going on to the fourth question, which we'll deal with, what are the other associated features? We'll talk about ocular, and I'll leave systemic out of the picture, but just briefly touch upon them. So the ocular features, we'll mainly look at the posterior segment features because it's common to have posterior segment rather than the anterior segment involved in most cases. Vitritis, snow banking, cotton balls, or retinitis and chorioretinitis lesions may be noted. And the various lists of differentials have been listed there. But the other interesting signs like microaneurysms, vascular tortuosity, healed scars, and occlusions should be given due attention. Also, systemic involvement like lung involvement or cutaneous involvement or skin involvement has to be looked for actively and needing systemic investigations for them. Now, a point which I can share is if a patient presents with retinitis with associated vasculitis, think of infectious unless proven otherwise. So rule out the infectious causes first. However, there are two exceptions to this rule. One of them is sarcoidosis and Behçet's. Both of these can have retinitis lesions. associated. We'll go into a few brief description of disease-specific signs in retinal vasculitis, and Dr. Basu mentioned about tuberculosis, and a few lesions which can be noted with tubercular retinal vasculitis include focal lesions like this, or sometimes you can find scars overlying the blood vessels. You can have focal vascular tortuosities, like this one here, where the proposed mechanism is an underlying granuloma leading to VEGF expression, and this VEGF expression causing a endothelial cleavage plane differentiation leading to the tortuosity. And sometimes you can have occlusive vasculitis, which we just talked about. In sarcoidosis, you might have anterior segment signs, but in addition, you can have candle wax drippings, continuous vasculitis, and you can have inferior multiple pinpoint or hypopigmented chorioretinitis lesions, which may be suggestive of healed lesions. You can have multiple microaneurysms at the disc or along the bifurcation of the arteries, which is quite characteristic for IRWAN. And in addition, in Takayasu arthritis, sometimes you may find multiple microaneurysms in the periphery of the apparently normal eye. The left eye was involved here. However, the right eye had good visual acuity. But if you look at the periphery of the angiogram, you'll find that there were multiple microaneurysms, so numerous in each of the quadrants, suggestive of a widespread ischemia. So let's revisit the 5 points which we looked at. The first point to evaluate for is perivascular sheathing or cuffing. Second would be to differentiate between arterial, venous versus mixed. The third one would be to look at the morphological pattern of presentation and try and classify it according to one. And finally, looking at the other associated ocular features or signs and systemic signs. So let's go back to the first case. Again, trying to keep it clinical. I'm not going into the systemic investigations here. So what we find here is that it's unilateral, venous predominant, associated with hemorrhages, focal lesions, candle wax drippings, and confluent vasculitis. So the possible etiologies which we can narrow down on clinical examination are these two. Thank you for your patient listening. I can take questions if...
Thanks, Rishi, for a wonderful talk on approach to a patient of retinal vasculitis. So, is my slide visible?
Yes, Rohit. Your slides are visible.
Can I start, Samir?
Yes, Rohit.
So, in changing gears, something which is quite radically different I'm going to present is how diet can make a difference in controlling ocular inflammation. Well, there's a disclaimer, I'm not a dietitian. I'm a fellow ophthalmologist and I'm still exploring dietetics, and there is no financial interest. So what I came across basically last year was a concept of whole food plant-based diet, where you eliminate all the animal products, which include all types of meat, poultry, and dairy. Eliminate processed foods also. So most of the packed foodstuffs we get nowadays are processed foods. And the focus is on consuming plant-based diet, especially a good proportion of raw salads. And this is a concept of power plate, which is recommended by the Physicians Committee for Responsible Medicine, which, if you have a mixture of enough raw fruits, vegetables, grains, and legumes, that takes care of all your nutrition. So there's another concept which is known as Nutritarian diet by Dr. Joel Fuhrman. He has even published the effect of such a diet on monotherapy in a case of psoriasis. What he recommends is a G-bombs concept, basically having more of green leafy vegetables, beans as any legumes, onions, cooked mushrooms, berries. For me, berries would mean most of the fruits, and seeds, something like flax seeds or chia seeds. There is another concept, especially for autoimmune diseases, that is a hypernourishment protocol by Dr. Brooke Goldman. Now, she herself had lupus nephritis, and she herself cured by the hypernourishment protocol, which she recommends to patients. She has published a case series of patients with lupus nephritis. The main focus of her hypernourishment protocol for the 6 weeks is on raw salads, especially the green cruciferous vegetables, which can be combined with seeds which are rich in omega-3 fatty acids such as grounded flax seeds. And if a person is not able to eat that much raw, you can blend it and make a green smoothie out of it. She runs a Facebook group known as Smoothie Shred. So let's look at the pathophysiology, why we are saying that a diet can have such a revolutionary effect in terms of inflammation control. Now, when we look at the sources of arachidonic acid, you can see they are majorly from your meat and dairy and poultry. And this arachidonic acid is basically responsible for all the inflammatory mediators. When we take a diet rich in omega-3 fatty acids, something like grounded flax seeds or chia seeds, they supply us anti-inflammatory mediators. The only rate-limiting step for alpha-linoleic acid is delta-6 desaturase. Now, this alpha desaturates limitation can be overcome by increasing the intake of flax seeds and chia seeds and restricting the intake of omega-6. That is by restricting vegetable oils, animal fats, processed foods, and meat, seafoods, and eggs. And when we are taking adequate amount of omega-3 fatty acids, there is a inhibitory effect on the arachidonic acid pathway. So this forms the anti-inflammatory nature of the diet which was discussed before. Now let's look at the effect of this kind of a diet on some of the autoimmune diseases. One is rheumatoid arthritis, where effects of low-fat vegan diet was studied, and it was found that the weight decreased significantly. At 4 weeks, C-reactive protein decreased, RA factor decreased, although ESR was unchanged. Now, some people can attribute this to the weight loss because many of us know it's a common fact that when we are on a low-fat vegan diet, we tend to lose weight, and when we lose that extra fat, it automatically has an anti-inflammatory nature. But when one of the papers studied the effect of weight loss and weight maintenance with diet varying in protein and glycemic index, and they found that a low glycemic, high fiber, low protein diet. Now, when you have a mixture of, let's say, vegetables, legumes, and your fruits, all these things are low glycemic in nature, and the fiber content is very high, and the protein content is relatively lower as compared to animal products. Now, this kind of a diet could mediate inflammation by decreasing the pro-inflammatory gene expression. So even if the people are prone for developing autoimmune diseases, it can decrease the gene expression, improve insulin sensitivity, even after significant reductions in inflammatory markers due to weight loss have been balanced. Another important thing is when we increase the intake of dietary fiber, it basically prevents the degradation of colonic mucus barrier. Now, this mucus barrier is absolutely essential to maintain the gut microbiome. The fiber acts as a probiotic, and these gut microbiome basically feed on them. They release short chain fatty acids which are anti-inflammatory, and also they act on the satiety center to reduce the calorie intake. Now, this is affected when we have a predominantly animal product based diet or a processed food diet. And now this has been shown that a translocation of gut pathobiome drives autoimmunity in mice and humans. So the translocation of gut pathomicrobiome to the liver and other systemic tissues to the autoimmune responses in a genetically background predisposing to autoimmunity. And this discovery show how gut pathobiome can translocate and promote autoimmunity in genetically predisposed host. So it's very important, and nowadays there's a lot of focus on the maintenance of good gut microbiome is being done. Another thing is that we know that type 2 diabetes is a pro-inflammatory state, and 15 years back when a randomized study was done to compare a low-fat vegan diet versus an American Diabetic Association recommended diet, the low-fat vegan diet showed that there was a greater weight reduction, also significant changes in the glycosylated hemoglobin. The total cholesterol in fact dropped significantly in the low-fat vegan group, and also the LDL cholesterol, the bad cholesterol also dropped. So these are the certain byproducts of a diet, and we recommend to our uveitic patients they can get benefited. In conclusion, whole food plant-based diet has an anti-inflammatory effect. It can be useful as an adjunct tool in our hands to help patients in terms of inflammation control along with standard treatment. Much more data is required, a lot of exploring is required on how this diet can be useful in a uveitis, and will customization help with respect to disease customization or patient customization. There are a lot of things which are unanswered in this field. My acknowledgements, especially Dr. Basu, who inculcated curiosity in me with respect to uveitis, Dr. Anand Kumta, who introduced me to the concept of whole food plant-based diet, and all the others through whom I have learned directly or indirectly about this. Thanks.
Thank you, Rohit. This is an interesting addition to our uveitis topics. I think it's a welcome change for all of us. I think we can open the panel discussion now.
Yeah, absolutely.
I think first of all, I would like to congratulate all the speakers and Rohit for choosing the topics, and I think all the speakers have done an excellent job. So I will not be again reiterating the fact for each of the speakers because all of them have done an excellent job. Second thing I want to state was, now I was looking at the participants and I was feeling very happy, a lot of retinal surgeons are coming, but then I realized that the next session is about to start now. Otherwise, we don't have too many people in the uvea setup. So let's take the anterior uveitis. I think Dr. Chetan is not there. So the anterior uveitis, I think the questions we can direct it to Dr. Partho. Is he there?
Yes, I'm there.
Yeah, good. So in Dr. Chetan's talk, I think he did not mention too much about HLA-B27 uveitis. I think we need to know a little bit more about that. Could you throw us some more light on this entity, and how do we differentiate it between the virals, which you have mentioned? So what investigations do we need to do apart from HLA-B27 once it's positive?
Yeah, HLA-B27, mainly when a patient comes, the most important thing is to ask the patient that how recurrent he is getting the inflammation. And the second question is whether he is having any systemic issue like any joint pain, backache. So if these two are there, like backache and he is getting recurrence, then this is the time you have to send the patient to a rheumatologist, and you have to start the patient on an immunosuppressive. So these two are very important, sending a patient to rheumatologist and considering immunosuppressive instead of recurrent to use topical steroid for recurrent to control the recurrent inflammation.
Gaurav, you want to add something on that?
Go ahead to the rest of the questions because I don't think we have much time left.
I think a couple of questions on each of the topics. So, Partho, I wanted to ask one more thing. You had said that AC tap we should be doing for PCR to look for the herpetic group. So now, in a general ophthalmologist scenario, when do you think that this AC tap should be done?
Yeah, very good question actually, and I understand that sometimes it's not available also. So mainly, I'll recommend treating a case of viral uveitis based on the clinical suspicion. That's why there was only one slide on laboratory investigation. So if you can do better, you can't go for a PCR test, and that can be done anytime. When there is inflammation, you can't do a aqueous paracentesis and send the patient for PCR. So in absence of that, it's highly recommended that you examine the patient carefully, and whatever signs and signs we have described, based on that, try to treat the patient, give acyclovir empirically, and see the response. So it becomes very important that when you are suspecting or you are treating the patient with acyclovir, the patient is not responding, that time you have to consider CMV, because acyclovir won't work for cytomegalovirus.
Thank you, Dr. Partho. Samyak, in your talk on intermediate uveitis, just one basic thing. How do we differentiate between the cells which you said, no, retro-lentil cells? How do we differentiate that between the pigments that we many times see?
Okay, can you repeat the question? I just missed out in between. Sorry.
So in your talk, you said the very good feature, the clinical sign which we pick up for intermediate uveitis is the cells just behind the lens.
Right, right.
So many times these cells and pigment, we are not able to differentiate between the two.
Right. So pigmented cells and the WBCs, you mean to say. So pigmented
active inflammation versus an old one.
Right. So the pigmented cells will shine through. You can see, sometimes you can see the pigment actually there when you put a slit lamp beam and you focus it beyond the posterior lens surface. The other thing is that the pigmented cells will be significantly smaller in size than the WBCs. So that is one another point. The third thing is, if the pigment has been released, you can see either the release of the pigment from the anterior, the iris tissue, or it has to be somewhere from the posterior. So, for example, in like Dr. Partho said, you can see pigment release in viral uveitis. That is one of the clues that you may see an area of pigment release. So
one more thing that I can possibly add is that in pigmented cells, it's basically as a small cell with discrete boundaries. Whereas in active inflammation, there will always be like a halo around the cells, or there will not be a distinct border, which is what helps in differentiating between an active inflammation and when it is trying to become inactive and is.
Yes. The other thing is there will be clumps of WBCs. So cells like WBCs, they would tend to stick together, while pigmented cells will be solitary and they'll be floating on in the anterior. The other question, which I will just whether HRCT or CECT is preferred. HRCT does give a very good resolution. In fact, that will be the preferred investigation. But for us who are, suspecting something and looking for some biopsy or intervention, then a contrast enhanced CT is important, because if you get a HRCT and if you have a finding in that and you send the patient for a biopsy, inadvertently the interventional radiologist or the pulmonologist will ask to get a contrast enhanced CT, because that will help them visualize the nodes further to plan the biopsy. So just to prevent a double exposure to radiation, I personally prefer, and our pulmonologists do prefer to get a CECT done rather than an HRCT. I mean, that's simple.
Do we have a HRCT with contrast?
You can have, but then you will have to talk to the radiologist to be specific.
We can have the best of both the worlds.
We can have the best of both, yes.
Yeah, usually we can ask for a CECT with few HR cuts. That makes the trick.
Yes. That's the way.
Dr. Soumya Baba, do you think that HRCT, if we order, it can better rule out sarcoidosis also? So as a diagnostic tool, when we are still not very sure as the etiology, does HRCT have an edge over CECT in that, or not really?
I would still go for CECT, because that's what the pulmonologist told us, that that will be better in finding out. Especially, we are mainly looking for the lymph nodes actually, we rarely get to see active pulmonary TB or even healed fibrosis in these patients. I mean, fibrosis is still seen, but active pulmonary TB is rarely seen. So what we look for is the lymph nodes. It could be sarcoid, it could be TB, whichever we are finding. And apparently that is better seen with the CECT. So that's what I go for. Of course, the cost increases because of the contrast, but you have to explain that to the patient.
In your talk, you had shown us a beautiful case presentation where you had treated the patient only with ATT. So now, many of the times we are starting the patients on steroids. So if we are planning to start the steroids, what is your timeline? When do you want the patients to be on steroids?
Soumya, you need to unmute yourself.
Yeah. So TB as an infection is a little different from the other infections, common infections that we deal with, be it viral or say Toxoplasma retinochoroiditis. So here we would like, when we need steroids, we would like to start the steroids along with the anti-TB medication. The only exception would be, let's say, a tuberculoma with a large exudative detachment. You might start with the ATT first, and then if you do not see the exudative detachment resolving well, you can add the corticosteroids. But otherwise, my dictum, and this is not a hard and fast rule, is that when I see a focal choroiditis lesion, when I see a tuberculoma, when I see a retinal vasculitis with overlying retinitis lesions, I treat them only with anti-TB therapy, and we now have a big series to show that these work very well. On the other hand, in serpiginous like choroiditis, in granulomatous anti-uveitis, you have to add some form of steroid. It could be an oral steroid, it could be a topical steroid. And you start it along with the ATT.
Great. Are there any more questions to any of the panelists?
I think the retina.
If not, I think Rohit.
Yes.
Yeah, I was saying the retina people must be itching to get started now. Yeah.
I think we will bring this session to an end. Thank you, Rohit, especially thank you all the speakers, people who have attended this session, and Team MOS. And we hand it over to the next session.
Thank you all.
Thank you so much. Thank you, Rohit.
Thank you, Dr. Gaurav, and thank you for handing it over on time. I request you guys to please stay back. We are having an interesting session of videos. So please stay put, and it will be a good fun. Is Dr. Swapnesh here?
Yes. Yeah, I can see.
Yeah. So may I invite chairpersons Dr. Pramod Bende sir, Dr. Manoj Sasavde sir, and Dr. Nitin Prabhudesai.
How are you, PV sir? Nice to see you.
Unmute yourself.
Nitin Prabhudesai sir, how are you?
Fine. Pramod, please unmute yourself.
Yeah.
We can't hear you.
We can't hear you, sir. There is some. Are you on a headphone? You're on mute. We can't hear you. Yes.
So try log out and log in again.
So Manish, you can go ahead, introduce the session, why we made a session like this.
Yes. Good afternoon, everybody. So the session we are having is amazing retina roller coaster session. where we will be displaying 17 videos of various topics related to various topics of vitreoretinal surgery. This is, we have some of the excellent speakers from all over India and all over Maharashtra. So we have received some videos. So me and Dr. Swapnesh will alternately start the video, run videos from our screens. Those which do not have their commentary embedded in the video, we request the speakers to unmute themselves and give the commentary along with the talk. So to begin with, can I ask Dr. Sharang Vartikar to
share his video?
Sorry?
Dr. Starang can share his own video.
Yeah, he will share his own video.
I request everybody to stick to the time because we have 17 videos. So,
yeah, can you hear me?
Yes.
Right. So I'll be starting my video. Yeah, so today I'm going to talk about what happens when the nucleus decides to do a Titanic on us. Right, and I thank the MOS committee for allowing me this opportunity. I'm going to tell you a basic don'ts, right? So don't panic, that's the first thing, because it's not really the end of the world. Don't go fishing for the nucleus, because this situation can be salvaged. Don't try to force in an IOL, don't make it an ego issue. And don't push your VR colleague for an immediate surgery as if it's an ophthalmic emergency. What you should be done is do a good anterior vitrectomy, provided your cutter lying somewhere works. Place the IOL only if you are truly comfortable. Suture the wound. Counsel the patient honestly. I think we could have a whole talk on this topic itself, and then refer to your VR colleague. And what the VR surgeon does, I'm going to show you some videos to explain this. So the standard procedure is that we clean up the AC, we clear the anterior segment from vitreous and cortical material, do a pars pena vitrectomy, remove the cortex with the cutter, and then we use a phacophragometome to clear the nucleus. We lift it and bring it into the mid vitreous, and then we clear the thing. And finally, we place the IOL.
Again,
in general, if the capsular
automatically.
support is good, then we prefer to put in a 3-piece IOL in the sulcus. So that's about how the procedure goes. Now, sometimes if it's a soft nucleus, we can even use the cutter itself and do a kind of lensectomy rather than using the phragmatome. That prevents the making of a separate port. No suturing is required. It's a closed chamber surgery. With 20 gauge earlier, it used to be a lot easier, but even with 23 gauge, these things can be managed. One variation that can be done is initially after we have cleared the AC, at that time itself, we can place in the IOL. The advantage being that there will be a well-dilated pupil right up with a clear cornea, and the capsule is much better seen right than at the end of the surgery. And that gives us a more added confidence of putting in the lens the way we want. And of course, the rest remains the same. All the videos I have sped up in like 2x thing to try and fit it into the time. But the reason why we have this confidence of putting in the lens right up first, or even we ask the anterior segment colleagues to do it themselves, is that the modern phragmatomes are very capable of removing the hardest of nuclei. And the light pipe is a good useful tool as a secondary instrument. You can either hold the nucleus with the phragmatome and then cut it with the light pipe, or impinge it into the light pipe and then use the phragmatome to kind of fragment it over it. Or you can go directly bimanual by using a chandelier system itself. PVD induction, there are two schools of thought. If it's done right at the start, then you get a much cleaner vitrectomy, but the nucleus is then sitting right on the retina, and there are chances of bleeds or tears occurring. Some recommend the use of PFCL or visco in order to provide a cushion. And always remember to do a good peripheral examination at the end of the procedure. The other way is that we leave the vitreous behind right till the ends, which kind of gives a cushion for the nuclear fragments. And then we remove it, the nucleus up front. But the problem there is you are using a phragmatome with vitreous inside the eye. Finally, for the IOL implantation, my preferred practice is the posterior iris claw lens when there is no capsular support there. it works well in my hands. But of course, there are other options, all of which are equally good. So finally, in order to tackle the Titanic, the clues are try not to panic, take it in your stride, and trust your VR colleague to bail you out. Thank you.
Thank you, Dr. Sharang. That was an excellent presentation. Dr. PV sir, can you please make a expert comment?
I think sir has tried to log out and log in.
Okay. Next is
Dr.
Dr.
Vikram.
Vikram Mahenka. So he is there.
Yeah, he has already shared his screen, sir.
Yeah, can you see my slides?
Yes, yes, yes.
Very good afternoon.
Yeah,
Sharan, that was a wonderful video, very well edited video.
Thank you.
Thank you so much. And
Thank
over
you
to
so much.
Vikram. Looking forward, Vikram. Go ahead.
Thank you, Preetham. Very good afternoon, respected seniors and colleagues. Thank you, Sapnesh, for this
Can
opportunity.
you hear me in between? So I just wanted to know.
Yes, sir, we can hear you.
Yes.
Okay, that's fine. Then please go ahead. Then I'll mute again myself. Yeah.
I would be talking on IOL drop and in these different scenarios with the help of 5 short surgical clips and try to discuss some take-home points. So in the first scenario, it's an almost drop-like situation. Case posted for combined phaco with oil removal. And let's join the surgery at a stage where the phaco has been done, 3-piece IOL has been implanted, and there is Helon in the AC. And right now, a 25-gauge automated pars plana silicone oil removal is underway. After having removed the silicone oil, the ports will be removed. But prior to that, I make a small central posterior capsulotomy for a PC plaque. And now all that remains to be done is washing off the visco. And now some of it is hydro-expressed. And as you know, Helon comes off as a bolus. And now what happens is when I'm about to wash off the final visco, I enter with the irrigation. And because of the fluid gush, there is a posterior bellowing of the PC and the PC blows out. Now let's see that again in slow motion. I enter with the irrigation, the sudden gush, bellowing of the PC, and there's a large rectangular blowout. This is one margin of the PC. This is the other margin of the PC. And hence an almost drop-like situation. Thankfully, the 3-piece IOL stayed put there, stayed stable, and it all ended well. But take-home points to avoid such a mishap would be that you wash off the visco with a low bottle height, always have a high threshold for a posterior capsulotomy, and the pars plana infusion cannula should be the last to be removed because that would balance the anterior and the posterior forces and that would avoid bellowing. The next scenario is where the IOL is caught in the act of dropping. 2 small clips. In the first clip, the drop nucleus has been managed and the IOL implantation in the sulcus has been attempted. Now, without really confirming that the leading haptic is in the sulcus or not, the trailing haptic is dialed deep right onto the surface of the retina, and that's where the IOL lands up. The next clip is in the next clip, looks like a seemingly uneventful phaco surgery. Visco is being washed, and then you realize that the IOL is tilting, and actually tilts dangerously to the extent that the nasal haptic is almost hanging vertically down. And because one of the haptic is still anteriorly, it is decided to gently hook the haptic into the wound. Once that is done, the second haptic is also nudged above the iris. After that, a bimanual anterior vitrectomy is carried out with the help of a 23-gauge cutter. And after having ensured that all the vitreous is cleared, the IOL is dialed into the sulcus sequentially. And because of the haptics being bulky, a small PI is made with the cutter. So just the points here would be that you should avoid blind and violent fishing. If the visibility is poor and you're attempting IOL implantation in the sulcus, make sure you either use hooks or you use bimanual maneuvers, or maybe you could just inject it above the iris and reposition it into the sulcus sequentially. Ensure a complete anterior vitrectomy, and 3-piece IOLs are better suited for sulcus implantation. The last scenario is when you get an SOS call where the IOL has been dropped and is lying on the retinal surface. 2 small clips here. In the first video, the surgery begins with a standard pars plana vitrectomy where the aim is to free the vitreous all around the IOL so that there is no traction when you're handling or picking up the IOL. Once the retina is free and lying on the retinal surface, though I was planning to reposition the same IOL in the sulcus, I make a small clear corneal wound. Then I like to pick up the IOL, viewing it under a plano lens, because that gives me better depth, and I'm sure that I'm pinching the haptic and not pinching the retina. And once the IOL comes up anteriorly in the pupillary plane, through the main wound that I've made, I temporarily, with the help of McPherson forceps, exteriorize that haptic, so that way I'm sure that the IOL will not drop back again, and then I can sequentially dial it into the sulcus. The last video where there is a late spontaneous subluxation of the IOL and the bag complex on the retinal surface. Again, the surgery begins the same way. You complete the vitrectomy. Because I'm planning an explantation, I need a bigger wound, so I prefer a scleral tunnel in such a situation. I use a plano lens for viewing when I pick up the IOL and the bag complex. Hold it carefully, gently, make sure I'm not pinching the retina, get it up in the pupillary plane, and with the help of the left hand, I grasp the IOL and it's delivered out of the body.
नहीं मेरा fourth or fifth है, just it's a second going on. तुम लोग चालू करो.
So the last slide, the points that I would like to highlight is the IOLs per se are not as hostile. They don't go around slashing at the retina. So you can wait to ensure a good intraoperative visibility, achieve a good control of inflammation and IOP before you take up the case. See the IOP of all the surrounding vitreous. Use the viewing system as per comfort. Ensure that the wound size is sufficient. You don't want the IOL dropping down again and again, or traumatize the iris for that matter. And lastly, handshake maneuvers would ensure a cornea-friendly and controlled IOL removal. Thank you.
A lovely video, Vikram. Especially the handshake technique was clearly demonstrated. You can stop sharing. Bape, sir. So you're muted, sir.
Yeah. Next video will be by Dr. Ketan Jathar, and I'll be sharing his video. Just hang on. He's presenting on Yamane technique. Dr. Ketan.
Yes.
Can you please give commentary to your talk or your video?
Sir, the video is not seen. Your folder is seen.
One sec. I'll start again.
I am audible. Good afternoon. Am I audible?
Yes, yes.
Is it visible now?
Yes.
Okay.
Yeah, we have seen in previous videos the different techniques. Now this is a technique wherein there is a no capsular support. Iris claw is a good technique, but sometimes there can be some pupillary distortion. So nowadays I have shifted to this Yamane technique, which I feel is really a good technique when I have a more physiological position of the lens. There is no rubbing of the iris on the lens. So the markings are made at 2 millimeters posterior to the limbus and horizontal, and we use a 26 gauge needle to railroad the haptic. So once the 3-piece foldable IOL is inserted, ideally the haptic should open onto the iris so that it's easy to grasp them with a 23 gauge forceps. This is a 26 gauge bed needle which I've inserted. The first, it goes parallel to the limbus, and then there's an oblique entry to create the scleral tunnel. It is grasped with a 23 gauge forceps and it's railroaded and it is externalized. You don't need to push in so much. This was one of my earlier cases where, to be sure, I've pushed in quite, but if you just pushed in about a millimeter or so, you can still railroad it out. Once it is out, externalized, you keep another McPherson handy and use a heat cautery to create a flange at the tip of the haptic. The goal is that the heat cautery should be quite hot and you need not touch the haptic. Actually, it should be kept far away, slightly away, so that there is a flange created. And a similar procedure is then repeated on the other side. This is the tricky part because at this, when you try to railroad the grading haptic, there are chances of breakage. So it's always better to have a standby lens. And the similar technique is then repeated on the other side, and the tip is cauterized so that a flange is created. It's a perfectly stable lens. Once it has been centralized, you can just check for the centration and the stability. You can get a nice round pupil with a good centered and well stable lens. This technique can be done in variety of cases. Suppose the iris has been, suppose a traumatic hydrodialysis or there is loss of iris tissue. In these cases as well, where you can't do an iris claw, this technique can be used to fix in a lens. So this showing this another video. This was a case of a picture status post penetrating injury. Ideally, I feel when you're doing a Yamane SF scleral fixation, it's better to complete the vitrectomy. A standard past vena port vitrectomy should be done. A 3-piece foldable IL as it's used. We can even do this technique with using a 3-piece rigid lens. The earlier, with the first initial lenses which were there available, the 3-piece rigid lens can also be used. Only difference I felt was the haptics of these 3-piece rigid lenses are quite thin. So a foldable lens, I personally feel it's preferable. Some of the Indian companies have also come up with this 3-piece foldable lenses, especially Aurolab. I've got no financial interest, but I've done using that Aurolab lens and it's really good. The same thing, use a 23 gauge forceps to railroad the haptic and externalize it, and then use a heat cautery to create a flange. We need not touch the haptic, we have to just be slightly away. A similar process on the other side. There are a lot of variations which have been described now. Some people prefer to take out the 26 gauge bent needle right up outside the opening and then railroad it outside the wound and then draw it inside. So these are the variations which are being used. So once you tuck in the apis, you can see a nicely stable intraocular lens. Thank you.
Very nice videos, Ketan. I would like to ask PV sir. So what is your choice of secondary IOL in case where the capsular support is not good?
Yeah, can you hear me?
Yes, sir.
I still prefer suture one. The only thing is I switched to maybe Gore-Tex now because somehow I just have that feel because even if you are the knob you are cauterizing, it may remain subconjunctival and the long term, because here we are looking at 20 years, 30 years, 40 years. The chances of erosion, I feel, is always there. And this is just one single point contact, and the arch, the curve of the haptic is basically not designed for coming into the scleral tunnel, because it just IOL is gently pulled off and the tilt is, there's one possibility. But somehow the more concern for me is I have that knob eroding through the conjunctiva. Of course, maybe it's I'm overcautious. Down the line, maybe few years experience, when you have a 5-year, 10-year experience, probably I may just myself switch on. But as of now, I still have that concern. Somehow I feel a little bit uneasy leaving that knob just like this at a conjunctival level or a scleral level, I would say.
So no glued IOL also?
Not really. I not tried glued IOL. I still feel more comfortable with having an eyelet and going ahead with suture. But what Ketan said, I agree. All these IOL, I definitely would go ahead with a complete vitrectomy. That's no compromise on that from my side. And particularly vitreous around the IOL, if you left behind, particularly when you do just anti-vitrectomy, there are chances vitreous fiber can entangle through and can create a like a subsequent lateral on traction, and these eyes can come back with a tiny tears at the periphery.
Luckily, I haven't encountered a retinal detachment in the Yamane fixation. So
because you are a VR surgeon and you are doing a good vitrectomy. That's what I'm saying. What happened, unfortunately, when you have most of these eyes are having a clear vitreous, and when you do only anti-vitrectomy, you feel you have done good vitrectomy, but because vitreous is clear, it's very easy to miss some amount of vitreous around with most of these eyes. And being a trained VR surgeon, probably most of you will not have problem. I'm talking about person who's doing only cataract surgery and they do just limited anti-vitrectomy. even though you put a tricot in anterior chamber and doing anterior, because when you cut vitreous, this vitreous, more and more vitreous will start coming up. Some amount of vitreous will be stuck to the iris or pupillary margin, which sometimes very easy to miss. And round air bubble in anterior chamber does not ensure that there is no vitreous. Okay, so that's what. So the trained VR surgeon, obviously, you know how far deep you can go, and you can do a good job. If probably most of us still would prefer to go through pars plana because you are trained when you are not hesitant entering through pars plana. That exactly the difference between a trained VR surgeon doing the same technique and doing anterior segment, only anterior segment. Those are having a both. I think that's a boon if you have.
I have a technical question for Dr. Ketan.
Yes, sir.
When you cauterize, I'm sure the haptic could shrink, right?
Yes.
So how do you know how much to cauterize? Is there some kind of a mark you make on the haptic before inserting it, or is the IOL manufactured with a mark on the haptic? How do you know? Because you could have, suppose if the haptic shrinks unequally on both sides, can there be a tilt of the IOL?
Yeah, the first thing when initial cases, what I used to do was I was to touch the heat cautery to the haptic. So when we touch the heat cautery to the haptic, there can be more of shrinkage. So what I learned later on is that the heat cautery should be slightly away from the haptic, so that because the heat radiation, there's a flange which is created, the proper flange, and we stop at that, both ends. If we touch the haptic, it was a shrinkage. There had been a couple of times where I had to cut the haptic and again do a cautery so that I get a proper flange. So the trick is to keep the cautery slightly away from the tip, that only a flange is created and there is no shrinkage. And tilt, yes, sometimes I had surprises in my initial cases where there was a bit of tilt, but slowly, maybe after the learning curve, you can adjust it and tighten it accordingly, and the tilt is less. The only thing I noticed is you do get a hyperopic shift in these cases. So presently I've started using a slightly higher powered lenses so that the refractive error, post-refractive error is a bit less.
Yeah, the key here is, if you look at it, there is only one point of contact. So you go through the sclera, just one point of contact, and IOL can't wobble. So what is important here is you have to go exactly through the sulcus. So sulcus acts as a platform where your haptics are stable. If you are posterior to the sulcus, then there is a chances of tilt. So your measurement has to be very, very precise. And as a kid, if you remember, Preetham, to answer a question once again, as a kid, remember we used to do some plastic wire, cable or wire, something, filament, you use a heat there, it just retract and melts and form the knob layer. Something like that, that's what exactly happened here. It holds on.
Thank you, Dr.
Thank you so
Ketan.
much.
Thank you.
Next, we have Dr. Shamik Mukadam from Nagpur. He will be talking about vitreous hemorrhage, vitrectomy for vitreous hemorrhage.
Vitrectomy in vitreous hemorrhage is like a Pandora's box. The first thing that we have to secure is the infusion port. And once we have put the infusion port in place, we need to check whether the infusion port is well into the vitreous cavity. I generally follow doing the retrolental vitrectomy post putting the ports in place. Once the ports are in place and retrolental vitrectomy is done, I proceed towards doing the peripheral vitreous cutting and the core vitreous cutting. While doing the peripheral vitreous cutting, I take care that I don't cause any retinotomy in the peripheral retina, and also I try to do the peripheral subincisional, that is the vitreous posterior to the ports, in a detailed manner. Then I would proceed with doing a fluidary exchange followed by endolaser. Once endolaser is done, I would do a gas exchange. However, as I said in the beginning, that vitrectomy for vitreous hemorrhage is like a Pandora's box. So sometimes when we
Hello.
we're doing a retrolental vitrectomy,
Can you hear?
we end up seeing something like posterior highlight phase with a pre-retinal hemorrhage. In such cases,
We can hear, but the volume is less.
taking care that we do not induce any posterior retinotomies. While doing this, we have to cut around the fibrous membranes and gently clear the posterior vitreous and do a good vitrectomy after we have done a PVD induction. Once we have done a good vitrectomy, we then proceed to do clearing up all the pre-retinal hemorrhage, and then hemostasis is achieved by doing endodiathermy. This is followed by fluidary and gas injection.
That's a video, Dr. Shramik.
Yes, sir.
Any comment, Dr. Prabhudesai, sir? One quick comment.
Shamik, I have seen that in your video, your pressures were all around 40 plus always. So do you keep your intraocular pressure on that level every time for all surgeries?
Sir, actually it's around 35 to 40, but sometimes the machine doesn't calibrate it properly, and then I have to keep it on a higher level because it keeps on oozing otherwise.
Okay.
So that has been there. Now I've started keeping the infusion while the machine does the calibration. I've started keeping the infusion port on a higher level. That's what was told to me by the engineer from Alcon. That keep it on the higher level so that it calibrates properly.
One trick would be to keep looking at the optic disc. In the initial phases, you can keep the pressure on the higher side and see, because I have seen in some patients, especially diabetics, it starts pulsating even at 25. So that would be a clue before we go ahead and set the pressure. If you see that there is pulsations coming at lower pressures, you have to still go down. Maybe
the only issue is here because all these diabetic, we know it is a compromised circulation for optic disc also, not only in glaucoma. And with added laser, quite some time we really wonder, intraoperative everything was fine, preoperatively everything was fine, and post-operative period we see why disc is pale. Probably with keeping this pressure continuously high, and again we raise higher level if it bleeds inside, and if you're keeping it longer time, I think that again compromise the optic nerve circulation significantly. And then in spite of doing excellent surgical intervention, surgery, and everything goes well, final outcome may be not as good as you desire. That's the thing. I also have a question for you. Why you injected gas? Was there a break intraoperatively?
No, sir, but just to be sure that there are no more bleeds at the end of the surgery, we do gas injections.
So there's a saying, if all of you read that Steve Charles' book, first thing, no break, no gas. Okay.
Okay.
Next, Dr. Manoj Sasawade sir will be sharing his video on TRD, tractional retinal detachment in diabetic.
Manish, what I'll advise is when you are taking comments, you can ask the next person to start sharing so we can gain on time.
Okay.
Yes.
Dr. Sasawade sir.
Am I audible?
Yes.
Yes. Good afternoon, everyone. Especially PB sir, in front of him, I am presenting today his very favorite topic of advanced diabetic eye diseases. You know that there are various presentations of the advanced diabetic eye diseases in which you get various types of uveitis, tractional detachments, sclerosed vessels, optic atrophy, and the neovascular glaucoma. What is important is that in these cases, if it is an advanced ocular disease, it is also advanced in other organs also, like in cardiac as well as the kidneys, and patients might have the amputations. So thorough evaluation, ocular as well as systemic, is warranted. Particularly
not able to see video. Is it video not shared? Not able to see the video? Manoj?
It is not.
We can see you, but video is not there.
Is it not seen?
So your folder is seen. So you have to share the video program, whatever your VLC player.
You just double click on the video, sir.
Okay, 1 second.
Yeah. No.
Is it visible now?
Yes.
Yes.
Okay. So the thorough evaluation, including visual acuity, particularly in such cases, most of the time there is no visual, no light perception, and one most important thing is missed is rubiosis. Those things are very important. Apart from that, you have to do the OCT, ultrasonography, and thorough counseling is important. Where the important thing is that promise less and deliver more. Preoperative laser and vitreal anti-VEGF helps a lot in cases of advanced diabetic eye diseases. One has to stop anticoagulants 3 days preoperatively. The various types of manifestations, you can see that as large TRDs, and in such cases you have to give less promises. You can see this fluorid rubeous neovascularizations on the disc. Elsewhere also, these cases really need intraoperative, preoperative anti-VEGFs. Here you can see the subretinal hemorrhage, exudation, subretinal glyosis, various types of, this is a combined detachment, and thick, dense fibrosis with the apart from that, you can see cupping, sclerosed vessels. So other eye status is also important, so that you can get little bit of boldness to do the surgery during the surgery. And the most important thing, that principle is that you have to trim the peripheral vitreous. The principle is to go from posterior to the anterior or outside in. Most of the times, the fibrovascular membranes or TRD is attached on the posterior pole. So when you go from the anterior to posterior, most of the traction is relieved and you get the proper plane. Here you can see that the 360-degree mid-vitreous PVD is usually present in such cases. It is trimmed 360-degree from anterior to posterior or outside in, and then you can get a good plane to do the surgeries. These cases are highly vascular, and there are various types of attachments, and most important thing is that you should go into the right plane. If you are not in the right plane, you can go cause the more tears as well as the bleeding. But if you are in the right plane, you can see there that you can get a good plane, and your dissection time is also less, and bleeding is also less, and you get a good result. Here also you can see that if you have to spend a little bit of time to get a proper plane. Once you get a proper plane, whole membrane comes out very easily. Don't hesitate to use the scissors as well as the forceps in such cases. Otherwise, nowadays there is a fashion of using 27 and 25, and they don't use the scissors. But whenever it is required, you have to use it. And here also you can see that the pressure, whenever there is a bleeding, you can increase the pressure in intraocular pressure instantly to stop the bleeding. Here also another case of getting the right plane dissection, so that there is less bleeding as well as less number of iatrogenic breaks. Another thing is that you have to understand the anatomy of these membranes. These are pinned to the retina, and this can be pinned or can have a broad attachment. You have to lift the retina a little bit with the suction, and in between space, keep on dissecting either with the cutters or with the scissors or forceps, and you get a good plane. This pinning or these attachments need to be isolated, separated, and trimmed one by one. And whenever it is required, you can go for the ILM peeling also. These are the broad attachments, and these attachments are near to the arcade or large vessels. So there is high chances of getting the bleeding during the intra-op. So you should be prepared to increase the intraocular pressure instantly, so that you need not have to stop during the surgeries. Then delamination and segmentation is also important with the suction. And beforehand, you have done the proper laser, and in intravitreal anti-VEGF is given, you get a good avascular membrane, and you can delaminate and then segment it with the cutter itself, and you don't require any scissor or forceps. And whenever it is required, you can see that a crumpled ILM one has to remove it. Usually in some cases it comes out in a piecemeal, then you can remove it at the time of silicone oil removal. There are certain surprises also you can get in a vitreous hemorrhage cases. You can come out, encounter with the sclerosed vessels, all 360-degree or all over the fundus sclerosed vessels, and dense fibrovascular sheets are attached on the disc and near to the macula. So such types of cases comes, then in beforehand counseling helps in giving the patients good outcome or to avoid the medical legal cases. These are the cases where you have to work in a small people also. They are resistant to dilate also. And once you have a good visualizing system, then it is possible to do the good dissection through the small pupil also. You don't require any pupil dilator. So the thing is that take home message is that if you are in a right case selection, then there won't be a problem. You should be right pre-operative preparation should be there. Right equipment you should have. A right plane of dissection is very important. Right time of hemostasis instantly is to be achieved. Right time to convert the bimanual is also important. And right vitreous substitute should be selected depending upon the patient general conditions. You have to put most of the time oil, even though you don't like, but the patient can't maintain the supine position, prone position most of the time because of the general debility. And you should have a good equipment like which are having the wall cannulas and pressurized intraocular surgery system. Thank you very much for patiently hearing.
Thank you, Dr. Sasode, sir. We have 2 more videos related to this, so we'll have discussion after that. Can I request Dr. Swapnesh to share his video, please, on combined retinal detachment? Dr. Swapnesh, please go ahead.
So can you see my screen?
Yes, yes.
Yes, we can.
Sorry. So thank you, MOS, for this opportunity. Combined retinal detachment is always a difficult case to approach to, and the main aim is to enter the posterior hyaloid space. So once the posterior hyaloid space has been entered, you can either induce the PVD from disc to the periphery or from periphery to the disc. So in this case, particularly, I am using forceps to try and induce the PVD. We all know there can be vitreoschisis, so we have to be very vigilant about a second membrane. And with the modern cutters, with the optimization of the cutter port, we can really dissect around the epicenters of the FPP quite easily using a combination of a delamination as well as a desegmentation. With the modern machines, with the raised IOP control in the foot switch, we can always raise the IOP for 1 or 2 minutes to catch the bleeder. In this particular case, I have used a pre-operative Avastin so that the intraoperative bleeding is much less. You can also have a rollback technique where you sit on the FPP and with vacuum control, you can actually see the FPP folding onto itself and entering the cutter mouth. Also important to dissect in all 360-degree areas because the hyaloid may be stuck somewhere and may be flush to the retina somewhere. So it is very important once you get a plane, try to go all around it. We should not be satisfied, especially in a combined RD, just to dissect the FPPs. We should also look for this thick SRF as well as if there is any underlying subretinal gliosis. Like in this particular case, there was an underlying subretinal gliosis, and try to make the retinotomy port in such a way that your dominant hand is in a good direction to pull that SRG out. Even if you are managed to cut that SRG, it will help. You can do a fluid-fluid exchange. In this particular case, had a redetachment only inferiorly, rest of the retina was all at. So we attempted to go in, and this time I tried to do ILM peeling and also peeling of the fine membranes over the area of the elevated break. So as you can see, I don't attempt ILM peeling in each and every case, but wherever it comes out easily and one piece, then in those cases I can do that. So this was the post-operative result. So one more case, if my indication to put a BB is if the CRD is beyond the equator, then I don't hesitate and put a band buckle in all my cases, provided the SRF is extending beyond the equator. So similar technique where we are trying to go around the epicenters, but sometimes the retina is very mobile, and in spite of all the efforts, it may be difficult to really tease off these membranes. So the PFCL comes to our help in these particular cases. It acts like a third hand and keeps the posterior pole behind. also stabilizes the retina so that we can do the same maneuvers all over again. So in this video, you can see that once the PFCL is put, the dissection is much more easier and the retina is much more stable. Also important to use your left hand. You have to be ambidextrous because you have to dissect the PVD without causing many iatrogenic breaks with your left hand also. So again, this particular case had a SRG which is looking very small to start with. And once I start pulling it out, you will realize that how large the SRG was. I'm using a max grip forceps to pull it out, and it's really a huge SRG, which didn't seem so. So preoperative evaluation is very important, and I still do my own B scans for all vitreous hemorrhages, be it CRD or TRD, to plan my surgery. This surgery is ended with oil in situ and with adequate endolaser done around the breaks as well as to the anoxic area. Thank you very much. It was a pleasure to present in front of PB sir and all the seniors.
Yeah, excellent, great video, Dr. Swapnesh. Next, we have Dr. Mandar Zogrekar talking about combined phaco and vitrectomy for diabetic patients. Dr. Mandar, can you please share your video?
Yeah. Can you see the video? Hello?
No, we can't see it yet, but I think your screen is coming up.
Yeah, I can see the VLC. Huh? So,
okay.
You can see.
Can see, yes.
Yeah. So, cataract is a common complication in common accompaniment in the diabetic cases. And one of the things you have to do is a lot of times you have to do the combined surgery for these cases. One problem we can enter, and we are very much have a problem, is that there is no glow when you do the phaco. For that, what we use is the moonlight phaco, where we use put the one trocar and put the chandelier light, and you can see the moonlight effect from this, where there is no glow in first side, and left side you can show there is a good red glow. This red glow allows you to do the good rexis. You don't have to stain the capsule just to see it because of the red glow coming of the chandelier coming from that. You can see the capsule well. You can complete your rexis without any staining. Then you can continue your phaco, and you can see your lens well. You can, one flip side of this putting chandelier is that there is a surge and scleral elasticity is reduced. So there is some amount of surge there. You can continue by switching off the microscope light as well, and you can see very clearly the light shines through the lens and the matter, and you can see them very clearly. And you can do your phaco. Most of the times the nucleus is very soft in these cases, so you have to do a piecemeal approach sometimes. So we continue the phaco like that. Once the phaco is complete, cortex is removed, we put down the lens. You have noticed I have put the trocars first before starting the surgery. That helps me not putting the sutures to the main tunnel when I start the phaco or when I start the vitrectomy. So once the lens is in situ, I keep the visco in the AC itself. We remove the. We are using a beveled cutter here. We start to remove the dense vitreous, dehomogenized vitreous hemorrhage, and then we reveal the. You can see there is a tough hyaloid membrane, and we are not getting any purchase to the posterior membrane. So we go in the magnification mode and we nick the membrane and peel it over, so that we have got the posterior membrane now, and we can enlarge this. We go back. Now, without removing the blood too much, we go back and enlarge this opening of the hyaloid and try to open it on all the sides. Remove the vitreous, remove the membranes in the place, and try to create a PVD. In this case, it comes out easily on the inferior side because there are no nails. superior also, then we go and try. I have not still removed the blood because it helps me to give a contrast for the membrane. As you can see, these beveled cutters are very much of helpful. You can go inside beneath the membrane and cut them, segment them very well. Then we remove the other membranes. We hover over the membrane so that it comes into the port, and we remove them. After cauterizing the bleeders, if any, we remove the blood with the cutter itself, and laser is applied to the areas where there is no laser done. We take special care around the diathermy sites. And once the laser is complete, you can get this view. I can compare with the intraoperative pictures, what was before and after. Thank you for your hearing.
Thank you, Dr. Mandar. That was excellent video. Can I request Dr. PB sir to have a comment on diabetic vitrectomy in 1 minute, and so we can proceed.
I think all three videos are, I think, exceptionally good. I think very well presented, and I'm really proud that everybody is doing really good job. The only concern, what I would like to say here is, even they are small, tiny bleeders, never take them lightly. Particularly, just take care of that bleeder then and there only, because we don't realize, we think it's a small bleeder. Over a period of time, as you complete vitrectomy in other part or peeling other part, this will be small, oozing slowly start pulling up, and then form a nice layer between retina and the remaining membrane. And then once it's clotted, it's excellent glue, and then your life can be extremely difficult when you peel. So best way, just to take care then and then only. Easiest way, you just raise little bit bottle height and stop your vitrectomy. What happens when you have turbulence of fluid? This blood is getting washed. You are not giving time for it to cauterize, to clot. I mean, if a small user, if you just hold on for a while, let I be stable like this, bottle height little bit on higher side, even 35, 40, just few seconds, and it stop on his own, and then you proceed with the surgery. But if you even raise bottle height and you keep on continue vitrectomy, because the fluid is flowing, turbulence is there, this blood keeps on getting washed, not getting clot, so it keeps on oozing long time. So take care of bleeders, then and then only your life can be much easier. And others, I think they are describing everything very well. Actually, all the techniques, I really are happy with that.
So can I request you to go ahead with your presentation, sir?
Yeah.
Share the screen, please. Next up is Dr. Aditya Kelkar after sir. So
I would take this opportunity on behalf of MOS to wish PB sir a happy anniversary from entire MOS team. And thank you for sparing your time on this day.
Thanks, Swapnesh. Thank you so much. Can you see the video?
Yes, sir.
Yes, I've just make it full screen. And just basically, Swapnesh wanted this coloboma with a detachment. There are a few just catches here. As you see, you can get away without removing the lens. With our experience with lens sparing vitrectomy and ROPs, we realized previously dictum was all the coloboma lens has to go. It's really not necessary. And you look at the anatomy, probably corneas are small, but pars plana may be exactly same. In fact, maybe even further away. So safely you can make a sclerotomy without touching the lens. So you can go ahead. This is a 23 gauge. Almost all these eyes, I prefer to use a bell buckle. And the important thing is most of these eyes are young, so, and extremely difficult, and then almost never they have PVD. So and the PVD induction is the key for a success, and it takes a hell lot of a time, extremely difficult to induce. And as you see here, totally detached retina, posterior hyaloid is stuck, and when you try to induce along with the posterior hyaloid, along with the vitreous, retina start jumping up, and there's a high risk of creating iatrogenic problem. So debulk the vitreous, and probably now we are comfortable with the tricot. So as much as debulking, if you're doing carefully, use a high cutting rate and low suction. And as you see, vitrectomy being completed, and I use a tricot, but in spite of tricot, and as you see, this will show you when you try to use the suction, There's no fixed rule where you should start, but probably avoid traction along this coloboma margin, because that can sometimes create a break underneath here. But if you see us try to pull it up, the retina is getting pulled, as you see here, and retina being more mobile, and you try to keep on cutting very close to the retina, and port is facing retina, you just see few seconds and you say what is going to happen now. Okay, just trying to keeping port away, but sometimes you cannot catch and vitreous properly, and if you do go turn the port around, and this is what you see. What happened here is, see, part of vitreous has come, but this is what happened now. I was fortunate enough that managed to get without break, but you see there's a pinching here. You can assume that, probably take it for granted there is a break, even though it was not obvious, but so don't miss that point, laser it at the end. This is not one, and once as you start pulling the vitreous, retina become more and more vipile, and sometimes even then things become a lot of more difficult, because retina is coming anteriorly up. If it's a partial RD, become a complete RD, and then you have a less space to work around. And as I'm using light pipe to press the retina back and holding the vitreous, and this is another, I created break here, the little share. This is what happened again. So what happened, you use a bimanual, in the sense, suck the vitreous in a port and hold it there, don't pull. And then you press the retina back, and that's way slowly helps you to separate entire vitreous. This I cauterize the break because the bleeder also, and that will help me to identify as well. And once you ensure that entire vitreous is removed, okay, you do not know a original break where is. Sometimes there is, you can identify within a coloboma area. This was a break iatrogenic that helped me to drain SRF in this scenario. But sometimes breaks will be always along coloboma margin, and it is difficult to identify. And that's the reason we generally do laser all along coloboma margin. This is what FG being done. I drain through this break. You see how deep the scapuloma is. You can still see age when with detached retina. So fortunately, because break was a little bit inferior and posteriorly, it was easier for me to flatten whole thing off, try to drain SRF within this coloboma area. There is no need to remove entire, if there is intercalary membrane, there is no need to remove actually it is, because sometimes large blood vessels running across it and you may damage those, and it does not cause much distraction. And as you see here, all along the edge of coloboma, this break is treated. That other contact point also a laser, and all around 360-degree laser, and that's what we're done with. Yes, back to Manish.
Yeah, so thank you for the excellent tips. We are slightly running short of time. So can I request Dr. Aditya Kelkar to please share his screen, and we can go ahead with next presentation. So after RD videos, we can have a small question answer or whatever comments. Dr. Aditya Kelkar, you are there?
Yes, I'm there. I'll just share the screen. Just let me know if you are able to see the video.
Not yet.
Not yet.
After Dr. Aditya, we have Dr. Shilpi Nanavre from Dr. Bhavan Kule's hospital. So,
now, are you able to see the screen?
Yes.
Yes, sir.
Yes, no.
And the video is also visible?
No, sir. We can see your folder.
Not yet?
We can see
Dr.
your folder.
We can see the folder.
And can you see now?
No.
No? I think you can take the next speaker. In the meantime, I'll just sort out what.
Dr. Shilpi, are you ready?
Aditya, sir, you can stop share.
Yeah, I'll just try once. See if it works.
So try sharing the player instead of the folder.
In the meantime, I will play Dr. Sharad Bhomat's video.
Okay, right. I'll just stop sharing here.
Yeah, please. Dr. Aditya, can you please stop sharing the video? I'll play Dr. Bhomat's video. Dr. Bhomat, you are in there. You are there.
So please
I'm there. I'm there.
Yeah.
It is inbuilt in that.
Yes, sir.
Now visualize
Is there some problem with the audio?
You can speak. So
Yeah.
you can speak as the video is being played.
Yeah. So this is the hyaloid which is being removed, the temporal part, and we're trimming the hyaloid now with the cutter. Basically, the aim is first to relieve the epiretinal traction completely, and once that is done, we inject PFCL now after maximal possible relief of the epiretinal traction. We could visualize a macular hole now, and what we are doing is we are staining the ILM under the PFCL with the brilliant blue, and it provides very good counter traction and good contrast when we stain under the PFCL. So you can see that once we have flattened the retina, it is easier to peel. In between, we can restain the membranes, and you can see that once the traction has been completely relieved, the hole is closed. So now you can see the hole is closed. So the next step is now we go to the periphery and do the peripheral vitreous base removal. Again, your PFCL helps you to stabilize the posterior pole, and the peripheral retina flutter is less. So once you've completed the vitreous base removal, scan the retina, entire retina for all the breaks and mark them with the endodiathermy. At this point of time, we felt that we need to put a encircling element, and that we have done, because sometimes in such cases we end up doing a retinectomy, so we didn't do it pre-operatively. I mean at the beginning of the surgery. So after placing the encircling element, now we are doing the fluid air exchange and draining the subretinal fluid through the breaks, and we proceed to do the laser. Generally, I prefer to do the laser only to the breaks, and only in some exceptional cases where multiple tears are there in all the quadrants, I will do the 360-degree endo laser. Most of these times, we need to inject silicone oil in these patients. Though sometimes with superior breaks and superior PVR, we can do away with the gas. So you can see this is before the surgery, and this is post-operatively. You can see a well-attached retina. The hole is closed, and we could achieve a vision of 6 meters after silicone oil removal, which was done around 5 to 6 months. So the most crucial steps for PPR management are to ensure the complete hyaloid removal by repeated triamcinolone staining. So if you detect the vitreous cases, all the membranes, then you can be assured that there will be no reproliferation. Adequate traction relief in the form of membrane peeling, subretinal, epiretinal, and retinectomy when it is required. And we need to place the scleral explant, whichever type is needed according to the case, especially in case of inferior PVR, inferior breaks, and posterior migration of the vitreous space. Again, to re-emphasize, we need to treat all the breaks, find them. Post-operative management is equally important in these cases. The management of inflammation, the glaucoma, the reproliferation, and the exact time of the oil removal. Thank you.
Manish, I'm ready if we can just try it once.
Yeah, Dr. Aditya, please go ahead.
Ready?
Go ahead, Aditya.
Yeah, are you seeing the screen or no?
Yes.
And the video is there. I mean, the title of
Yes. 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 You can share your video also, so we can see you.
Yes. Now I'm sharing the video. I don't know if you are able to see it.
Yes, we can see it, but we can't see you.
Okay. That's all right. As long as you can see the video.
Yeah.
So this is a chandelier-assisted scleral buckle. This patient had a slight amount of cataract, inferior vitreous hemorrhage, and the media was slightly hazy. And therefore, I thought that this is the ideal case for doing a chandelier-assisted scleral buckle. So first of all, I isolated all the muscles. I use this chandelier in the inferonasal quadrant here because the break is in the inferotemporal quadrant.
Chandelier assisted scale.
So I mark the break.
I could have done a radial buckle here, but I wasn't very sure about the adjacent areas because the media was not very clear. So I did a segmental buckle in this case. So this is cryopexy being done. And 511 sponge, that is 7 millimeter, is passed segmental between 6 o'clock and 9 o'clock. Just confirming once again the placement of the suture. So you are seated comfortably, you are not changing your head gear because the indirect ophthalmoscope is not there right now. And so it becomes quite a comfortable surgery to perform. At the same time, the fellows and residents who are assisting also get to know what exactly is being done inside the eye. Confirming the indentation effect, you can see the amount of vitreous hemorrhage the patient had, and it would have been very difficult to do scleral buckling using indirect ophthalmoscope. So in the end, removing the chandelier, cutting the threads which isolated the muscles, and then the peritomy is closed. And this is the buccal effect that you see post-operatively and attached retina. Thank you.
Excellent video, Dr. Aditya. You make it look very simple, though obviously it is not. Next, I will request Dr. Shilpi from Sarakshi Netralaya. She is presenting on behalf of Dr. Bavankure, who could not be here with us because of some preoccupation. Dr. Shilpi, can you please share your screen?
Aditya sir, you need to stop sharing your screen. Is my screen visible? Hello. Is my screen visible?
Yes, madam, your screen
Yes.
is visible. Please go ahead.
Yeah, thank you. So good afternoon, everyone. I'm sharing the top 7 pearls for managing case of GRTs. As we all know, it involves more than 3 clock hours with edges having tendency to roll or curl back. Yes, GRT is definitely a challenge as much to a novice surgeon as to a veteran. Attaining PVD is one of the most critical steps. Luckily, in most of the cases, it is present. But yes, challenge in this case was because it was a closed funnel case and the ports were coming into the cutter during the active suction. So the tip here was to create the space by training the SRF, avoid active or passive suction, and use forceps to induce the PVD. You can also create space by putting a small PFCL bubble over the disc. Another important step is base excision, which has to be done at high speed and low vacuum. Base excision has to be done after supporting the posterior retina with PFCL and also by depression. Phakic base excision can be done by bringing the ora into the mid-vitreous cavity so that you don't have to cross with the cutter and touch the posterior capsule. Anterior flap excision is a devitalized tissue which acts as a scaffold for anterior loop PDR. To remove the last bit of the tissue, you can also depress in that quadrant, and it also leads to the base excision in toto in that region. While unfolding a retina is another pearl, one needs to open the funnel and the collapsed retina with the 2 instruments. And then you need to put the PFCL as a single bubble over the disc, slowly watching for the disc pulsations and also for the opening of the peripheral funnel. In this video, the peripheral edge of free flap did not unfold with PFCL. So you have to slowly stroke or massage the fold with closed blunt tip forceps or the soft tip cannula so that it can be unrolled. After flattening, you need to laser You have to laser the posterior edge of the GRTs with the laser burns, which are almost confluent in 3 to 5 rows with moderately intense burns. Important is to specifically take care of the horns of the GRT, where you should put additional rows of the lasers. If any residual anterior flap is left, you should not forget to ablate it. The most important step is to prevent the slippage of the posterior flap. To prevent the slippage, you have to avoid the hide and broad buckles. Put PFCL to the posterior edge of GRT and milk out the last drop of SRF during PFCL air exchange. Less than 90 degrees GRT, there is hardly a fear of any slippage. Remember to milk out by repeatedly tapping at the edge of GRT. The more you spend time in this step, less are the chances of the slippage. Also, the 360 degrees GRTs does not warrant PFCL SOI exchange if meticulous tapping is done during PFCL air exchange. GRTs can be associated with concomitant pathologies like epiretinal membranes, FTMH, and severe PVR. In cases of severe PVR, membrane peel has to be done with forceps or with the vitreous cutter in the shave mode. Who says 360 degrees GRTs make your life difficult? Here, intentional retinectomy is done to convert a 180 degree GRT to 360 degrees GRT to relieve severe peripheral contraction and PVR and intrinsic contraction of the retina so as to facilitate the flattening of the retina. So friends, follow the tips and hope it will bring smiley on your patients and your face as well. Thank you.
Thank you, Dr. Shilpi. Excellent presentation. I will be sharing next video from Dr. Rupak Kanti Biswas. He is from Calcutta, and this is for macular grafts in failed cases. This is a ASRS Red Buckler Award winning video. Please enjoy.
Manish, this video also, I think Uretina also is award winning video it was.
Yes, yes, sir, it is. Rupak, you are there, right?
Yes, I'm here only. I'm here only.
Welcome.
Anteroposterior and horizontal traction by ILM peeling is standard method of treatment in full thickness macular hole. But it can fail. You close the hole like this. Inverse flap technique for larger holes can improve anatomical closure.
This is actually when you fail, and if you don't have, then you have to put a free ILM filling flap there. But what we noticed there, probably, I think you are hearing me?
Yes,
Manish, you are
very clearly. Yes, we can hear you.
Yes. So
would you like to talk? Rupak, I can mute this.
Sizes. The hole size is measured.
No, carry on.
Previously failed macro hole surgeries. 100 micron bigger.
Yeah, but Manish, in
Manish,
between
when
the
you're muting yourself, the video is getting muted. Manish, don't mute yourself.
Can you see Can everybody see it?
With the help of a Caesar, while the graft is being held.
Yes.
Very, very clear. Yeah, very clear.
Fluid air exchange done, and graft tissue is placed gently over the macula with photoreceptor side down. Gently tucked. And residual fluid taken out. Donor side lasered. Vitreous cavity filled with gas. Postoperatively, wow, amazing. Nicely taken graft, filling the gap properly with somewhat similar architecture of graft tissue and surrounding neural tissue. To know the graft function, I did multifocal ERG. Amazing near normal macular spike is seen in our grafting cases, which is not seen in other scenarios. We did Humphrey visual field 10-2 as well, which showed near normal foveal threshold of 25 to 27 decibels. Literature says few surgeons have tried larger retinal graft in failed cases without refination where there is no end-to-end touch. In our method, predetermined size of retinal refination done and tucked into the hole to have end-to-end touch. And we hypothesize that an aberrant regeneration takes place at this graft host junction. To prevent any hindrance into it, we did not use any viscoelastic agent or liquid perfluorocarbon. Till now, 7th eyes of macular grafting is performed in failed macular hole surgeries only. Well taken graft in 70% cases and visual improvement in 40% cases were observed. The key success here is harvesting exact size of retinal graft by trefination and not using any substance which can hinder end-to-end touch. A larger series can open up a new horizon of retinal grafting.
Brilliant video and brilliant device, Dr. Rupak. Is it available now for everyone?
Yeah, this is available with Epsilon. But the only thing is that there are two things. Most important is that you need to measure the size of the hole and then increase of 15% of it. So the series what we have from 400 to 1200 as of now is available. So take the nearest one. So for example, if you have a 600 of size of the hole, and then 15% is around 90 microns. Again, you add it up. So you take 700 size of the trifine and then take it.
Okay, great.
That is the
Thank
calculation
you.
we need to do. Yes.
Thank you. Dr. Swapnesh, I think you will share videos.
Thank you all.
Oh, it's brilliant. Brilliant. Great technique.
Thanks, Manisha. This is an excellent program going on. This is a world class, all videos. Thank you.
Thank you. Thank you. Dr. Swapnesh, can you please share video from
Dr. Nitin Prabhudesai sir is there? Dr. Nitin Prabhudesai sir?
Yeah, I'm there. I'll share my...
No, I've already shared it, sir.
Okay, fine.
Yeah.
So this...
You can speak over.
This is a case of double staining. We have first we are removing the apparent membrane, and you can see that it comes off very smoothly. That means it is not the ILM which we have been rasping. So after removing that membrane, we restain, and after restaining, we really see that we are getting the ILM now. So and whenever you are in doubt that what you have peeled is ILM or not, you can always stain again and check. That's what the message we want to give through this video. Thank you. It is a short one just to show the double staining technique.
Is the video that was
Yeah, it's I just had to show this.
So can I share Dr. Madhusadan's video in succession?
Yeah, please go ahead, Swapnesh. Then we can discuss.
Madhu?
Yes, Dr. Swapnesh, I'm there.
Yeah, please give your commentary.
I can't see the video, Dr. Swapnesh.
Okay.
You need to stop sharing this and again go back.
Yeah. Yes.
Yeah, I can see the video now. Good afternoon, everyone. I thank the MOS for giving me this opportunity. I am going to be talking about vitreomacular traction. It's a tug of war between the vitreous, which is pulling the retina in the anterior posterior direction. Case one is a scenario where we suspected a potential deroofing. So this is how we start. I always prefer to do anterior vitrectomy under direct visualization, follow up with a core vitrectomy. In VMT cases, I do prefer to put IVTI because it gives me a clear demarcation. And while inducing the posterior vitreous detachment, it has to be very gentle and in a very limited manner, so that you have to try not to move across the fovea at all points of time, so that you do not deroof the thin layer of the vitreous slash ILM over it. Now, if you notice in this case, the vitreous is in fact, even though we are careful, it does tend to go across the fovea, and therefore you'll see a change in our approach later on. So what has happened, it's just crossed across the fovea, doesn't matter. We are just going ahead and inducing the remaining part of the posterior vitreous detachment. This is a complete posterior vitreous detachment with the help of the microvit. You finish off the vitrectomy. Now in this case, because we suspected that we may have caused a potential deroofing, we are going to stain the internal limiting membrane with the help of brilliant blue-green. Now what we did in this case is we peeled the internal limiting membrane all around the macular hole, but we tried and spared the fovea. So you'll see that we are going all around the macular hole. This video is in 2x, so it will appear a little bit shaky. Here we are going all across the macular hole. The ILM has been stained obviously with brilliant blue-green. You're just going across the macular hole in all the directions. Once you had a peel, you just try and trim this residual ILM to whatever extent is possible. And you can just do a fluid air exchange. There is no need to even put gas in these cases. You can see the ILM flap over there, and the hole is nicely closed. This is scenario 2 wherein we are starting with an anterior vitrectomy. This is a case where there is vitreomacular traction with a very thick sheet of posterior vitreous. What we are going to do now is just try and create a hole in the posterior vitreous with the help of a cutter, like what you do in cases of subhyaloid hemorrhage. You can see a very clear hole in the posterior vitreous. Again, we are going to go kind of try and go around the macular hole, sorry, not macular hole, but around the area of the vitreomacular traction, taking care that one does not cross or does not allow the vitreous to cross from one side to the other. So we've done a temporal vitrectomy. Now we are going to trim the vitreous from the nasal side, a bit like truncating the cone. And we are just going all around the area of the vitreomacular traction. Fortunately, in this case, the vitreous was quite opaque, so we did not require the use of triamcinolone. Here we are just ensuring that there's a clear separation of the end. This vitreous was very firmly adherent and would not come with the cutter. Therefore, we've used forceps, which gives a more controlled separation of the posterior vitreous. We're gently nudging, gently separating the vitreous from the optic disc with the help of the forceps. And then we would just go around, complete the remainder of the vitrectomy, including peripheral vitrectomy and base excision. Again, what we would do is we would just want to confirm that there is adequate clearance. Here, I'm not going to be peeling the ILM. We're going to trim this residual vitreous to the extent possible. Just do a fluid air exchange and just leave it at that. So these are two approaches. One can also do a complete ILM peeling with an inverted flap technique if one feels. So this is the pre and post-op result of this patient. Thank you so much.
Beautiful video, Dr. Madhu. And next up is Dr. Atul Dhawan. Swapnesh, can you please share his video?
Yes.
None of the producers have any financial interest in the products or procedures.
Can't see the video, Swapnesh.
Okay, just hold on. So can you see now?
Yes.
Does have any financial interest in the products or procedures shown.
In front of all the stalwarts, I am going to present how to make VR surgery easy. First is Osdalex in the AC. Osdalex has come because of the zonulodialysis. Just make 2.5 mm incision. Place the implant in the line of incision with the help of faucet. Inject low molecular weight viscoelastic.
Lost voice.
Tilt the head towards the primary brain and make it most dependent part of the eye. Do the maximum SRF drainage with the help of the gravity. In this case, the break is at the 12 o'clock position, so just chin up and breathe the flow. Subretinal oil removal in the case of posterior subretinal PVR, which has gone posteriorly. Inject the saline in the subretinal space. Place the retinotomy at the higher level by adjusting the head position. Push the oil by silicone tip towards the retinotomy side. Remove the silicone tip and aspirate the oil by the active suction. Dosa technique. You can learn the temporal flap from any South Indian restaurant very easily. Temporal flap with Bromo-Visco tempo technique. In this case, 180-degree flap I have used. Create the initial part of the flap with the finish loop. You can adjust the length of the finish loop. This is a wonderful instrument. Do it very gently. Don't tear the flap. Start peeling from one side, extend to the other side. Be very gentle. Place the flap over the hole and support it with the help of the color visco, followed by the fluid air exchange. Lamella macular hole. Surgeon has given the hypothesis that the vision loss in the tractional lamella macular hole is because of the synaptic breakage at the outer plexiform layer. So, remove the epiretinal membrane. Put the iLem transplantation. iLem flap will lead to the restoration of the fovea and the synapses and increase in the vision. Creation of the iLem flap, in this case, by temporal area and stabilized by the color visco. At the end of the fluid air exchange, rolled over flap was correctly placed over the hole by color visco suction assisted iLem flap transplantation technique. It is very easy to maneuver the flap under the visco. Epiretinal membrane removal. Hold the edge with the end grasping forceps. Hold the membrane very close to the macular surface to prevent its tearing. If abnormal adhesions are present, regress and strip towards the area of adhesion from the another direction. By this, you can take out the full ERM in toto. Diabetic membrane removal. Use bimanual surgery. Place the chandelier directly, not obliquely. Don't pull too much from the non-dominant hand. Find plane of dissection, inside out, outside in. Any technique you can choose. And with patient, give very small snips. At the end of the surgery, you can take out the membrane in one piece. Always remember, don't make the tractional artery into the combined artery. Thank you very much.
None of the producers have.
Excellent video, Dr. Atul. Nice tips. Next, I will be sharing my video from about optic disc pit. Optic pit maculopathy, vitrectomy for optic pit maculopathy. Can you see the video?
It's a rare congenital cavitary anomaly of optic
No, sir. Video is not seen. Folder is seen.
Now can you see?
This pit a rare congenital cavity
Can it be
anomaly.
seen?
Yes.
It is associated with maculopathy in about 50%
Yes.
of the cases and is characterized by intra and subretinal fluid. Origin of this fluid remains debated. We present a case of 12-year-old female patient diagnosed of dimness of vision secondary to optic pit maculopathy and had visual acuity of 6/60 at presentation. Intraoperatively, after 25 gauge transconjunctival cannulae were inserted, core vitrectomy was done and PVD induction was attempted with cutter before and after staining the posterior hyaloid with triamcinolone acetonide. Was used to achieve PVD induction at posterior core. It involves alternate scraping and aspiration of posterior hyaloid from retinal surface till vitreoretinal separation is achieved. PVD is used near posterior pole. It is extended to the extent possible with aspiration scraper. Peripheral PVD is completed and vitrectomy done with cutter. PVD is extended up to pora serrata with cutter in all quadrants, and thorough vitrectomy is performed. After vitrectomy, brilliant blue dye is used to stain ILM under air. Pinch technique is used to initiate peeling of the ILM. Arcade to arcade peeling of ILM is done. ILM close to optic nerve head is peeled and draped over the area of optic. Of the surgery, fluid air exchange is done. C3A-fed gas is injected and cannulae are removed. Post-operative follow-ups show progressive regression of maculopathy with reduction in intra and subretinal fluid and progressive improvement in visual acuity. In conclusion, primary vitrectomy with ILM peeling is effective technique of management of optic pit macular.
Thank you. The last video of the day is by our boss, Dr. Preetham Samant. He is going to present cases of...
Thanks, Manish. Before you start the video, a quick word. First of all, many thanks to Manish and Swapnish for making this session possible. I just gave them the idea. They conceptualized it and executed it. Very well done. We've covered 17 presentations in 1.5 hours. That is quite a feat. And I thank you for all the faculty who participated and gave us the fantastically edited videos. I know the oculoplasty dignitaries are waiting for us. Give me another 4 minutes and I'll be handing the session to you. Please start, Manish.
Can you see the video?
Yeah.
Yeah, please go ahead with the commentary.
Okay, thank you. So as we know, primary hyperplastic primary vitreous is persistent vitreous vasculature, and it can cause diminution on vision in various ways. So I'm going to show you 3 scenarios which were tackled surgically. In the first situation, there was vitreous hemorrhage just behind the lens in the patella fossa. You can see the ultrasound showing the PHPB. And this kind of vitreous hemorrhage can, in very small children, can cause amblyopia. And it's important to do a vitrectomy and clear the visual axis. So as you can see here, you have to go very close to the limbus because these are very infantile pediatric eyes. And with the cutter, first I am getting rid of the vitreous hemorrhage all around the stalk of the PHPV. Now the aim of the surgery here is not just to fracture the PHPV. Now in this case, it was a very small PHPV, just a single point. So I used a scissor and fractured it away from the lens. You can see it falling back and you can see the stalk of the PHPV lying on the retinal surface. I really don't have to pull it from the disc. I just amputate it as much as possible and we are done. So this was a much simpler case. You can see here the post-op result, the vitreous cavity is clear. Now in the second situation, although there was no vitreous hemorrhage, the attachment to the posterior capsule was pretty large, like a medusa head. You can see here the attachment. You can see how the stalk has attached itself. So here I can't just cut the PHPV and leave this opacity behind the lens, because when the pupil constrict, this opacity is going to be bad. So I cut all those attachments. Now this is why I say this is living on the edge, because I'm working behind the lens, making sure I don't touch the lens. A small lens touch can cause cataract. So while I pull the PHPV down with a little tug on the forceps, it's a bimanual dissection, I cut the stalk. And as I cut the stalk, you can see the stalk again falling back on the retina. And then the remaining stem of the stalk is just simply amputated with the help of a cutter. So it is very important to clear the visual axis in these PHPV patients. If many a times we may even have to sacrifice the lens if we are not able to do it, but that would be the last option. And so you can see this, the vitreous, minimal vitrectomy being done. You really don't have to do a total vitrectomy in these cases. And it is sutureless at the end of the day. So that is the advantage of this MIVS. The third scenario was there was actual tractional retinal detachment. So it was imperative that I get rid of this traction and cut the PHPV, more like the medusa, which is stuck not only to the lens but also to the retina. So you can see here the connections the PHPV has to all around the lens. So first I debulk the anterior vitreous all around the stalk. Hold the stalk gently with a forceps. Very important is not to yank it off, and then gently release, a little bit of tug and going in between the lens and the stalk. I'm cutting it. Now here, there are some attachments which I leave behind. I really don't go after them because I know they are adherently stuck to the posterior capsule. Cutting it from all sides so that the central 2 to 3 millimeters of the posterior capsule is made free of this this is vascular frond. And once it is free, then the job becomes easy. Then to go inside the vitreous cavity under visualization and to cut the remaining stalk. This tractional retinal detachment will eventually settle on its own. In small children, the retina is quite elastic and the retina eventually settles. You can see this is the end result. I've left back some pieces of those tissue in the periphery, and you can see this is the end result. This is a 6-month post-op ultrasound showing a clear vitreous cavity. So these were the 3 cases which were successfully dealt with. Thank you so much.
So we are going to have concluding remarks from Dr. PB sir.
So Manish, you're mute. Maybe you can just conclude.
It was brilliant videos by everybody, and thank you for all faculty members. Can I request Dr. PB sir to have a concluding remark, please?
Yeah, I think it's a great session, and thanks to all the speakers. I think everybody has, I think Swapnesh and Manish has given a strict instruction. I think everybody followed, everybody stick to the time. And I think that's what the beauty of things now, like when your session finish everything in a time, everybody feels really happy. I really thank all of you. And it's really always when you see video, whether it's junior or senior, always you pick up 1 or 2 new points and you can implement in your practice. And that's the way we all keep on improving. Thanks, Pritam,
thank you so much,
Manish, and Swapnesh specifically for having me here. And thank you all speakers, and nice to see Dr. Nitin after a long time. Thanks. Thank you very much.
Thank you so much, sir.
Professor
I would like to thank also my president, Dr. Sunaina Malik, madam. She's just joined us probably for the next session.
Thank you. Thank you, Pramod. Nice to see you.
Thank you so
I'm
much,
not a posterior segment surgeon. You know that, Pritam.
Yes, yes.
But I like seeing all of you. See you all, guys.
Over to you, madam, for the oculoplasty.
Thank you so much, Zach. Thank you. Bye.
Bye. Thanks, everyone.
Bye. Bye. Bye.
Thank you for ending it in time.
Go ahead, Shubha. Start your session. You're already 5 minutes, 10 minutes delayed.
Yeah.
We need to finish by 4:30.
Yes. A warm welcome to all of you here at a wonderfully organized MOSVIRCON with an oculoplasty session, the session of oculoplasty capsule. And I am Dr. Shubha Jhabbar. I am oculoplastic surgeon and a professor academic at Government Medical College, Aurangabad. And me with Dr. Ramesh Murthy, and a very renowned oculoplastic surgeon from Pune, who is also a pediatric ophthalmologist, I should say, and working at Axis Eye Hospital. We both are chairing this session. And after the grand success of the MOS webinar on oculoplasty extravaganza that we did in July, which had 3,000 plus viewership, we are back here, we present here an array of stalwarts of oculoplasty. So we present here Dr. Usha Kim ma'am. She's come from all the way from Madurai. She's the president of Oculoplasty Association of India. Welcome, ma'am. I welcome the doyen of oncology, Dr. Santosh Honawar, Dr. Milind Naik, who takes pride in aesthetics. And we have Dr. Anuradha Iyer, our very own enthusiastic Dr. Aditi Watwe, who comes from Kolhapur and is a very energetic lacrimal specialist, and Dr. Ravindra Wankhede from Solapur, Dr. Mangesh Dobekar from Ganpati Netralaya Jalna, and Dr. Pallavi Khedkar from my Aurangabad. So we have all of us here. I welcome all of you speakers, and we start with the session. The first talk is by Dr. Usha Kim ma'am. I welcome you, ma'am. She heads the Department of Orbit Oculoplasty at Arvind Eye Hospital, Madurai. She is currently the president of Oculoplasty Association of India. And given her interest in oncology, she is currently involved in the genetic research pertaining to retinoblastoma, and that's what her talk is about today. She has around 57 international and national publications to her credit, and has co-authored the Atlas of Neuroophthalm and Orbit. Her unit is a preferred destination for training in orbit and oculoplasty, and she has set up a Ring of Hope, this initiative in 2004 to raise funds for the treatment of eye cancer. With this, I welcome you, ma'am, for your talk on role of genetics in retinoblastoma, a case-based study. Welcome, ma'am. You have to unmute yourself, ma'am. Please unmute.
Thank you so much, all the speakers and chairpersons. I really appreciate you having me over. And it's really nice that we could meet on this platform, because I think it's very difficult for all of us to travel this far and across the country. I think we've got all representatives. Thank you, all the organizing committee members, and Pallavi in particular, because that's the connect. I think she still has the cord connected to Madurai. Thank you so much for having me. And I would start off my talk on retinoblastoma and the role of genetics in the retinoblastoma. Oops, I think it's, yeah. So retinoblastoma, I don't think we need to go into the details of the tumor. It's the most common pediatric intraocular tumor world over, and we do see a lot of cases in our part of the world, and the cases are anywhere between 80 to 100 in a year in our institution, and that's how the interest started. So these are the various presentations of the retinoblastoma patients. They commonly present as leukocoria, which all of us know. So way back in about around 2003-04, I could see that there was a huge number of cases presenting to us, especially in the late stages of the tumor. And that's when the transition happened between the old and the new classification. And however I extrapolated in whatever method or classification, it all boiled down to being presented in the late stages of the disease, which I think every one of us would have come across in our practice. Now, what was most interesting was of the cases that we had accumulated in the about 5 to 6 years, I saw that a huge number of cases were heritable. We did see a familial history, and that's what arose the interest in me to go ahead with the genetics in retinoblastoma. So we did do a lot of investigations and treatment and put them in place. But what was actually missing was the genetic testing and prediction. And in fact, with the genetic testing, we were able to, and I'm sure all of you would agree, we would be able to bring the children earlier in the early stages of retinoblastoma, which would translate to better outcomes in whatever treatment we advocate. So the first thing we did was actually revisited the Knutson's hypothesis, and obviously for all known reasons, the bilateral cases did present early because of the visual loss in both the eyes, and the unilateral presented later in life. So which is the reason why we did see a lot of advanced cases. And whenever we had the bilateral cases, both of them would have a late stage presentation because they wait till the vision drops. And it's in the early part of the childhood, so the children may never notice it and would not be able to tell the parents as well. So it's only the parents to all of these and bring it to our notice. And unfortunately, whatever we saw was in the lower socioeconomic strata. So there was not much of an emphasis given to these children, and especially not much of a notice was given to the vision deficiency as well. And as you all know, retinoblastoma gene is not an easy gene. It's a very lengthy one, and it is a very heavy one, and it's very difficult to study this gene. So you know that, I'm sure all of you must have in your early days read about the functioning of the RB gene, which actually controls the cell proliferation. And inactivation of this would result in uncontrolled cell proliferation. So this is the background with which we started working. And what's interesting is with heritable condition, that is with the heritable retinoblastoma situation, it often we assumed that it would be in both eyes, but we did see some cases which was unilateral as well. And there is also another high risk of other cancers apart from retinoblastoma, and there is an increased risk of the retinoblastoma presenting in siblings as well as the offsprings. And the alterations are found in the blood. This is the basis, and this is what you need to remember as we go on with my presentation. And the non-heritable one clearly affected only one eye in most of our cases. There could be some outliers, but it's often only one eye. And there's been no reported case of other cancers. And there is also a reduced risk. I wouldn't say there's no risk, but there's a reduced risk of RB in the siblings and offsprings. And the alterations are identified only in the tumor. So this is the most important thing for us to understand. And with this basis in mind, we went ahead and did a tracing of a 4-generation family, which was affected with retinoblastoma. So the first, we added this element to all our retinoblastoma treatment protocol. So genetic testing was part of our treatment protocol now. So this is the first family which came to us. The mother was affected with retinoblastoma. She had her right eye enucleated way back, and she's completely all right otherwise. She had brought in with her first child with bilateral retinoblastoma. One eye was advanced and the other eye was treatable condition. So one eye was to be enucleated. And as we were following this child up with chemotherapy, she became pregnant with the next child. So when the question she asked, had in mind was, obviously she would ask, I am affected, my child is affected, would the next child also be affected? And then the answer came in very directly to us. The mother and the proband had a very similar mutation. So we predicted that the sibling is going to have the almost a 50% chance of having the tumor. So we had asked the mother to bring the child earlier on in life. That is, she brought the child at about 12 days after birth. And fortunately, unfortunately, the child had bilateral retinoblastoma, but in its early stage. So the child was treated with chemotherapy, and the child grew up to have both vision and the eyes salvaged in both the eyes. So this child had one eye removed and the other eye was salvaged. Whereas with the prediction, we brought in this child earlier and this child had both vision and eye salvage done eventually. Now there's another scenario where you have a set of twins. One eye had a squinting, and that one of the twin had one eye squinting, which had a retinoblastoma, and the other twin had a retinal scar. We weren't sure if it was a retinoblastoma scar or was it something else. So then we had to subject the parents and both the children for genetic screening, and fortunately we found de novo germline mutation in the blood of the twin A, whereas there was no mutation found in the blood of the twin B, which meant that the twin B is going to be absolutely normal, and we were almost sure that the child was not going to have retinoblastoma, which meant that we didn't have to go through the examination under anesthesia month after month or follow up the child as we had to follow up with the twin A. Now, this is another scenario where there is a child is had a unilateral RB, a group E, and it presented almost 36 months. You often notice that with a unilateral retinoblastoma, they present late in life. There's no family history. And as I had mentioned earlier, you often think that unilateral cases would not have a genetic, wouldn't be a heritable disease. But in this case, there was a mutation identified in the blood, which means that blood should be screened in all unilateral cases as well. What does this indicate? It indicates that there is a higher chance for the sibling to be affected and a very high chance for the offspring of this child to have a retinoblastoma. That's the indication that we need to keep in mind. Now again, there's another child which presented as a unilateral case at 26 months of age. There was a germline mutation found in the blood of this patient, and eventually the child turned to become a bilateral RB. So when we did the analysis of the parents, the father showed a mutation and he became an unaffected carrier of this disease. So we then predicted a higher risk for the next sibling. So remember all the scenarios, and everyone insists that there needs to be a genetic analysis done for every child with retinoblastoma. So this is one-off case of Waardenburg syndrome, which we found out incidentally. The patient was referred to us when the child was 5 months old with a suspected retinoblastoma. The patient had a history of colostomy done 2 days after birth, and then we realized this was a Hirschsprung's disease, and we actually reported this case, and you can see the mutation in this proband. So now the next question was, would the offspring be affected? And this child had a unilateral RB. Enucleation was done at 9 months of age. There was no family history of RB and any other cancer in the family. So the little girl grew up and she was ready to get married. She wanted to know if there is any risk of the offspring being affected with retinoblastoma, and a genetic analysis was done with no mutation found in the patient's blood. We predicted that there is less than 1% chance of the offspring getting affected with retinoblastoma, and fortunately the baby is disease-free. And another interesting scenario where a unilateral RB patient was enucleated when he was very young and in his early on, and then when presented at 18 years of age, wanted to know whether there is any risk of secondary cancers. With all the information available in the internet, I think these children become more and more aware, and they wanted to know more about themselves. Since there was no mutation found in the patient's blood, we predicted that there is a very less chance of getting a secondary cancer, and this is how it turned out. This child actually this is the same child who presented to us way back in 2003, and then when he had a bilateral retinoblastoma, one eye was group E, which had to be enucleated, and the other eye was group C. And we had subjected this child for chemotherapy. Vitrectomy was done and tried with intra-arterial melphalan infusion, and this child has grown up to be a young lad, and fortunately he has no risk of secondary cancers, which is proven through a genetic analysis, and this is the fundus picture post-treatment. Now, the other thing which also came up was a situation where the proband had bilateral RB, and there was one particular type of deletion which was found to be inherited from the father of this proband. There was, this risk of RB was explained to the family for the next sibling to be also affected, and we had educated the parents about it. So the one child was affected, which was inherited from the father, who was an unaffected carrier. So with this similar mutation happening, we predicted that there is a risk of the next sibling to be also affected. And immediately after birth, the newborn was brought to us, examined, and genetic testing was done to check for deletions. And fortunately, the sibling was found to be normal. And this eliminates the child having a risk of RB, which eliminates the need for repeated evaluations. Now, there is another scenario where there was a familial RB with 4 affected members in the family. The proband and the mother had heterozygous deletion, which is one particular type of deletion. And the family wanted to know the risk of RB in the growing embryo. And following an amniocentesis, unfortunately, we proved that there was a mutation in the growing embryo as well. And then this resulted in the termination of pregnancy, which was purely the decision of the proband and the family. So now the question next was, does this involve a lot of, a series of research? Yes, it did involve a lot of research in the institute. In our own patient population, of which 20 were very unique to our Arvind Eye Care system. And there were a lot of mutations, which was mainly missense, nonsense, splice. And fortunately or unfortunately, we did see a lot of variations in the amount of penetrance as well, all of which has its own implications in the disease pattern as well as in the outcome of the treatment. And I will have to state that we are on par with the international standards, but we are providing at a very less cost. What costs is about $1,800 is now costing us about $250 in our setup. And we have established the genetic testing lab in our own setup. And these are some of our happy patients, and all the relevant investigations, starting from the intravenous chemotherapy to high-dose chemotherapy, lasers, cryo, thermotherapy, intravitreal. Now we've started the intra-arterial as well. All the surgeries, reconstructions, prosthesis, rehabilitation, low vision aids, genetic testing are all done free of cost to patients who cannot afford through the Ring of Hope project. And thanks to our entire team who've been making this possible, and it's an ongoing process. There's a lot of research which we are pursuing, but what we like to emphasize is all of these are just not bedside, they are brought to the bed, and all these research activities are extrapolated, and it has been useful in our real life scenario. Thank you again for this opportunity.
Thanks, ma'am, for a wonderful talk on genetics. Very important for us to examine all the siblings of retinoblastoma that we are treating. I hand over to Dr. Ramesh Murthy to introduce Dr. Santosh Honawar. I should say he's been eluding the MOS conference since 2 to 3 years, mainly because the American Association of Ophthalmologists dates collide with MOS conferences. But due to this pandemic, we are lucky to have him here today. Welcome, sir.
It's indeed an honor to welcome Dr. Santosh Honawar, sir. There are no words which can describe him. He's amazing. He's definitely one of the leading ocular oncologists in the world. He's the only Indian who has won the foremost award from the American Academy, that is the Lifetime Achievement Honor, apart from the innumerable other awards that he has won. And maybe that's why this time he is stuck on to us. So I think he's won all the awards that they can offer. Honawar, sir, it will be a pleasure to listen to your talk on eyelid tumors, the good, bad, and the ugly.
Thank you, Ramesh. I thank MOSVIRCON for the wonderful opportunity. I'll be speaking on eyelid tumors, just the introduction basically, because in 10 minutes you can introduce what is good, what is bad, and possibly what is ugly. Now, if you look at these three gentlemen, of course, you may have seen the movie. It's very difficult to say who is good, who is bad, and who is ugly, isn't it? Unless you remember the movie very, very clearly. So in a land of confusion where everything looks similar, it's hard to tell who is good, who's bad, and who is ugly. Unless I tell you who is good, who is bad, and who is ugly, it's very difficult for you to recognize sometimes. But then even when you look at a white tiger, you can identify it by its unique identifier, that's the stripe. Similarly, every tumor comes with a signature. And if you're able to identify that, the job is nearly done. Everyone knows that this is a patient with multiple papilloma. Of course, patients with bilateral multiple papillomas have a possibility of being immunocompromised. That is one. Secondly, if you look at this particular papilloma and this, would these two differ from this? Yes, because there is some amount of change in color and vascularity. So, or if a papilloma were to have high degree of vascularity, bleeding, and induration at the base, instead of being pedunculated like this, if it becomes sessile and has induration at the base, that is one more sign. Or if it has change in morphology, which is of recent onset, and if there is surface ulceration, then you think that a particular papilloma is turning into squamous cell carcinoma. Papillomas are HPV-16 induced tumors, and they have a tendency to turn into squamous cell carcinomas, and you should be guarded against that. Or if a papilloma has marginal location, papilloma is generally supposed to be present on the skin. But if it is in the intermarginal strip and has a broader base, indurated broader base, that means that it's an inverted papilloma. Again, the chance of it being squamous cell carcinoma is much higher. So in a patient with papilloma, you should be worried about change in color, change in vascularity, ulceration, hemorrhage, and a larger, wider base and induration at the base, which are indicators of it being malignant. Now, this looks like papilloma, but much placoid, much flatter, sessile. This is seborrheic keratosis. Very typical manifestations of seborrheic keratosis, which is quite common. This is one more patient with seborrheic keratosis, which is nothing but benign proliferation of the basaloid cells of the skin. That's fine. But what if a patient were to have multifocal sebaceous keratosis? Multiple sebaceous keratosis, not just in the periocular area, but also on the face, chest, and abdomen. Then it is dangerous. It's called sign of Lesetrelet, which is a cancer predisposition or a paraneoplastic syndrome rather, and also is seen in patients who have immunocompromised status. So you should be worried about patients who suddenly develop a lot of seborrheic keratosis, not just in the periocular area, but also on the face, chest, and abdomen. Now, this is a tumor in a young individual. He's just 16, and this has a tumor with loss of lashes, looks definitely like a malignant tumor, but has central ulceration or umbilication. So that's a feature of keratoacanthoma. Just with this particular signature, the tumor, which is spheroid, but with a central umbilication, indicates that it is keratoacanthoma, which is a benign tumor. It may look like a malignant tumor, it may be locally destructive, but by nature it is quite benign, and you really don't have to be worried about it. You can simply excise and reconstruct the lid. As opposed to that, this looks very, very benign. All this patient wants is cataract surgery for his mature cataract, but he also has a cicatricial ectropion, or what looks like cicatricial ectropion, but there is loss of lid margin, and there is something pigmented with pudgy orange appearance. So there is some form of pigmented lesion which is non-ulcerative, placoid, with attachment to the underlying periosteum, causing ectropion of the lower eyelid. This is morphiform or sclerosing variant of basal cell carcinoma. So it can look very innocuous, but it is a malignant lesion. Of course, basal cell carcinomas are quite rare in Asia and India. They are more common in the lower eyelid, and there are three variants: nodular, ulcerative, and morphiform. It's rather easy to recognize except for the morphiform variant. This is how it looks like. It can be secondarily pigmented in Asian Indians. In fact, about 50 to 60% of basal cell carcinomas can become pigmented and may look like melanoma. But what if the bad gang up? Basal cell carcinomas, many of them presenting in a patient, indicates that there is a syndromic variant, that's Golin-Gott syndrome. These patients also have Palmar pits, odontogenic keratosis, and calcification of the falx cerebri. So if you have a patient with multifocal basal cell carcinomas, they should look for the syndromic variant or Goldenrod syndrome. Squamous cell carcinoma is evidently bad. Generally, about 80% of squamous cell carcinomas are nodulo-ulcerative. Nodular variant of squamous cell carcinoma is quite rare. Ulcerative form is, of course, present. But the problem with squamous cell carcinoma is that it has a high chance of orbital extension and also regional lymph node invasion. Let's go to something which is more common. All right, there are 4 ladies here, almost similar age group, except one this one is slightly younger. All of them are in the 50s, early 60s. So which one of this is a chalazion? It's difficult to tell, isn't it? All of them have nodular lesions in the upper eyelid. In this lady, you find that the eyelid margin is thickened. This one doesn't have eyelid margin involvement. This is slightly larger. This is slightly larger. Now, with all these in place, which one do you diagnose as chalazion? So you cannot diagnose a chalazion unless you look at the accompanying features, especially in an atypical age group. If you were to encounter a 20 or a 30-year-old with a nodular eyelid lesion, most likely it is chalazion. But when you have a atypical age, definitely you have to pay attention to rest of the features, which are eversion of the eyelid, showing a bluish discoloration with thinning of the tarsus, would mean that it is an epidermal inclusion cyst. Very straightforward diagnosis. Also look at the meibomian gland orifices, look at the thickness of the lids, and look at the lashes. If all these are fine, then it is an epidermal inclusion cyst. The other 2 ladies that I showed have other attendant features apart from the eyelid nodule. What are these? Let's look at these very carefully. There is widening of the lid margin. You can see the normal thickness of the lid margin there, and it is evidently widened. Posterior lid margin is all rounded. You can see meibomian gland orifices up to this point, but beyond that they are missing, and the lashes are sparse. There's a posterior lesion as well. So this is typically sebaceous gland carcinoma. So the dictum is that I'll just draw that and show you. If a young individual were to come to you with a lesion which is, which is restricted to the tarsus, or it is now higher than the height of the tarsus. If this is the height of the tarsus, if a lesion were to be just restricted to the tarsus in a young individual, most likely it is a chalazion unless it is accompanied by additional features. If it is higher than the level of the tarsus, most likely it is an epidermal inclusion cyst, in which case you look for thinning or bluish discoloration of the tarsus. If an elderly individual were to present with a lesion which is higher than the level of the tarsus, then you should look for attendant features of sebaceous gland carcinoma, and that is the most likely diagnosis. So that is why you should be worried about any eyelid lesion which presents in elderly individuals. Now this looks very innocuous, but the patient is 78-year-old. This looks like a small little bump in the eyelid, but what is happening is that the meibomian gland orifices are missing in that area. There is some amount of loss of eyelashes, and the eyelid margin seems to be thicker. So this was considered some sort of inflammation. The patient was left alone. After 3 months, you can see that it has evidently grown. So this was an early sebaceous gland carcinoma grown over 3 months of time. So anything which disturbs the posterior lid margin and results in loss of lashes and meibomian gland orifices can be sebaceous gland carcinoma. So in fact, it is written in books that any nodular variant of sebaceous gland carcinoma masquerades as chalazion, and any recurrent or atypical chalazion warrants biopsy. Atypical not just in terms of its morphology, but also atypical in terms of the age of the patient. Quickly showing you other variants of sebaceous gland carcinoma. This is nodular variant. You can see extreme degree of vascularity, which is not to be seen in a chalazion. So if you have a large nodule like this with a lot of vascularity, then sebaceous gland carcinoma. Nodular variant presenting on the tarsal side without involvement of the lid margin. Obviously, in these patients, you don't see that meibomian gland orifices will be occluded or the lid margin will be thickened. This is a nodule plus an ulcerated nodule. It's a bilobed sebaceous gland carcinoma. Apart from that, note thickening and congestion of the palpebral conjunctiva as well. So all this area is involved with intraepithelial or vegetoid sebaceous gland carcinoma. This is the early nodulo-ulcerative variant where there is evident central ulceration already, Rest of the features are of course present. Now this is a predominantly nodulo-ulcerative. The entire surface of the lesion is ulcerated. This is predominantly ulcerative variant of sebaceous gland carcinoma. When you talk about sebaceous gland carcinoma, you expect the lesion to be there on the posterior aspect, that is the tarsus and the tarsal conjunctiva. But when a lesion were to present predominantly in the lid margin, then it is most likely from the Zeiss glands. Now, Zeiss glands are anterior to the sebaceous glands. If these are sebaceous glands, these are Zeiss glands. Zeiss glands are associated with the eyelash follicles, and these lesions tend to be present in the lid margin and towards the lash follicles. So loss of lashes is more common in Zeiss gland lesions, but these tend to be very small. Finally, this is about a patient who has chronic unilateral blepharoconjunctivitis. This patient has red eye in the left eye, and when you look at the eye carefully, you find that there is some amount of conjunctivalization of the cornea, loss of limbal architecture, and when you invert the lid, you find that the lid margin is widened. Posterior lid margin is all rounded. You can see the thickness of the lid on the normal aspect, whereas it is very thick on the inward side, and meibomian gland orifices are all lost. So this patient has all the signs of sebaceous gland carcinoma, but no visible tumor. So this is vegetoid variant or intraepithelial variant of sebaceous gland carcinoma, which is also seen in this patient. You can see already that the lid margin is grossly thickened. So if you have a patient with unilateral blepharoconjunctivitis with gross thickening of the lid margin, loss of meibomian gland orifices, and sparse lashes, then you're justified in doing a biopsy. If a patient were to look like this, don't treat them, continue to treat them conservatively, but do a map biopsy. Even a patient who looks very, very innocuous, just blepharitis, chronic blepharitis of the upper eyelid, when you look at it on slit lamp, you find that the posterior lid margin is rounded, meibomian gland orifices are lost, and lashes are sparse. This is again an early variant of vegetoid sebaceous gland carcinoma. Look at it very, very carefully. You can, if you look at it on slit lamp, you find that there is vascularity of the intermarginal strip as well. Very, very early variant of vegetoid sebaceous gland carcinoma. This is, of course, a late manifestation of vegetoid sebaceous gland carcinoma with the entire ocular surface involved. There are other variants of intraepithelial called bovenoid and papillary as well. So you should be aware of these variations. So if you have a patient with diffuse unilateral chronic blepharoconjunctivitis, then you should suspect vegetoid sebaceous gland carcinoma and do a map biopsy. Otherwise, they end up resulting in orbital exenteration because of delayed diagnosis. Melanocytic tumors, of course, there are too many of them, but just to show the most common variant, that is a nevus. Nevus presents anywhere in the eyelid, eyelid margin, or on the skin, but it has a nice glistening surface, and the lashes, in fact, are hypertrophic. They could be hypertrichosis, they not be loss of lashes. That's one feature of a nevus. And nevi currently can simply be vaporized using a RF electrode or carbon dioxide laser. The advantage of vaporizing a lesion nevus, as opposed to excision, is that lashes very nicely grow back in 6 to 8 weeks. So you maintain the cosmesis and get rid of the lesion by this technique. Whereas malignant melanoma of the eyelid presents as a nodulo-ulcerative lesion, it is dull in surface and has vascularity and variable pigmentation. So asymmetry of its morphology, irregularity of its borders, variation within the lesion itself, color variation, and a larger diameter indicates melanoma, and there are 3 variants of melanoma. To quickly show you other malignant tumors, this is a highly vascular lesion in a young HIV positive individual. This is Kaposi sarcoma. And if you were to have a larger lesion, rubbery lesion in the eyelid, then you consider B-cell lymphoma. Similar lesion with skin necrosis, you consider mycosis fungoides or T-cell lymphoma, things like that. So there are certain sentinel signs for each eyelid tumor, and that will help you differentiate one from the other. Visual impression and clinical features are important in arriving at a differential diagnosis. Thank you so much.
Thank you, Hunawa sir, for a very informative talk. It is indeed an honor for me to have 3 of my teachers presenting in succession, Dr. Usha Kim Madam, Dr. Hunawa sir, and Dr. Milind Naik. Dr. Milind Naik is the Chief of Oculoplasty at L.V. Prasad Eye Institute. He's definitely one of the pioneers of cosmetic oculofacial surgery in India. And I still remember in the year 2002, he did pioneer work in Botox. I request him to speak on this very interesting topic of whether ophthalmologists should actually practice aesthetics. Dr. Milind Naik, sir, please.
Hello, everyone. Ramesh, can you hear me?
Yes, I can hear you, sir. Please share your video.
My
Your
screen share is disabled. If someone can help me with that.
In charge of the hall, please enable it. Who's the hall in charge? Just a second, Dr. Naik.
Sure.
Yes, ma'am. Yes, ma'am, doing it.
Yeah.
Okay, thank you. So thank you, MOSVIRCON and the entire oculoplasty team of MOSVIRCON for having me here in this program. I know this meeting in person was supposed to be in my hometown, Nasik. Nevertheless, it's great to see you all on the screen here. I'm just waiting for the screen sharing.
Since we have some time, maybe I can ask a question to Usha madam. Madam, when we do the genetic testing for children with retinoblastoma, do we have to screen the children, the other children in the family and the parents as well?
Yes, doctor.
Do you advise that?
Yeah. Whenever a child comes with retinoblastoma, I think it's mandatory to examine all the members of the family. One, when I say examination, it is both, physical examination of the eye as well as we include the genetic testing. It's important that we examine it.
Oh, okay.
Okay. Yeah. Dr. Milind, is your screen share on?
Yeah, he started.
Great.
Yes.
Thank you.
Okay, can you see my slides?
Yes.
And am I audible?
Yes.
Okay, so the topic for me is should ophthalmologists practice aesthetics? And I'll quickly take you through a few of my thoughts on this topic. So if some of you noticed the photographs that Dr. Honavar was showing, the white tiger that he showed also had an aesthetic problem. He had a left mild ptosis. So aesthetics is important, and we will touch upon the topics of should we do it as eye doctors, what is the scope, and how do we begin. So first asking the question, should we? Now facial aesthetics is a branch which is a no man's land. So there are dermatologists, plastic surgeons, oculoplastic surgeons, maxillofacial surgeons, and these days surprisingly even gynecologists are practicing it. And being an ophthalmologist, we certainly are in the area where we should be contributing to this. And here is a simple statistics. Per patient, you have only 2 cataracts, but you have 4 eyelids and you have multiple wrinkles. So it's better to defocus from cataract and focus on the other stuff. And sooner or later, someone who's right now sitting in the lab rather than attending a webinar is going to find a medicine for cataract. And that day, you might have to zoom out your slit lamp and look around the lens to find out other opportunities. One more reason why an ophthalmologist should get into aesthetics is this couple, Jean and Alastair Caruthers. These were the pioneers of popularizing botulinum toxin and its cosmetic uses, and one of them is an ophthalmologist. For someone who can do as complex a surgery as this, performing cosmetic surgery around the eye should not be a problem. Now this lady, moderate myopia both eyes, she underwent LASIK, and post LASIK she realized that the spectacle was actually hiding her appearance, which now needs repair, her tear trough and the overall sad look that she has. Another case where the glasses were eliminated, and now she claims that this mild ptosis on the left side is caused by this surgeon who eliminated her glasses. So aesthetics is around us every time and all the time. So should we get into it? Yes, definitely. And in the periocular region, we very much have an advantage. What is the scope? So the low-hanging fruits in this area would be obviously injectables. You can start off with botulinum toxin injections. You can easily start off with spasms first, and then graduate to periocular wrinkles. And next step would be fillers. Now, all ophthalmologists are indirectly handling a filler, which is your viscoelastic during your cataract surgery. So if you can slide it very beautifully inside the eye and fluid under the capsule, you can certainly put some of it into this groove and eliminate the valley that the patient has. It's just an external job, that's it. And we do have a very good opportunity to practice this. Every patient of DCR, we are going to take an incision exactly in the area where we are usually doing a filler injection. So use your local anesthetic and try and fill the tear trough as a practice so that you can learn fillers. This is data about blindness that can occur from fillers, and this is one thing which scares patients from visiting specialists like dermatologists, and this is where we have an advantage as an ophthalmologist. So taking all the precautions to avoid intravascular injections and being at an advantage of picking up this complication if at all it happens is a big advantage for eye doctors. And once you master the periocular area, you can always move on to the other areas of the face, of course, with relevant training. And these are surgical procedures, which is a step beyond a filler injection. Blepharoplasty, of course, comes into our purview, whether it's upper eyelid skin excision or the lower eyelid fat bags. With some amount of training, any ophthalmologist should be able to do a good job. We have a great advantage of doing this lower eyelid surgery transconjunctivally, which many plastic surgeons are uncomfortable, and avoiding a scar is a big advantage from the patient's point of view. You can also expand into lasers, although personally I have realized that that's not a good idea because it eats up into the dermatologist business and referrals can get affected. Of course, any major surgeries beyond the periocular region in the form of facelift, neck lift, and even maxillofacial cosmetic procedures has to come after a formal fellowship training. So with minimal training, you can start off with Botox and fillers. With moderate training, that is observing another oculoplastic surgeon, you can definitely do blepharoplasty. And for something more extensive, you will need a fellowship training. So what's the scope? The entire face. And how do you begin? I would suggest that you start by changing your mindset, which begins with defocusing your slit lamp away from the lens, making sure that the slit lamp, overhead lamp is working, and you also look at the patient's face and not just the eye. A little bit about understanding their world. And a cosmetic patient's world is always different from ours. We are functional people, we are doctors, and we always tend to judge them or we tend to trivialize their problems. But believe me, for them, the wrinkle is as important as a posterior subcapsular cataract may be for you. So another step is to spend time with experts. There are a couple of oculoplastic surgeons in our country who practice this, and just pay a visit, understand what are the nuances in this field. Attend non-ophthalmology conferences, go to dermatology meetings. There are many aesthetics meetings which are multi-speciality. You should attend that. And injectable workshops. Now, these could be cadaver or these could be real live injections, and they are relatively high price, but I would say that you compare this price to your 1 month phaco training that you would get anywhere, and acquire these skills before you start. So just to summarize, learning aesthetics before medical treatment for cataract hits the market. Good training is very important, and ophthalmologist is already at an advantage. Thank you.
Thank you, Dr. Milind, for a wonderful presentation. Well, surely aesthetics is really important these days. It's in demand as well as it's a demanding branch. Do we have any questions for Usha Kim ma'am, since she needs to leave? Well, we have a question from Dr. Pallavi. She asks, is there a prognostic connection with genetics, ma'am?
Yeah, there's a lot of work that's going on. There's been a difference in the pattern of outcomes with regard to the differentiation also. And so we realize that we need to link the differentiation pattern versus the genetic mutation pattern. So there is work in progress, and I'm sure we'll come out soon with the results.
Shubha, I'd like to ask something.
Yeah, please, ma'am.
Mentioned in your talk that the cost of the genetic testing is about 16,000 rupees, I think, $250 US.
It's all, yeah.
So do most of your patients afford it? Can they afford it?
That's why I mentioned that all of these, for those who cannot afford, we are providing it free of cost.
Yes.
And for those who can afford it, which is like I would say about 10 to 15% of my patients that can only pay. The rest of it is all non-patients. So we, through that fund, which I was mentioning earlier, we are trying to accommodate all of it.
Thank you.
Okay, one more query for the pregnant lady that you said you tested. So what is the protocol that you follow, like when to do and how?
Yeah, so amniocentesis is typically like how it's being followed. You have to make sure that the gynecologist, the obstetrician is there with you, and it's performed by the obstetrician, obviously. But the thing is, we need to ensure that you need to let them know before 3 months of termination is not possible after that, and the decision of continuing the pregnancy is entirely on there. The onus is not on the the ophthalmologist. We let them know the outcome, and unfortunately, in this particular scenario which I mentioned, it was a triplet, and they couldn't afford to go ahead with the pregnancy. That's the reason they had.
Thank you, ma'am. Any more questions, Dr. Ramesh Murthy? Well, okay. Thank you, ma'am. Thank you for being present. Thank you very much. We move on to the next speaker now. She's Dr. Anuradha Iyer, and her topic is ptosis, diagnosis and management. She is a fellowship trained in oculoplasty, facial aesthetics and oncology at L.V. Prasad Institute, Hyderabad. She completed her post-graduation from Arvind Eye Institute, and she primarily consults at her hospital, OSS Care Center, in Thane city, with special focus on oculoplastic and aesthetics. Welcome, Dr. Anuradha.
Good afternoon, everyone. Dr. Shubha, am I audible and is my slide seen?
Yes.
A very good afternoon, especially to my colleagues and to the elite panel. I'm going to speak on ptosis today. I saw ptosis for the first time in Dr. Usha Kim's operation theatre. I have a long association with oculoplasty because my examination was at LVP. Dr. Honavar was the examiner for a case of ptosis in my DNB, while Aditi was the expert there, and Dr. Milind is my much cherished mentor. So on that note, I shall begin my talk today. So the case one is that of a 30-year-old male who had droopy eyelid from birth. As you can see, there's a mild ptosis with full ocular movements, but the story was not complete here. The LPSA was 13 millimeters, but as you can see here, this was a congenital ptosis with a moderate Marcus Van Jauwinking phenomenon. Now, when you have this particular problem, you have to ask the patient whether his concern is predominantly the drooping of eyelid or the Jauwinking. If the patient is bothered about the drooping alone, you can perform a levator resection because it is more physiological than a frontalis sling. To this surgery, while in case the patient needs the Jauwinking, can always be done at a later stage with excision and sling. But if the concern is jaw winking more than drooping, I would suggest go ahead with the levator excision and the sling surgery. Of course, there is some degree of sulcus asymmetry that the patient will have to accept, and the longevity is lesser than that of the former surgery. So this patient was not very bothered about his Marcus gun, underwent levator resection with decent results. Now, this was his 3-month follow-up. As you can see, the lid lag over time reduces, and the skin scar pretty much becomes invisible over time on the upper eyelid. Now, the Marcus gun phenomenon after this surgery, it sort of begins where you have left the eyelid. So the patient might feel it is a little larger when actually it is not. So in any case of congenital ptosis, it's a great idea to look at older photographs and ask the patient for the same and document it. Any child with ptosis, keep monitoring the visual axis vision properly so that you do intervene timely and perform surgery either for vision results or for cosmetic basis. So the case 2 is that of a 55-year-old woman who had drooping of eyelids since 1 year. There was absolutely no other significant history apart from this. Now, on closer examination, I noticed some degree of variability in ptosis. In any case of acquired ptosis, it's a good idea to look at Herring's phenomenon, in which you lift up the ptotic eyelid and observe a droop in the upper eyelid. As is noticed here, approximately the upper eyelid has fallen by 1 to 1.5 millimeter. Any acquired ptosis, spend 2 minutes of your time in OPD, use an ice cube or an ice pack and place it on the ptotic eyelid. So, bingo, just the ice compress itself solved her ptosis. So she was diagnosed as a case of ocular myasthenia and the absence of any systemic features of myasthenia. So in ocular myasthenia, ptosis and diplopia are the common presentations that you notice. An eyes test has very high sensitivity and specificity and does not cost anything. So please do it in all acquired ptosis. Systemic involvement is very common in patients of ocular myasthenia approximately 2 years after onset of ocular problems. So follow up these patients regularly. It's a good idea to treat this patient in collaboration with a neurologist who can further investigate for other problems. And treatment is, of course, with acetylcholinesterase inhibitors and corticosteroids. Case 3 was a very interesting gentleman who came to me with progressive ptosis and a family history of the same. So this is him. He came with severe ptosis and a very prominent chin elevation. So this picture was taken in primary gaze with his chin placed in the normal position. Now the history goes that 22 members in his family on his mother's side across 3 generations have ptosis, which all started in their 40s or 50s. All the patients have variable severity of dysphagia. Men, women, both were affected almost in equal proportion. Now, the story goes that one of their ancestors, a great-grand-uncle or somebody who suffered from severe exposure keratopathy post-ptosis surgery, following which the entire family was scarred. And despite the fact they were scattered across the globe, not one of these 22 members underwent any ptosis surgery. Now, some of the family members who were abroad did undergo some genetic analysis, following which they were diagnosed with oculopharyngeal muscular dystrophy. Now, this is a rare disorder and the onset is commonly in the 4th and 5th decade. It starts with eyelid ptosis, ophthalmoplegia, progressive dysphagia, which could be for solids or liquids. Certain patients can develop proximal limb weakness. Among the genetic inheritance pattern style, the dominant variety is more common than the autosomal recessive variant. Now, all we can offer to the patient is supportive treatment. So the levator action for the right was 7 and the left was 6 millimeters. While traditionally what is recommended is a frontalis sling, the patient was quite well educated, a professor, very intelligent in understanding the pros and cons of all surgery. So we decided to go in for a trial of levator resection with guarded prognosis. The goal was simply to clear the visual axis and get rid of the chin elevation, and also to obtain tissue for histopathological analysis. So here are the photographs of the patient a month after surgery. Around 1 year follow-up of the patient, the right eye has been maintained till date, while the left eye drooped down by 1 millimeter, but he's still able to see in there. And since the orbicularis is affected in this case, you have to monitor the lag of thalamus, which was not very significant in him. So the histopathology of report, which revealed atrophic muscles, fibrotic hyalinoized areas, was sent across to his entire family, who were very excited about it. So in any ptosis, the management plan will depend on history, severity, levator action, Bell's phenomenon, and the lid crease. Bell's phenomenon, look at the Bell's phenomenon in gentle and tight closure. The results could be variable. You could combine these ptosis procedures along with blepharoplasty, lateral canthoplasty, etc. Be aware of cases of pseudotosis, like this patient who actually has a low brow and not ptosis. So the takeaway pointers are that detailed assessment, patient guardian counseling, set feasible goals, under-promise, over-deliver. It's important to learn from your previous cases and follow up with patients, anticipate complications, prepare patient for those much in advance, plan the suture readjustment at the appropriate time when necessary, and photographic documentation at each visit. Thank you so much. I hope I presented something of value today. Thank you again.
Thank you, Anuradha. That was a nice case study. Of course, I have almost 15 of them in one single family. So it was nice to hear. I would now, I would now like to welcome the next speaker, Dr. Aditi Vatve. She is a fellow oculoplasty from LB Prasad Eye Institute, Hyderabad, and she is a founder and consultant of this oculoplasty surgery at Sunaina Oculoplasty Center, Kolhapur. And she is also a consultant of oculoplasty at Aster Aadhar Hospital, Kolhapur. So she has various publications to her credit and has won numerous prizes, and of course very actively takes part in lacrimal workshops these days. So over to you, Aditi, for your talk on reconstruction, basic principles of eyelid reconstruction.
Yeah, thank you. Am I audible and are my slides visible to everyone?
Yes.
Am I audible to everyone?
Yes, Aditi.
Yeah, so I'll start with my presentation. As the quote says, excision with reconstruction in mind is science and art combined, whereas excision without reconstruction is mindless butchery. With this quote, I will start my presentation on basic principles of eyelid reconstruction. The reconstruction starts from the assessment of the lid defect, which includes the assessment of the defect, whether it is full thickness or partial thickness, involvement of margins, canaliculi, any presence of tissue loss, the actual size of the lid defect, and involvement of the levator muscle. So now these kind of lesions where we can just excise the lesion and leave it for secondary intentional healing, there is no reconstruction needed. When these kind of small lesions, we can just shave off the lesion without disturbing the lid margin. But these kind of lesions where the lesion is sitting on the eyelid margin, involving the lash line, and sometimes extend on the posterior lamella also. Sometimes we need to take a decision of pentagon excision with direct closure in order to maintain the lash line continuity. So going to the first simplest eyelid reconstruction of direct closure for small defects, which can arise either from excision of a lesion of the eyelid or post-trauma. So first thing, how should we start repairing the eyelid margin? It starts from repairing the tarsus, and it is not gray line, which is sometimes mentioned in few textbooks or told sometimes. So tarsus is a solid structure which will give support to the wound. So we first enter with a 6-0 MOSVIRCON through the third meibomian orifice on one side, come out equidistant on the other side, re-enter in the first meibomian gland orifice and take it out through the first part of the eyelid and complete the mattress suture. So this is the first margin suture taken through the tarsus. This is also called as 3311 or far near suture, which is the first suture which will give the support, which is followed by closure of the tarsus with absorbable suture, which is generally from the anterior side. 90% of the tarsus is taken in the bite so as to avoid any conjunctival irritation, globe irritation by the conjunctival bite, which is followed by orbicularis and skin closure in layers. So what is the end point which we expect post-operatively is a pouting of the lid margin, which should be seen at the end of the surgery, so that when the wound heals, the lid margin is flat and does not cause complications like ectropion or notch because of the misalignment or because of the depressed scar which can be present in the lid margin. The other complications of this simple procedure may include lagophthalmos, scarring, or symplepharon due to some small conjunctival bite which can be taken into the skin or like lid margin suturing. So these kind of small lesions can be treated with excision with clear margins. Here this lady also had a small sebaceous gland carcinoma which was excised with clear margins. We can give very good cosmetic outcome without much of lash line or lid contour disturbance. So now coming to large upper eyelid defects which are not amenable for direct closure even with canthotomy or cantholysis. Now this old gentleman presented with a lid mass. On eversion there was a pagetoid spread, so clinically this was a sebaceous gland carcinoma, and with clinically clear 4 millimeters margin which were confirmed on frozen section, we had to remove almost 75% of the upper eyelid. So these kind of defects can be repaired with full thickness lid sharing techniques. Here the Cutler beard surgery was done where full thickness flap is borrowed from the lower eyelid, which gives a very good outcome after the stage 2 of the flap division. The only part which is missing is the upper eyelid, which is most of the time is fine with people with if the lid contour and rest of the things is managed well. These kind of lesions, this lady had a chronic blepharitis which turned out to be a sebaceous gland carcinoma, or simple eyelid lesion like this will give a large defect which needs to be addressed with this lid sharing techniques like Cutler beard. Now large eyelid defects with canthus involvement need some extra attention for the canthus formation, which will give a better contour and cosmetic appearance. So with this lady who had a basal cell carcinoma, even the lateral canthus was sacrificed while obtaining 4 millimeters clear margin. So for this lady, we need to address both the lamella, anterior as well as posterior lamella. So posterior lamella was addressed by borrowing a tarsoconjunctival Hughes flap from upper eyelid. The canthal thing was taken care by borrowing periosteal flap forming the lateral canthus. This made the posterior lamella, and anterior lamella was formed by full thickness skin graft. So this was the final appearance, giving a good lid contour as well as the lateral canthus. The only thing missing was lashes because we had borrowed a tarsoconjunctival flap and the lashes were already destroyed with the tumor. So these kind of basal cell carcinomas are actually somewhat bothersome because many of the times we clinically do not expect that most lid margin is involved. So after frozen section control, clear margin, this is the lid defect. The posterior lamella was formed again by Hughes flap. The anterior was formed by ipsilateral monopedicle flap, and the rest of the defect was covered by taking skin from the contralateral upper eyelid. So if you notice, the monopedicle graft actually gives a better contour to the lower lid because of the upper sided traction, the upward traction, and gives better color match. So to conclude, for any eyelid reconstruction, we first need to assess the lid defect. In case of lid tumors, anticipate the defect and plan reconstruction pre-operatively, because we need to obtain a consent even from the graft harvesting site. Be ready with alternate reconstruction plan in case frozen section control surprises us with more of a excision. Replacement of both anterior as well as posterior lamella, very important. Plan reconstructions considering the blood supply of the tissue, and never give graft over a graft. Always combine flap with graft or both flaps. Yeah, that's all. So that if you follow all these principles, you can get very good cosmetic as well as functional outcome post lid reconstruction. Thank you.
Thank you, Aditi, for your nice overview of all leg reconstruction techniques. I now invite Dr. Ravindra Wankhede to give his talk. Dr. Ravindra Wankhede is a dear friend, and he has done his post-graduation from Nagri Eye Research Foundation, Ahmedabad, and he's also done his fellowship in orbit oculoplasty with Dr. Nitin Trivedi at Nagri Eye Hospital. At present, he is the leading oculoplasty and orbit surgeon at Solapur and adjoining areas of Maharashtra.
Am I audible?
Yes, Ravi. Yes. Please.
Yeah.
Please go ahead. Thank you.
Yeah. Good afternoon, everyone, and thank you, MOS, for giving me opportunity to speak. The topic allotted to me is entropion, and I will talk upon few of the basic cases related to general ophthalmologist. A 70-year-old lady presenting to me with irritation in the right eye with foreign body sensation. On examination of the eyelids, there is inwardly turned lower eyelid. There is overriding of the septal fibers of the orbicularis, which is noted on squeezing of the eyelids, I mean on closure. After assessing the horizontal eyelid laxity, the snapback test is positive. That is, the lid doesn't go back immediately after the drag, and the pinch test is positive, that you can hold the eyelid very easily, which is possible when there is excessive lid laxity. So obviously the clinical diagnosis in this case is a senile involutional entropion. The important thing is to be differentiated from other conditions like trichiasis, which is misdirection of the eyelash, dystichiasis, which is an extra row of eyelash, and other types of entropion. The involutional is the most common variety, and the causative factor is dehiscence or disinsertion of the lower lid retractors, or increase in horizontal eyelid laxity, and in ophthalmos because of age-related factors. The various treatment options available were sutures, Jones procedure, Weiss technique, Quickerts, or lateral tarsal strip. The simplest is the Jones procedure, which is application or reattaching the lower eyelid retractors back to the lower border of the tarsal plate. We just take a incision 4 millimeter below the eyelid margin, do a subocularis, get a subocularis plane, dissect till the septum, then incise the septum and retract the fat. And there lies a retractor, which is a white band, generally seen as a white band structure. We just have to hold it and attach it to the lower border of the tarsal plate. On table, you can assess the outward turning of the eyelid. This is one of very easy technique to overcome the senile entropion, where normal anatomy is retained back. The results can be titrated on table, and there are very less chances of recurrence. It can be combined with other surgeries such as lateral tarsal strip for a greater success. These are the results which I could get. The second case is a 60-year-old lady coming with irritation, photophobia, watering in both eyes since 3 to 4 years. On examination of her eyelids, there was difficulty with a digital inversion of both the upper lids. There was a thickened tarsal plate, marked palpebral conjunctival scarring was seen. The multiple tracheitic lashes were seen, and almost about 75% of the meibomian glands were posteriorly displaced. So obviously the clinical diagnosis in this case was severe cicatricial entropion. The various causes in this cicatricial entropion are trachoma, which is very common, chronic allergies, Stevens-Johnson syndrome, ocular cicatricial pemphigoid, and chemical injuries, which are quite common. The basic pathophysiology is scarring and shortening of the posterior lamella, which leads to turning of the lid margin. In turning of the lid margin, the basic management principles are recession of the anterior lamella or lengthening of the posterior lamella, or both. Few of the important surgical options we have is electroepilation, which is done whenever there is just a few of the trachytic lashes. We can go ahead with a tarsal fracture with a marginal rotation, a tarsal wedge resection, or a lamellar separation with or without buccal mucous membrane graft. A few of the important factors affecting the surgical choice are severity of the entropion, thickness of the tarsal plate, if the margin of the tarsus is keratinized, or is there any history of previous surgery. The most very excellent procedure for mild to moderate kind of entropion with adequate tarsus is a tarsal fracture or tarsotomy with tarsal rotation. The basic method is the lid is everted and the incision is taken about 3 mm parallel to the lid margin. Incision is limited to the tarsus and the conjunctiva. The 3 double arm sutures are passed through the proximal cut end of the tarsus to exit through the skin just near the lash line. Once we tighten all these 3 sutures, the lashes are everted. The advantage is it is very simple and effective primary procedure. There is no need of external incision or grafting. One more procedure is tarsal wedge resection. This is preferred whenever there is tarsus is thickened. About 3 mm wedge of full 90% thickness of the tarsal plate is excised. Then the remaining edges of the groove are approximated with a double arm suture, leading to eversion of the lid margin. A few of the simple advantages are there are very less chances of overcorrection in this procedure. The disadvantage is obviously the undercorrection. One more important procedure is lamellar split with lamellar recession, anterior lamellar recession, where what we do is terminal 2 to 3 mm of the tarsal plate is incised and dissected and rotated to recession of anterior lamella. The uncovered part of the tarsus is covered with the buccal mucous membrane, or it can be left just to keratinize. The advantage is it can be done in any severe cicatricial entropion associated with keratinization of the lid margin. The only disadvantage which it can give is it can create a lag of thalmos or a lid retraction. The lady with this cicatricial entropion underwent the lamellar split with buccal mucous membrane grafting, and these were the post-operative results. The third is just a simple and very common case, which is common in day-to-day practice of a general ophthalmologist. About a 1-year-old infant presenting with few eyelashes rubbing to cornea in the right eye. Obviously, the diagnosis is epiblepharon. It's an excessive skin across the lid margin. The differential diagnosis is obviously congenital entropion, which we have to differentiate. In congenital entropion, there is turning of the entire lower eyelid with the lashes, and in epiblepharon, extra horizontal row of the skin there is just an extra row of the skin along the lid margin. Absence or presence of lower lid crease in case of epiblepharon. It resolves spontaneously if wetted, or if not, if creating some corneal irritation, we can go ahead with the HOTS procedure, which is excising the excessive skin and the orbicularis along the subciliary incision. Yes, thank you.
Thank you, Dr. Avi, for an excellent talk where you have covered all the possible surgeries for entropion.
Yeah, thank you.
We would now like to invite Dr. Mangesh Dhobekar, who is again a dear friend who has also done his fellowship from Shankar Netralaya, and he's also the head of oculoplasty at Ganpati Netralaya Jalna, to give his talk. Dr. Mangesh?
Yeah. Just
please share your screen.
Yeah.
Yes, but voice?
Hello.
Yes.
Am I audible?
You're audible.
Yeah. So, good afternoon, all of you. So, I'll be talking on the clinical pulse in the management of eyelid injury based on the interesting case scenarios. I will take you through the 10 clinical points which are very important in the management of lead injuries. The first one is always rule out the associated serious injuries. If there is any associated serious systemic trauma, upper globe injuries, traumatic optic neuropathies, then it's very important to rule out all those injuries because the lead injury is not that important as the intraocular or the systemic lead injuries, and these injuries take precedence over the lead injuries. As far as the lead injuries are concerned, I assume that it's present in all cases unless proven otherwise. So it's very important to rule out the canalicular tear in all injuries. For example, the blouse hook injury, we have to actually insist and we have to convince in a very hard way to the parents that there is a canalicular involvement. It can involve upper, lower lid or any part of the lacrimal system. Always rule out the deeper lead injuries like it may involve even a LPS. It can have a retained deeper foreign bodies based on the mode of injury. So you have to rule out all the deeper lead injuries in all cases. Next thing is that we have to actually rule out whether there is a tissue loss or not. Because tissue loss is very rare in the eyelid injuries, and orbicularis contraction of the lead tear can give pseudo appearance of the tissue loss. Is there any role of imaging in the eyelid injuries? Yes, is there. In some of the adnexal injuries like the suspected orbital fractures, retained orbital foreign bodies, traumatic optic neuropathy, occult globe ruptures, then CT orbits remains the first investigation of choice. This patient presented only with the lateral canthal lead tear, but the patient had globe luxation into the ethmoidal sinus, which was revealed on imaging. Infection prophylaxis is very important basically in a dog bite injury when you have to use the anti-rabies vaccine as well as the anti-rabies serum. Nowadays we use monoclonal antibodies in the dose of 3.3 international units per kg body weight. Proper and detailed documentation, because this is the case in which I was called in court of law for the expert witness. So it's very important that you go ahead with the history, detailed examination findings and the photographs in all of the lead injury cases. Isolated lead tear is not an emergency unless there is a tissue loss and there is a preserved amputated tissue. Then you have to go to the operating room as soon as possible. Otherwise, you can safely wait for 2 to 3 days. Non-surgical management, especially with the strips and glue in young children only when there are clean lacerations which are parallel to the relaxing skin tension lines. You can use derma-bound tissue adhesives or the sterile strips, no financial interest. Proper wound decontamination is very important before going ahead with any surgical procedures. You can use the normal saline with the pressure. Removal of the particulate foreign bodies and the dust particle is very important because they can give rise to the pigmentation in the wound later on. You can use the saline sedative which is attached to the suction catheter. It acts as a very efficient debrider for most of the cases, and Bitarian of course for the 30 seconds. Surgical management of the lead injury, it's a puzzle, so you have to identify the landmarks and solid. This was a case in which you were not able to identify the upper lid structures, and then you have to underline the deeper injuries like the LPS. You have to align the brow part, you have to align the lead margin, and then only you will get the gratifying results in most of the cases after solving the difficult puzzle. As Aditya already informed and instructed that the lead margin, keep it everted. This is very important. I will not talk upon that. Coming to the canalicular tear, minimonoca is the answer. It's a very simple, minimally invasive, and the efficient procedure. Doesn't matter whether a single canaliculus is involved, like in this case, or it might be a 2 canalicular involved in case of the medial canthal avulsion, and I do not go ahead with a bicanalicular intubation which needs additional nasal manipulation in all such cases. Coming to the tissue loss, you have to, I have already stated that you have to actually see if there is any tissue loss, because this was the case in which we were assuming that the upper lid is totally lost after a road traffic accident in this baby. And after exploring in the infraorbital area and the retroorbital area, we could found that there is a upper lid which was down there behind the globe. And so we could retrieve it and attach it to the normal position with the gratifying results. If there is a tissue loss, then you have to follow the principles of the lead reconstruction as Adi told. Coming to this scarring part, it actually starts with the first stitch itself. It needs proper closure of the wound without tension, avoiding infection and wound breaking. The wound is very important. Antibiotic steroid ointment I usually start after a week onwards. In selected cases, triamcinolone or 5-fluorouracil injection periregional. Otherwise, you have to go ahead and you have to resort to the revision surgery in selected cases. This is the late presentation, medial canthal scarring. You have to go ahead with the J-plasty based on the cases, or you have to go ahead with the full thickness skin graft based on the cases. So this is the last case, and this is the case I had been quarantined for 2 weeks. This was a case of a polytrauma which was referred for the lid tear repair from elsewhere. Patient had other non-serious systemic injuries, so the patient was first referred here for the lid tear management, and I did, but it was in the initial days of the lockdown, and we were about to not to do the longer surgeries, and I managed this case, but still it took me 2.5 hours. But there was a total lower lid loss. But I didn't want to go ahead with the longer surgery in this case. So I plan to go ahead with the lower lid construction in the next setting, because the globe and the cornea is very well protected. So these are the very important clinical points as far as the management of lid injuries are concerned. Thank you all of you for the kind listening.
Thank you, Mangesh, for a wonderful elaborative talk and highlighting almost all the important points in eyelid injuries. I now welcome for Dr. Pallavi Khedkar for her last talk of this session, which is on custom-made ocular prosthesis, the soul of socket reconstruction. Dr. Pallavi Khedkar is a PG from Shankara Netralaya and has done her fellowship from IA
Is like anophthalmia, microphthalmia in a younger age or an adult patient can present. Acquired deformities like phthisis bulbi, post-evisceration, which we do for anterior staphylomas or a painful blind eye, or post-traumatic. Post-enucleation for a malignancy, post-chemical injuries, contracted socket, post-radiation, or a post-orbital accentration. There is an entity, if after enucleation, if the socket is left empty, it causes a socket contracture, which is called post-enucleation socket syndrome, in which we see enophthalmos, upper eyelid sulcus deformity, laxity of the lower lid, ptosis, or a backward tilt of the ocular prosthesis. So how do we go ahead? What are the types of prosthesis? They are ocular and orbital. In orbital, spectacle mounted prosthesis and adhesive retained prosthesis. So what are the stock eye and customized ocular prosthesis? The stock eye is the production is a mass production. It can rehabilitate cosmetically in post-evisceration or a post-enucleated patient. It gives an artificial look. Patient may complain of discharge, discomfort, or watering. It has restricted wearing period. Patient has to remove the stalk eye in between, and it has restricted or no movement. And in the long run, the lower eyelid will sag due to the weight of the stalk eye, and it needs a surgical correction. So why is there discharge and sagging? Note the gap between the stalk eye and the ready-made stalk or ready-made eye and the implant or a surface of the socket. There is a gap between that, so there is a discharge, and also the stalk eye is resting on the superior and inferior fornices, which leads to sagging of the lower lid. And in custom made artificial eye, it is snugly fitted with the socket surface, so there is a less chances of discharge collection, and in addition it gives a greater area for movements. So these are the differences. There is no, there is need for removal of the stalk eye in the night, and there is no removal of the customized or artificial shell in the night, or even during the week or a month. So a patient can remove it only for cleaning and immediately fit in the socket. So these are the results. Few patients who are using stalk eye, we can see a squinting appearance. Here is the ill fitting of the stalk eye. Then here there is a color mismatch. So these are the few disadvantages of stalk eye, and these are the results of a customized ocular prosthesis. And at any age, every age, we can see a beautiful outcome, which gives a natural look to the facial features. So this is one patient where we can see the difference. Here the volume loss is seen, that the inophthalmos can be noted. And with the customized ocular prosthesis, we can see a good matching with the contralateral eye. Even in this case, we can see a lid retraction. There is no color matching. Even the iris color is different with the stalk eye. And in the customized ocular prosthesis, everything is matched so well. So how do we make the customized ocular prosthesis? It is definitely a painstaking and technically demanding process. It is a skillful, trained job. It takes time to prepare, at least 1 to 2 days for one single patient. So how do you go ahead with that? It is clean the debris and secretions from the anophthalmic socket, and an impression socket was made with an irreversible hydrocolloid material after you put it in the custom impression tray. Once you take the impression is set, then remove the set impression from the socket, look for accuracy, look for the edges of the impression, and then later it is boxed in a dental stone. And the set cast is retrieved after putting the molten wax on the dental stone. So the wax pattern is removed and it is carved to achieve an arbitrary contour of the eye. Then check the fitting of this wax mold and remove all the extensions in the fornices. Then contour is adjusted to match it with the contralateral eye. The iris location is marked on the wax mold, and in both the gazes when patient is gaze is fixed, and also in all the gazes. The color matched iris is retrieved from the stalk and it is incorporated in the wax mold. Then the wax mold is finally tried in the patient and assessed in terms of fit, accuracy in fixed gaze, as well as functional movements. So the prepared wax pattern is again invested in the special ocular flask and de-waxed. After all these, heat cured tooth colored PMMA is mixed and packed in the de-waxed mold cavity. The red nylon fibers are used for characterization of the vessels on the sclera, which actually gives a good color matching with the contralateral eye. And this flask is again cured for 1 hour in a boiling water, and cured pattern is retrieved from the flask. And then you do the finishing, like polishing, then painting, and after all this, a final coat is given and polishing is done. So now the whole custom ocular prosthesis is ready. This finished prosthesis is disinfected and inserted into the ocular socket and assessed for the fit again. Patient is instructed to wet the prosthesis before insertion. It can be worn in the day and night with regular cleaning of the prosthesis. So in few cases, there is definitely a good role of congenital customized ocular prosthesis, like these congenital microphthalmos cases. After a surgical process of fornix deepening sutures, a good customized ocular prosthesis can be fit in where the stalk eye has no role. In anophthalmia, conservatively, we can expand the socket with regular and expanding conformer sizes, and finally we can give this result to the patient. But if these patients can be treated in a younger age, we can see such a good result with regular expanding conformers, and at the end of 2 years this boy is having almost similar looking contralateral eye on the face. So it needs timely intervention as early as possible. Regular customized conformers help in growth of the orbit comparable to the contralateral eye. Sometimes it needs orbital expanders along with these conformers, and definitely a stitch in time saves 9 has a promising result. We can see in both these patients, if this lady would have gone for early treatment, definitely she would have had a comparable this eye to her normal eye. So if this is also not possible, what we can go for is use of illusion. If we give a hyperopic correction to this artificial eye and have the maximized look of this artificial eye and achieve the good comparable size of this eye. Thank you.
Yeah, thank you, Pallavi, for a wonderful lecture. And of course, when we think of aesthetics, this part is really very important. Well, any questions for our speakers?
Madam, one question for Hunavar sir. Hunavar sir, are you there?
Yeah.
I also wish to thank
Hunavar
Professor Sunaina Malik Madam for having joined this session. She's the president of MOS and she's an outstanding work. Madam, we wish we could have seen you in person. Thank you, Madam, for joining in.
I also wish, but I am no longer president of MOS.
I know.
I handed over.
Till yesterday. Till yesterday.
I'm immediate past president now.
Yes, madam. Yes. Thank you, madam.
But Pam is always interested in oculoplasty.
Yes, it's
Yeah,
one of my
she's
favorite
always-
subjects.
Hunavar sir, one quick question. I want you to share your experience about the use of topical medication like imiquimod or vismodegib for basal cell carcinomas around the eyelids.
Well, vismodegib is an oral medication. It's a monoclonal antibody against hedgehog pathway inhibitor. It's a hedgehog pathway inhibitor. It's mainly useful in syndromic variant of basal cell carcinoma, but it's also currently used for inoperable patients where surgery may not be feasible because of systemic conditions or the lesion may be very large to shrink it. So that is one. Sonodegib is the other alternative. Vismodegib and sonodegib are the two drugs. Imiquimod is a topical applicant. If you have a very superficial basal cell carcinoma, then you can use imiquimod. So it's not useful for eyelid lesions particularly, periocular lesions possibly.
Yes, thank you, sir.
Any criteria like how big the lesion is, so you use that, something like that?
But, basal cell carcinoma is a surgical disease. So if surgery is not possible, or following surgery, if a patient has a small residual lesion or if has a small recurrence, then medical treatment such as imiquimod may be useful. But basically it's a surgical disease, and inoperable cases of multifocal basal cell carcinomas are treated with monoclonal antibodies.
Yeah, because I've seen some of my skin colleagues using imiquimod in periocular skin basal cell carcinomas.
Right. So if it's very superficial, it works. If it's not superficial, then it will keep progressing.
Okay, thank you, sir. One more question for Aditi. When you can use rotational grafts, when will you prefer free grafts?
Yeah, personally, whenever I can actually use advancement or rotational grafts, I will flaps. I will prefer that because it will have its own blood supply. It will give a very good like matching texture and color is more in those flaps than free grafts. But whenever local advancement or rotation is not possible, I will prefer taking a graft. That too, if it is possible from the ipsilateral or contralateral eyelid, which will have a best color and texture matching. I will prefer that. Sometimes patient is also happy to get a free blepharoplasty surgery in addition to the reconstruction sometimes. Maybe Dr. Amin can also add his points in this. Dr. Milind.
Dr. Milind, you would like to add?
Yeah, no, I think I agree with what Aditi was saying. I was just listening to her comments.
Dr. Honavar?
Sir, unmute yourself.
Okay.
The question, what is the question?
Well, when do you use free grafts in place of rotational grafts?
Well, there are very few occasions when you want to use a full thickness free graft. Most of the time we're able to do with a rotational graft. But if we have to use full thickness free graft, we have to be ready for failure as well. So it's always better to use a lamellar free graft rather than a full thickness free graft. Lamellar free graft in the sense that tarsoconjunctiva can be easily replaced by tarsus and conjunctiva of the upper eyelid if you're reconstructing the lower eyelid or contralateral upper eyelid. That's possible. Skin can be replaced. When you want to replace both the lamina, then it becomes high risk for failure.
Yeah. Thank you, sir. There's one more question for Dr. Ravindra. In case of failure of an entropion or a recurrence of an entropion, how do you go ahead?
Depending on the type of entropion, I had few of the senile entropions which recurred, and then I redid my Jones procedure in that case, and along with the lateral tarsal strip, it worked well. If I reduce the laxity, it works better. And in case of tarsal wedge or tarsal fracture, then better to go ahead with lamellar suppression with buccal mucous membrane. That is what is my experiences.
Okay. So I think we are finished with the questions now. So it was a wonderful session with all the stalwarts with us. Thank you so much. And we missed OPI session this year. And as we were planning to do our midterm, and hope so we postpone it for further 1 year. I think so. So let's wish that everybody is safe. Nice to see you, Milind sir. Happy
Thank you,
and
everyone.
hearty.
It was a wonderful session.
Thank you so much.
Thank you
Thank
so
you,
much, all of you.
Thank you, everyone.
On behalf of
Thank you,
MOS,
madam.
I'd like to thank all of you. Thank you,
Thank you,
for
ma'am.
being with
Thank
us,
you.
for joining us.
Thank you.
Thank you. It was
Always
nice.
a pleasure having you. Thank you. Bye.
Thank you.
Bye.